Hello everyone, this is Dr. Priyanka Sachdev and I welcome you all for second Prof Final Year, this 75-hour rapid revision course. It's a rapid revision course to ace your university exams. Before starting, I would like to address few FAQs which I've frequently received on chats.
I haven't read anything. I did terribly in my pre university exams. Can FG save me?
FG is exactly for you. It is for the student who are starting late, starting scared, or starting after a bad pre-university result. But a bad pre-university result couldn't predict your university result. What predict is what you are actually doing between PUs and Us. FG is a system, and the system works regardless of the starting point. Distinction is the target, and we go for it together.
I've already read the entire Robbins. What will FG add for me?
Everything, because knowing is not equal to scoring. Reading Robbins and revising Robbins are two completely different things. You have knowledge, but you need a system that organize it, integrate it, and convert into exam ready output.
What is integration?
It is a technique to organize similar systems across patho, micro, and pharma. So we have three subjects, that is patho, micro, pharma, and we have 9 major systems in human body. You are knowing that. So I don't teach you subject wise. I teach you system wise. We take system one by one and we cover that system from all three subjects one by one. For example, day 7 is our CVS day. So on that day, you have to complete watching CVS diseases and pathology, CVS infections in microbiology, and CVS drugs and pharmacology. So it is for better retention, better understanding.
Your course is fast paced. How will I keep up in so little time?
The workbooks. That's the answer. It is not a textbook. It is not a printout. It is a strategic companion designed by me. On every page, you can see 80% is filled and 20% is intentionally blank. The blanks you have to fill during the session in real time so that revision goes on simultaneously. So by the end of all session, it will be no more workbook. It is converted into revision copy. The only revision copy you have to revise before your university exam. And the biggest catch is that the videos and the workbooks are 100% synchronized. Slide to slide, page to page. Whatever I'm teaching you in the videos, word to word, it is matching with the workbook. And we have done page stamping on the videos for easy navigation into the workbook. But if some students feels that they want 100% filled notes, they do not want to fill the workbooks. So the Prof Buster hard copy books for pathology and microbiology, they are already available on our website. You can go with that. It's 100% filled version of the workbook. It's already available. And along with the microbiology, master charts are also available. Master chart is a calendar. You can hang it on your wall, you can flip it, and you can compress the entire microbiology in just 50 pages.
I always start strong and then fall off schedule. What's different this time?
This time, you don't rely on willpower alone. You make public commitment. I dare you to take 16-day FG challenge. It's not just a timetable, it's a social contract. So, there are total 49 chapters in Patho, Micro, and Pharma. I have completed the 49 chapters in 75-hour lectures. You cannot watch 75-hour lectures in one go. That's why I divided the 75-hour lectures in 16 days. I have already decided that what you have to watch on what day. From day 1 to day 16, the complete schedule is in your hand. You can see one system, one day. For example, on day 9, it's respiratory system. So you have to finish watching respiratory disorders in pathology, respiratory microorganisms in microbiology, and the respiratory drugs and pharmacology. Likewise, one system, one day, the 16-day go, the complete syllabus, the 49 chapters in just 75 hours. So you already know what you have to watch today, what you have to read today, and that's the 16-day challenge. Every day you finish your target, every day you tag me on Insta, daily, no hiding. When the entire FG community is watching, showing up feels different. So make a commitment, show up, tag me. Let's go. The 16-day challenge.
I understood what FG is. But, ma'am, can you please tell the exact steps to be followed?
There are 7 steps to be repeated 16 times for 16 days in a loop. Step number 1. Plan the next day the night before. It will take just 60 seconds but save your 60 minutes in the morning. Step number 2. Declare it publicly. Make a social contract. Make a public commitment. Step number 3. Start your day with coffee, tea, stay hydrated, and start watching the videos dedicated for that particular day in that folder with a timer. Don't forget to put the timer. Step number 4, fill the workbook simultaneously. Simultaneously means simultaneously. It should not be later, it should not be tonight, it should be simultaneously. Step number 5, after watching the video and filling the workbook simultaneously, there are two type of quiz we have attached with every video, the university exam quiz and the MCQ quiz. You have to solve both the quiz immediately after watching the video and filling the workbook to analyze yourself. Step number 6, track your progress either on the physical tracker, we are sending this to you, or the digital tracker which is already there on the app. You have to make a tick mark to analyze yourself and track your progress. Step number 7, analyze, reset, repeat for 16 days. If you are following these 7 steps for 16 days, take my word, the distinction is yours.
I am having my exams after 6 months. Is FG for me?
No, not useful now. You can enroll for Prof Buster comprehensive batch. You have time, you understand the concepts in detail. FG might be useful just before the exams.
I am an intern preparing for NEET PG. Is FG good for me?
No, FG is exclusively for second Prof university exams. Concept for both the exams might be same, but approach differs.
Is FG enough for university exam?
Yes, so 100% coverage of all LAQs and SAQs of all universities, pan India. So 100% strike rate in just 75 hours. So, are you ready? Today we are at day 10 of 16-day second Prof FG challenge. So, what's your today's target? What you have to finish watching today only? So, let's trace on the day 10 to say today's target. Starting from day 1, you can see we are proceeding towards the day 10. Day 10 is integrated GIT, integrated gastrointestinal system. You have to finish GIT from patho, GIT from micro, and GIT from pharma. So in patho, you have to finish watching GIT disorders. I hope you have already watched it. In micro, today I will teach you gastrointestinal infections, and in pharmacology, gastrointestinal drugs. So today day is integrated GITD, and the total duration of all this is the 6 to 8 hours, and you have to finish it watching it today only. There should be no pending. You have to watch it today only to complete the 16-day challenge. So this session is microbiology, in which we are studying. I'm going to teach you gastrointestinal infections in one shot. Likewise, we are going to continue till day 16 to make this challenge successful, right? So this session is microbiology session, in which I am teaching you gastrointestinal infection, in which there are total 30 organisms. Out of the 30 organisms, 13 are bacteria, 1 is virus, and 16 are parasite. The 16 parasites are there. Okay, so I'm going to teach you all 30 organisms in 2 parts. In this video, I will be teaching you 24 out of them. So I will be teaching you except 6 parasites, 6 nematodes parasite. I will teach you remaining all organisms in this session in one shot. So these are very important from a university exam point of view. You get many long question and short question from these 30 or maybe these 24 organisms. Have a look on them. Have a look on the LAQs, SAQs, which are already asked in the previous 5-year university exams. See the LAQs, see the LAQs, see the SAQs, see the SAQs. You can see how important they are. See very short questions in front of you. How many questions comes from these topics in your university exam, especially long, short, very short question. All these questions are already uploaded on the app, university wise as well as topic wise. So whatever comfortable to you, please go through these questions once you finish watching this session. Once you finish watching this session, there are 2 quiz attached with this video. One is university quiz and one is MCQ quiz. I want each one of you to watch the video, to make the notes in the workbook, and after that solve these 2 quiz and analyze yourself and let me know what is your analysis, how much you are confident in this topic. So let's start it. The gastrointestinal tract infections in microbiology. Let's start the systemic microbiology. General microbiology we have already done. Systemic microbiology we have started. We know in human body, in microbiology, we have divided the human body into 8 systems. You can appreciate the 8 systems in front of you. These are our 8 systems. 8 systems, 8 color coding. This is my color coding. It is not universal color coding. You can use any color of your choice. Okay. I have allotted 8 colors, unique color to each system. Okay, so that we can make master chart using that color coding. Okay, we have already covered the red, that is CVS. We have already covered the orange, that is respiratory tract, and today's color is purple. So today is purple day. I will cover all the organisms. I will cover basically 30 organisms today, the purple organisms. The organisms, bacteria, virus, parasite, and fungus, which cause GIT infection. So can we start? Can we start? So today's day is purple day. Okay. Now in each of these systems, we divide the system into 4 categories. The bacteria, which are causing infection in that system, the viruses, the parasite, and the fungus, which cause infection in that system. So that's what we are doing with GIT also. So the first and foremost step before we all start GIT, you must know the organisms, the 30 organisms which cause infection in GIT. They are in front of you. You can see total 30 organisms. So today's target is 30 and we will finish it. Okay. So you can see the 30 organisms. I'm dividing it into 4 categories. You can see these are the bacteria. I will teach you all these bacteria, 13 bacteria today. I will teach you all in this sequence. Okay. You can see virus, single virus, only one virus. I will teach you this virus. You can see parasites. I will teach you all these parasites. The biggest chunk today is parasite. Okay. So I will teach you all the parasites. Nearly 16 parasites are there. We don't have fungus, thankfully. We don't have any fungal infection in the GIT. So here there is a cross. You can see here there is a cross. So you can appreciate whatever is there. The same thing is over here. So hang it on your wall, your room wall. Hang this calendar and have a look on that. Aate, jate, whenever you are eating, coming to the room, going back. So have a look. It will fit in your mind. Okay, so this is the complete GIT infection. Okay, now we will start the organisms. Before I start the organism, now you all are doctors. Whenever any patient comes to you with GIT complaints, what are the GIT complaints? Patient comes with abdominal pain, patient comes with vomiting, patient comes with diarrhea or dysentery. I assume you understand the difference between diarrhea and dysentery. Diarrhea is simple loose motion, increased frequency of motion and loose motion. So that is diarrhea. But if the motion contains blood also, it is known as dysentery. So if the patient complains of diarrhea, patient complains of dysentery, patient complains of vomiting, patient complains of abdominal pain, and you are suspecting infectious etiology, with these symptoms, you are doctor, with these symptoms, immediately 30 differentials should come in your mind. So first of all, you should learn these 30 differentials. These are the 30 differentials. It should come in your mind. Okay, out of these 30 differentials, 13 are bacteria, 1 is virus, and 16 is parasite. So we are going to cover all these 30 organisms today only. In a comparative way, we will cover. Okay, I will compact these 30 organisms in just 8 pages. 8 pages of my master chart. 8 pages, you can count, total 8 pages. If you see the textbook, any standard textbook, this chapter is 80 to 100 pages. Okay, so I am converting 100 pages into 8 pages. And without skipping any important information that can be asked in your exam, without skipping, mark my words. Okay, so this is a way you should study. So total 8 master charts we will make, 3 of parasite, 13 bacteria will be compacted in 3 master charts, 1 virus for 1, and 16 parasites will be compacted in 4 master charts. So total 8 master charts we will make. So with this overview, now before starting the description of each organism, I would like to give you a bird's eye view. Okay, so that we can read the name of these 30 organisms and start their description. Okay, so let's start it. Let me give you a bird's eye view and let's start the session. Okay, now see the bacteria. First of all, have a look on the bacteria, so 13 bacteria. So see, I have divided into 3 categories. These 4 bacteria cause food poisoning. So first of all, I will start my session today with the bacteria causing food poisoning. Have a look on the 4 bacteria causing food poisoning. So I will start with Staph aureus, okay, followed by Bacillus, Clostridium perfringens and Clostridium botulinum. So I will teach you 2 Clostridium over here, perfringens and botulinum, along with Staph aureus and Bacillus. These 4 cause food poisoning. Okay, the next set of the bacteria, these 4 bacteria, they don't cause food poisoning. They cause gastroenteritis and other infection, but not the food poisoning exactly, but they all belong to Enterobacteriaceae family. So these are the bacteria from the Enterobacteriaceae family. You know the complete Enterobacteriaceae family? Yesterday also I taught you. So it's E. coli, Shigella, non-typhoidal salmonella. By the way, typhoidal salmonella I taught you in CVS. My first bacteria in CVS on YouTube I taught you. I guess you have attended. So typhoidal salmonella is covered there. Here I will cover non-typhoidal salmonella. I will tell you which organisms will come here. And Yersinia anthropolitica and Yersinia pseudotuberculosis. Mind it, it's not Yersinia pestis. Okay, Yersinia pestis is a zoonotic disease. It's not covered over here. Okay, but Yersinia enteropolitica and Yersinia pseudotuberculosis we are covering. So these are the 4 organisms which belong to Enterobacteriaceae. So first we will see these 4, then we will see these 4, and after that we will come on the remaining gram-negative bacteria, Vibrio cholerae, H. pylori, Campylobacter jejuni, and Clostridium difficile. See, this is the 3rd Clostridium. So I will teach you 3 Clostridium. The first 2 were coming over here, Perfringens and Botulinum, and the 3rd one is coming here. You may be thinking, ma'am, why you are not teaching the 3 Clostridium together? No, I can't. Okay, because these 2 cause food poisoning. So I have to take them over here with the organisms causing food poisoning, and this one causes simple infection, not the food poisoning. So I have to take it separately. Okay, so these are the bacteria, the biggest chunk for today. Virus is only one, rotavirus. I will cover that quickly. Only one small virus, rotavirus. Just in 2 minutes we will cover that. After that we will come on parasites, the big chunk, parasites. In parasites we divide them into 4 categories: protozoa and metazoa. Protozoa, metazoa. Metazoa is helminth. We have cystode, trematode, and nematode. Okay. So protozoa, cystode, trematode, nematode. You can see these 3 protozoa I will teach you. In the metazoa, these are the cystodes, these are trematodes, and these are nematodes. I will read their names when I will teach you. Okay, so let's start it. This was an overview, bird's eye view. It was necessary. You can understand my pattern. And now we will run quickly. So can we start? How many of you with me? Everyone give me a thumbs up. Everyone. Can we start? Chalo. We will run now. Let's start my first organism. I will divide my session into 3 parts. First, I will teach you the bacterial infections of GIT. That will take 2 hours or maybe more than that, because we have to cover 13 big bacteria over here. Then my second part of the session will be just of 5 minutes. Viral infections of GIT. I will teach you 1 virus, rotavirus. That is hardly 5 minutes. So 2 hours over here or 2.5 hours over here. Okay, maybe 5-10 minutes over here. Then we will come on parasites. The number is 16, but they are comparative. So again 2 hours over here. So total session duration will be 4.5 hours to 5 hours. Okay, so this is my the complete overview. And fungus, we don't have fungus over here. So these all organisms we will take in this sequence. So first of all, I am teaching you the bacteria, the bacterial infections of GIT. Let's start it. You can see the 13 names. First of all, you have to learn these 13 names and divide them into 3 categories as I have done. You can see I'm dividing them into 3 categories. What 3 categories I have divided them? These 4 cause food poisoning. We'll make one master chart for them. These 5 are the members of Enterobacteriaceae. We will make one master chart of them. We will take the 2 Yersinia together in one column only. And lastly, the others, we will take them together. So total 13 bacteria we will cover. Okay, now I cannot start their details directly unless and until we revise the classification of bacteria. You know my pattern. Okay, so this classification, I guess everyone know. Those who ever attended even my single class, you know this classification? Yes or no? We divide the bacteria into 4 categories: GPC, GNC, GPB, GNB. Total 60 bacteria in our syllabus. Yes, I will teach you all 60. But today I will not teach you 60. Today I will teach you 13 out of 60. 13 out of 60. Yes or no? My purple 13. So where are my purple 13? The 13 bacteria which cause GIT infection. Where are those? First you highlight those over here. So you come to know that out of these 13, how many are coming in each category? Okay, so you can see these are my 13 bacteria, you can see, and I'm highlighting them over here. So you can see the highlight. You can count if you wish. Total 13 bacteria. You can see how many of them are gram-positive cocci and none of them actually. How many of them are gram-negative cocci? Oh, gram-positive cocci, one of them is there, Steph Aureus. Gram-negative cocci is none. Some of them are gram-positive bacilli and most of them are gram-negative bacilli. So you should have an overview. The 13 bacteria which I'm going to teach you, each one of them is coming in which category. That is the basic. Now see this classification. See this page. Everyone have a look and see this page. Okay, both are same. Here it is not color coded, and here I have given 8 colors for 8 systems. Search for the purple. Today's color is purple. At the same location where I have circled them, you can see the same location at the purple. Okay, you can count. There are 13 purple. You can make a count if you wish. So total 13 purples are there, and you can see in this master chart. Okay, again and again I am showing you so that you can hang it on your wall and you can understand the importance of that. You can see the same classification. What I'm showing you here, what is shown here, and you can see the purple over here. You can see the purple over here. So total 13 purple bacteria I'm going to teach you today, the purple one. So let's start it. It was an overview. Let's start. Let's start The first category, the bacteria which cause food poisoning. Can you understand food poisoning? The bacteria cause food poisoning unless and until you don't know what is food poisoning. So first you tell me what is food poisoning. नाम में ही उसका मतलब है. The name itself depicts its meaning. Food poisoning. So what do you mean by it? So I will start these bacteria. I will start filling with you. But first you tell me what is food poisoning, people. What is food poisoning? Food poisoning is an illness. It's an illness which occurs after consumption of contaminated food and water. So someone is consuming contaminated food and water, and after consuming, okay, this food and water either contain bacteria or the toxin secreted by the bacteria. Okay, one of them. The food contain either bacteria itself or the toxin secreted by, so it is contaminated. Whether it contain bacteria or whether it contain toxin secreted by the bacteria, I will say the food is contaminated, the water is contaminated, and the person is consuming it. And after consuming, person have three possibilities. The person have diarrhea, the person have vomiting, or person have both. In some food poisoning, only diarrhea is there. In some food poisoning, only vomiting is there. And in some food poisoning, both are there. So I will teach you 4 food poisoning. I will let you know in which of them which occur, and you have to mind it. You have to mind it. Okay. So like in Staph aureus, both occur. Okay. Bacillus cereus, both occur. Okay. Here only diarrhea occur. Here only vomiting occur. So you have to mind it, and you have to write down in the clinical feature what I am saying. So this is food poisoning after consumption. Now the most important feature here in the food poisoning is the incubation period. You will say, what do you mean by incubation period? क्या होता है भैया? Boss, what is incubation period? Incubation period is the duration after consumption of the food, after how much time the person presents with symptom. Suppose I am consuming the food now. So after how many hours I am having diarrhea and vomiting? That is the incubation period. So based on incubation period, we divide the food poisoning into 3 categories. You can see the 3 categories. We divide the food poisoning into 3 categories. 1 to 6 hours, 6 to 16 hours, more than 16 hours. In 1 to 6, we have only 2 organisms. Okay, I will teach you these both. I will teach you Staph aureus, Bacillus cereus. In 6 to 16, again, we have 2 organisms. I will teach you both. It is Clostridium perfringens and Bacillus cereus. Bacillus cereus coming in both. You will say, ma'am, how Bacillus cereus coming in both? Suppose I am eating the food which contain Bacillus cereus. So will I have symptom within 6 hours? Or will I have symptoms within 6 to 16? I will explain. I will explain when I will come on Bacillus cereus. But it is the same Bacillus cereus. It is causing 2 types of food poisoning, emetic type and diarrheogenic type. I will explain both. And remaining all will come here. So I will teach you this 2. I will teach you this 2. Actually, this one is the same. So this is my first bacteria. This is my second bacteria. This is my third bacteria. And from here, I will teach you only one. This one. Okay. I will teach you only one. This one. So this is my fourth bacteria. But you should know this classification. So this is how I'm going to teach you these 4 bacteria. Okay. So you should know their incubation period. This one caused food poisoning between 1 to 6 hours. This one between 1 to 6 or 6 to 16, both. It caused 2 types of food poisoning. This one caused 6 to 16 and this one more than 16. So we have already seen their incubation period. Now all these food poisoning have their food predilection. Okay, different organisms love different food. Okay, so like some occur in the dairy products, some occur in the meat, some in Chinese fried rice. So you have to learn the food predilection for all of them, and I will teach you food predilection for these food. So that was the overview of food poisoning. Let's start it. Can I start? I'm inaugurating and cutting the ribbon out of 30. Let's start my first bacteria. Can I start? Are you people with me? Let's start Staph aureus quickly. Okay, yes. So let's start Staph aureus. Okay, Staph aureus. Introduction. You know my fixed set of heading. In introduction, I mostly tell you the scientist. Yesterday also I taught you, I guess, this. Staphylococcus is named by Alexander. Alexander Oxton named the term Staphylococcus. Okay, and Rosenberg named Staph aureus. This is complete Staphylococcus, and it is one of the Staphylococcus Staph aureus. So learn the 2 scientists, Alexander and Rosenberg. Alexander named Staphylococcus, Rosenberg named Staphylococcus aureus, the main hero over here. So this is the introduction. Okay, in the introduction, you should know the classification. Tell me the classification. I am teaching you Staph aureus, but how many type of Staph is there? Staph aureus is one of the Staph, one main hero. But how many type of Staph is there? So see first classification of Staph. Where Staph aureus is coming, then only I can proceed. How many gram-positive cocci is there? I guess yes, this I told you yesterday also. We have 2 gram-positive cocci, Staphylococcus and Streptococcus. You can see gram-positive cocci are of 2 types. Okay. Can you differentiate them based on their shape? No, both are cocci. Can you differentiate them based on their color? No, both are gram-positive violet color. So how to differentiate them? We can differentiate them based on their arrangement. Appreciate the arrangement. So Staphylococcus are arranged in clusters and Streptococcus are arranged in chains. It is arranged in chains. Today I am teaching you Staphylococcus. So with this, you can see this is clusters, this is chains. So based on the arrangement we can differentiate them, but I need a good test to differentiate them. Based on the arrangement I can do so. This is cluster, this is chain, but I need a test. What was the test I told you yesterday also? Tell me a test which differentiate the two. The name of the test is catalase test. Okay, catalase test differentiates Staphylococcus from the Streptococcus. It's a good test to differentiate. Okay, it's an objective test to differentiate the two. Okay, so catalase test differentiates step from strep. It is positive in step and it is negative in strep. You can see here bubbles are coming. So this is positive in step. Here no bubbles are coming. So it is negative in strep. Okay, you remember what's the principle? Yesterday I taught you, quickly we will revise. See the diagram of the two bacteria. You can see step and strep. God has made them like this. God, the great God. God has made them like this only. Not we humans. God has made them like this. My point is that all Staphylococcus have catalase enzyme in their cytoplasm. God has given them. Okay, it is inbuilt in them. God has given them. But Streptococcus, none of the Streptococcus God has given this enzyme. Okay, so all Staphylococcus have catalase enzyme. Catalase is the enzyme, by the way. And Streptococcus, none of the Streptococcus have this enzyme. We will utilize this property in differentiating that. So suppose I am having a specimen. Suppose this is a test tube in my hand and I am having a specimen. I know it's a GPC because I can see violet cocci. But at some places it is cluster, at some places it is chains, and I got confused. So I want to know, is it step or strep? No worries. You take a drop of your specimen on a slide. You take a drop of your specimen on a slide and you mix H2O2 in that. Here I am mixing 1 drop of H2O2. H2O2 is hydrogen peroxide. It's a liquid present in my laboratory commercially. So it's H2O2. So what will happen? Now it is one of them, either step or strep. वही तो पता करना है. That's what I want to know. Okay, suppose it is having step. This one is step. Okay, so God has given catalase enzyme. They have the catalase enzyme. Catalase enzyme degrade H2O2. Catalase act on H2O2. And catalase enzyme convert H2O2 into H2O plus O2. So as soon as you mix the two with a stirrer, bubbles of oxygen will come. The bubbles of oxygen will come. Here, this one is, suppose it is Streptococcus. God has not given this enzyme, na? So they don't have Streptococcus. So H2O2 will not be degraded. It will remain as liquid only. So suppose this is my specimen. I want to know, is it step or strep? I want to know. I will take a slide. I will take 1 drop of this specimen and I will put 1 drop of H2O2 mixed with a stirrer. Within 1 minute, I can tell you, if bubbles are coming, it's step. If no bubbles are coming, it's strep. So catalase test, differentiate step and strep. Okay, now we have differentiated. This is step, oxygen bubbles. This is strep, no oxygen bubbles. How to further classify step? Boss, ये तो पता चल गया कि ये step है. I know it is step. But how to further classify it? How many types of steps are there? How to further classify step? You are getting my point. How to further classify step? So based on catalase test, we are differentiating Staphylococcus from Streptococcus. We have already done so. Now I'm not interested in Streptococcus. Yesterday I taught you Streptococcus ka classification alpha, beta, gamma. But today I'm interested in telling you the classification of Staphylococcus. How to further classify it? Okay, for further classifying we need another test. The name of that test is coagulase test. Okay, coagulase test is positive in the main hero, and it is negative in the side heroes. Have you seen a movie in which multiple heroes are there? Maybe a movie in which 3 heroes are there. I don't know the name. One hero is main, okay, and other 2 heroes are side. So it's a movie like that. You can see the main hero is Staph aureus and the 2 side heroes are Staph epidermidis and Staph saprophyticus. These 2 are side heroes. This one is the main hero. Side heroes together known as cones. They are together known as cones. The main hero is coagulase positive. Okay, coagulase is positive in main hero and side heroes are coagulase negative, CONS. Coagulase, CO is coagulase negative Staphylococcus. So together I will call them cones. Okay, so ma'am, what is coagulase test? See the diagram. The great God, again. You believe in God? The great God have made them like that, not we humans. See the diagram of all main hero, Staph aureus, and see the diagram of side hero, that is cones. So God has given an enzyme coagulase to all main heroes. God has given them. We can't help. And none of the side hero God has given this enzyme. Cones don't have this enzyme. The name of the enzyme is coagulase. By the way, catalase is present in both of them. Both are staphylococcus, so catalase दोनों के पास है. Both of them have catalase, but we are talking about coagulase. Okay, it is present in main hero, not in side hero. We will utilize this property to differentiate them. How to differentiate them? अच्छा, what does coagulase do? What? जैसे catalase degrade H2O2, okay, and convert it into oxygen. What does coagulase do? नाम में ही मतलब है. The name itself depicts in the meaning. Meaning itself depicts in the name. Coagulase means you see the coagulation pathway. You know there are 12 factors. You know the cascade. 12, 11, 10, 9. You know the cascade. I'm not interested in that. I'm interested in the last step. How clot form? Blood clot. How does it form? When factor 1 get activated. When factor 1 get activated because of the cascade. Intrinsic, extrinsic pathway. Fibrinogen get converted to fibrin. Okay, it cannot be directly activated. It is via this, and then it get activated. It is known as cascade. Okay, but coagulase is an enzyme that can directly convert it. Coagulase, it is making the clot. That's why it is known as coagulase. Now, suppose I'm having a specimen. Where is my test tube? Suppose I'm having a specimen. I know it's step. It's catalase positive. It's giving oxygen bubbles, okay, with H2O2. I know it's staphylococcus, but I want to know, is it main hero or side hero? Okay, what should I do? No worries. Take a slide. Take one drop of specimen over here. Take one drop of specimen over here. Okay, and don't put H2O2. Okay, put rabbit serum. One drop of rabbit serum, commercially available. You know, serum is liquid. Serum is liquid. Put one drop and mix with a stirrer. Okay, now what will happen? Use your common sense. What will happen? Suppose this is the main hero, step aureus. So God has given coagulase enzyme to them. Okay, coagulase will clot it. It will convert factor 1, fibrinogen to fibrin. Okay, it will get clotted. Have you done blood grouping like that? It will get clotted. And here, cones are present. God has not given this enzyme to the cones. So this will not clot. So on mixing, suppose I am having a specimen. It is step. I want to know, is it main hero or side hero? No worries. In 1 minute, I will tell you. Take a drop on a slide. And put rabbit serum. I am having commercially available rabbit serum in my laboratory. I will mix the two. In 1 minute, I will tell you. If it is clotting, it's main hero. It's not clotting, it's side hero. So this is coagulase test. Okay, they asked image-based question and all. Principle. See here. You can see. You can perform this test on the slide as I have performed. You can perform the same test on the test tube also. You can see. So here you can see clot is formed. Here no clot is formed. I'm mixing two things. My specimen with the rabbit serum. Here clot is formed, no clot is formed. Here clot is formed, here no clot is formed. So based on which I can say, is it main hero or side hero? So first I have applied catalase test to differentiate step from strep. Then I have applied coagulase test to differentiate the main hero from the side hero. I know these are bechare side hero, side hero, but it doesn't mean side hero don't have their identity. It's not good to call them side hero. They also have their identity. They also have their name. How to differentiate the two? We have mixed the two and called them cones, the side heroes. How to differentiate the two side heroes from each other? How to differentiate epidermidis from saprophyticus? My question is that. How to differentiate these two? You are getting my point. So third and last test we are applying is Novo-Bayesian susceptibility test. What is Novo-Bayesian susceptibility test? Everyone have their own fear. Some get afraid of optochin. Some get afraid. Yesterday I taught you, no? So by basic person, some get afraid by Novo-Bayesian. Novo-Bayesian is a chemical like penicillin, cephalosporin. It's a chemical, Novo-Bayesian. It's available in my laboratory. So what I will do? I will take a agar plate. This is my agar plate. And I will cut a filter paper in a circular fashion and dip it in the Novo-Bayesian. Available in my laboratory. Novo-Bayesian. Keep it at the center of the agar plate. Okay, like this. You can see like this. So you can see this is Novo-Bayesian containing filter paper. This is Novo-Bayesian containing filter paper. Okay, on the agar plate. Now pour your specimen over here. Pour your specimen over here. One of them is epidermidis and one of them is saprophyticus. I want to know that. Now let the colonies grow for 24 to 48 hours. Now see the colonies, yellow color, lawn culture. See the colonies. Here the colonies are growing at the periphery, not coming closer because they are afraid. Once they go closer, Novo-Bayesian will kill them. So this is susceptible to Novo-Bayesian. Okay. And here colonies are nidder. They are strong. They don't get afraid of Novo-Bayesian. They are coming and touching the rim of the Novo-Bayesian, the yellow colonies. So they are resistant. So epidermidis is darpoke. coward. Epidermidis is coward. And saprophyticus is strong, resistant. S for saprophyticus, S for strong. This is how you can learn. People, summary. See the yellow color. See the three tests. See the three yellows, people. See my three tests. Are you people there? What are the three tests? Let me ask you leading question, leading answer. Tell me catalase test used to differentiate what from what. Tell me coagulase test used to differentiate what from what. And tell me Novobasin susceptibility test used to differentiate what from what. Three questions, three answers, waiting for your response. Fast. Catalase test differentiates staph and strap. Okay, better not to write like this. Better to write it is positive in staph and it is negative in strap. Okay, and we see for oxygen bubbles. Coagulase test differentiate the main hero from the side hero. So it is positive in staph aureus and it is negative in cones. Okay, and we look for the clotting by after putting rabbit serum. And Novobasin susceptibility test differentiate the two cones from each other. Okay, so it is susceptible in epidermidis, staph epidermidis, and it is resistant in staph saprophyticus. Got my point? So your questions, all these questions are asked in your exam. Got it? Now 7 questions, 7 answers. People are not responding. Are people understanding? Kindly respond this question. Are people understanding? Is the speed is good? You are getting it? You are getting the revision fast? All these things I assume you already know. We are just revising it. Yes? Chalo. So tell me the answer of these 7 questions quickly, fast. Enumerate GPC. There are 2 GPC. Answer of this is staph, strap. I don't know which one. Tell me a GPC which is catalase negative. Catalase negative, I will say, ma'am, it's strap. It's strap. Tell me a GPC which is catalase positive. I will say it's staph. Okay. Tell me a GPC which is catalase positive. Okay. But coagulase also positive. So it's the main hero. Staph aureus. Tell me a GPC which is catalase positive. Okay, but coagulase negative, so it is cones. And Novobasin susceptible is epidermidis and Novobasin resistant is saprophyticus. I hope you got it. All these are short questions, MCQ based questions in your exam. Okay, they don't ask you such directly, but they give you clues in the question. Introduction is over. Okay, classification is over. Okay, so introduction is over. Classification is over. Now coming on the morphology. What I'm teaching you? I'm not teaching you all these. I'm teaching you one of them. Which one? The main hero. My topic is this one because this one causes food poisoning. Others do not cause. I will teach you the other two in other systems. But this one I will teach you in GIT. Staph aureus I am teaching you in GIT because this one causes food poisoning. I will teach you Staph aureus again a day after tomorrow in skin because it causes skin infection. That time we will just quickly flip it. But I will teach you 2 times. Today in GIT and in skin infection also. Okay, chalo. So let's continue with Staph aureus. We have already seen the classification, introduction. Okay, it was a long introduction, but it was necessary. Let's come on morphology. How many of you were present yesterday? I guess you know my pattern. I ask you to do not learn morphology. People, we don't have to learn morphology. In morphology, I tell you 4 things. Standard in bacteria. It's gram stain, capsule, spore and motility and something extra if it is having. Okay, gram stain. You tell me gram stain. Where is Staph aureus? Here is Staph aureus. So it's gram positive cocci. You don't have to learn it if you know this classification. Learn it once. Don't learn it 60 times. Learn it once for all. So you see its category, it's GPC. It is GPC, but you should say it is in clusters. You can see the diagram. You can draw this diagram also. It is in clusters. You already know. Okay, what about capsule, spore and motility? Don't learn it. Learn 3 mnemonic once for all. Don't learn 60 different combinations. So you see the 9 bacteria which are capsulated, the 4 bacteria which form spore and 12 bacteria which are motile. Now my bacteria is Staph aureus. Is it coming in this mnemonic? No. Is it coming in this mnemonic? No. Is it coming in any of these mnemonic, motility mnemonic? No. So I will say it is non-capsulated, non-spore forming, non-motile. I don't learn. I apply it on the spot. So you can see my pattern, no capsule, no spore, no motility. The extra point is that you have to learn. It is the pigment producing bacteria. It produces golden yellow pigment. Yesterday also I taught you one bacteria. Can you tell me which one? Yesterday also I taught you one bacteria which produces pigment. I taught you Pseudomonas yesterday. If you were present in my yesterday's session, I taught you Pseudomonas aeruginosa. It produces 4 color pigment. Most common green, yellow, red, black. But Staph aureus produce only golden yellow. So only 2 bacteria produce pigment among these 60. है ना? Staph aureus, golden yellow, and Pseudomonas 4 color pigment. Most common green. It's a golden yellow pigment producing bacteria. Coming on the culture. Okay. If you can learn all, very good. If you can't learn all, learn the selective media. You should know the selective media of all bacteria at one place. Please, and that's why I've given the columns and rows. You can revise them in a row at one place before going your exam. Okay. Now nutrient agar. Nutrient agar. It from golden yellow colony. Appreciate the nutrient agar. Appreciate the golden yellow colony. Have you done paint at your home sometime during Diwali or sometime? We do oil paint. Most common color in oil paint we use is yellow color. Okay. So it is looking like the drops of oil paint are there. Okay. See the zoom version. You can see the drops of the oil paint. That's why it is known as oil paint appearance. It's very shiny. The colonies are looking like the drops of the oil paint. So please, it's a question. Oil paint appearance. You should draw like this or draw like this. Oil paint appearance. Okay. Nutrient agar. Nutrient agar, the background is gray color. Okay. On which golden yellow color is not very good appreciated. So in nutrient agar, we add milk, doodh, milk. You drink milk, the same milk. We add it here. So background become white. Opaque white, on which golden yellow color is much more appreciated. So here also oil paint appearance, here also oil paint appearance. So on nutrient agar, oil paint appearance. On milk agar, it's nutrient agar only. Only milk is added. Background become white. So oil paint appearance is more appreciated. So here also oil paint appearance. Okay, on blood agar, you have to say alpha, beta or gamma. You know what is alpha, beta, gamma? Yesterday I taught you. So on blood agar, it's beta. Beta means complete hemolysis. So around the colony, there is a clear zone. Translucent zone of discoloration. So it's beta. Okay, on McConkey, you have to say lactose fermenter or non-lactose fermenter. Is it pink or yellow? Yesterday I taught you. So it is lactose fermenter and it forms pink colonies. Lactose fermenter and form pink colonies on the McConkey agar. Okay, there is an indicator media that is known as PPA. The full form of PPA is phenolphthalein phosphate agar. Phenolphthalein phosphate agar. It's the indicator media on which also pink color is there when we pass the ammonia gas. It indicates that the colonies are grown. Okay, so PPA agar. Okay, the most important you have to learn after 5 is selective media. There are 3 selective media. Let me revise these 5 and then we will come on selective media. With the help of images you learn. Say nutrient agar pe, say nutrient agar, oil paint appearance. See, milk agar, again oil paint appearance. Better. Okay, blood agar, beta. Not alpha, not gamma, beta. McConkey agar, lactose fermenter, and that's why it formed the pink colony. It's lactose fermenter. Okay, PPA is indicator. PPA is indicator media. Phenolphthalein phosphate agar. So here also pink color is there. Now coming on last selective media. Okay, so take 3 nutrient agar. See ma'am, nutrient agar pe all the things will grow. The complete world grow. All bacteria grow on nutrient agar. I know. I will add something in that. So take 3 nutrient agar. In the first nutrient agar, add lithium chloride and telluride. Lithium chloride and telluride. Now try growing any bacteria. None of the bacteria will grow except Staph aureus. All other bacteria will die. Only Staph aureus will grow and none of the other bacteria will grow. So this becomes selective. And this media is known as Ludlam. Ludlam contains lithium chloride and telluride. Okay. The second nutrient agar. Here I will add mannitol. Here I will add mannitol. Mannitol and NaCl. Mannitol and salt. Now try growing any bacteria. No bacteria will grow. Only Staph aureus will grow. So it becomes selective. So it is known as mannitol salt agar. I am adding mannitol and salt. And third one. In the third nutrient agar, add 2 antibiotics. Add nalidixic acid and add colistin. These 2 antibiotics kill all bacteria except Staph aureus. Staph aureus is not killed by them. Now try growing any bacteria. None of them will grow except Staph aureus. So these are the 3 selective media. So the third one is known as Columbia because it is discovered in Colombia. So Ludlam media, mannitol salt agar and Columbia. You have to learn it, people. I have tried to explain to you. So Ludlam contain lithium chloride and telluride. Mannitol salt contain mannitol salt. And Columbia contain colistin and nalidixic acid. By adding these things in the nutrient agar, none of the bacteria can grow except Staph aureus. And only Staph aureus will grow. No other bacteria will grow. So that is the culture media of Staph aureus. Biochemical reaction? No need to learn. Don't see the answers. You tell me. This is not capsule. This is catalase. By mistake. You tell me catalase and coagulase. I am teaching you Staph aureus. Is it catalase positive or negative? Of course, it's catalase positive. Common sense. Okay. All staphylococcus are catalase positive. And this is the main hero. That's why it is also coagulase positive. See the clot of the rabbit serum. So it is catalase positive. You can see it is catalase positive as well as coagulase positive. We already know that. That is biochemical reaction. Coming on antigenic structure or virulence factor. Virulence factor. Give me a minute. Coming on the virulence factor. There are two virulence factors. It produces toxin, and it produces enzyme. Toxin and enzyme. It produces four type of toxin. What are the four type of toxin? Cytolytic toxin, enterotoxin, toxic shock syndrome toxin, and epidermolytic toxin. Four toxin cause four disease. Today, I am interested in one of them. Enterotoxin, which cause food poisoning. Because my topic today is GIT, people. So I am interested in enterotoxin. What is cytolytic toxin? Cytolytic. Cytolytic. It causes rupture of human cell. So suppose this Staph aureus enters in my blood. After entering in my blood, it is secreting all four toxin. So cytolytic toxin will rupture my cell. Okay, my cells of the body, and it causes pus filled lesion. So I will teach you the pus filled lesion in skin. Okay, enterotoxin, it goes inside the GIT and cause diarrhea and vomiting. So this one I am interested today. I am teaching you GIT. The third is toxic shock syndrome toxin causing toxic shock syndrome. I will explain. And fourth is epidermolytic toxin. It also causes skin infection, SSSS. I will explain. So learn the four toxins first. These are the four toxins. And two enzymes are there, bound coagulase and free coagulase. I told you, it is having coagulase, it is coagulase positive. See this bacteria. This is Staph aureus. God has given Staph aureus two enzymes. See, one in the cytoplasm. This is free coagulase because it is present freely in the cytoplasm. This is known as free coagulase. Okay, and one in the cell wall, a component of the cell wall. Okay, one component of the cell wall. It cannot move here and there. It is a component of cell wall. It is known as bound coagulase. Both are coagulase, but one is free in the cytoplasm. The one which is in the cytoplasm is known as free. And the one which is in the cell wall is known as bound. So two type of coagulase is there that you have to learn. So you can see the bound coagulase and you can see the free coagulase. Okay, both of them are giving the coagulase. This one is present as a component of cell wall, and this one is present in the cytoplasm. Both of them gives coagulase test positive. But I told you, we can apply coagulase test in this test tube as well as slide. So the free one give positivity in the test tube, and the bound one give positivity in the slide. Okay, so this is tube test positive, this is slide test positive. Very important MCQ asked in your competitive exams. Okay, so that is the virulence factor. Now, mode of transmission, most important mode of transmission for food poisoning is ingestion of contaminated food and water. Yesterday I taught you respiratory tract infection. The mode of transmission everywhere was droplet inhalation if you have attended my session. So today mode of transmission in most of them, in all of them I think, is ingestion, because I'm teaching you GIT today. So mode of transmission most important is ingestion, but it can enter via direct skin to skin contact and via fomites also. But most important is this. Now coming on clinical feature. I told you 4 toxin, 4 disease. I told you 4 toxin, 4 disease. Tell me the name of 4 toxin. Quick. So the first toxin is cytolytic toxin. Cytolytic toxin cause pus-filled lesion. The second toxin, toxic shock syndrome toxin, causing toxic shock syndrome. Okay. Third toxin, epidermolytic toxin, causing SSSS. Staphylococcus scarlet skin syndrome, SSSS. And the last one, today I'm interested in the last one, enterotoxin. Enterotoxin causes food poisoning. I do not want to teach you the remaining 3. Remaining 3 detail, please understand people. Staph aureus. Kya padha rahi hu main? What I am teaching you right now? A bacteria. The name of the bacteria is Staph aureus. Okay, I am teaching you under a fixed set of heading. Now this bacteria causes infection in 2 systems in human body. It causes skin infections also, and it causes GIT infections also. So it will come twice. Okay, obviously it will come twice. But its morphology, its biochemical properties, its antigenic structure, that will not change. That is fixed. Okay, organisms do not change. Only the disease it is causing, we are dividing the disease in 2 different systems. You are getting my point. So I will repeat the bacteria again in skin. Quickly I will flip. I will not teach you again. But you must understand, these 3 diseases will come mainly in the skin. And this one is coming in the GIT. Okay, so I will give you the detail of these 3 just in the images. But this one I will give you much more detail over here because today I am teaching you GIT. You got my point? Okay. So you have to learn a bacteria completely. Only its clinical feature and lab diagnosis keep on changing depending on the disease to disease. If one bacteria is involved in multiple systems, it is not that one bacteria involve one system. One bacteria can involve multiple systems. So all other properties are same. Its introduction, its morphology, its biochemical property, its antigenic structure, All the things are same except its clinical feature that we will divide according to the system wise, and lab diagnosis will differ accordingly. Maybe treatment also differ accordingly, but other things are constant. I hope you are getting my approach. Okay, yes or no? Yes or no? Got it? Is it buffering for everyone or only for Juhalom? Jushalom? Kindly respond. Is the video is buffering? Check your internet. I guess from my side there is no problem. At my end there is no problem. Check your internet, refresh your internet. Kindly other people respond quickly. Everything video audio is good. Can we go ahead? Please respond. Anyways, so these are the 4 things. The first 3 I will give you in just overview. What are the pus filled lesion? Cytolytic toxin. Cytolytic toxin cause pus filled lesion. It can cause cellulitis. It can cause abscess. It can cause furuncle. It can cause folliculitis. It can cause all of these. Okay. So that is due to cytolytic toxin. Okay. What is SSSS? What is Staphylococcus scalded skin syndrome? It is due to epidermolytic toxin. What is epidermolytic? It separates the epidermis from the dermis. Epidermolytic. It separates epidermis from dermis. So you can see here the epidermis is separated from dermis. Epidermis is separated from dermis. It is known as Nicolsi sign. What is known as Nicolsi sign? The name of the disease is SSS, Staphylococcus scalded skin syndrome. This is due to epidermolytic toxin. What is toxic shock syndrome? What do you mean by toxic shock syndrome? Toxic shock syndrome occurs only in menstruating female. She is a female. She is menstruating. During menstruation, she is using vaginal tampons. Okay. Now, maybe due to unhygienic condition and vaginal tampons get contaminated with the Staph aureus. And Staph aureus is forming toxic shock syndrome toxin in the tampon. That toxic shock syndrome toxin enters vaginally. The toxin is entering vaginally. Okay. And after entering vaginally, it involves multiple systems. It involves multiple organ failure. The multiple organ failure in a menstruating female due to using contaminated tampons is known as toxic shock syndrome. This is toxic shock syndrome which occurs typically in menstruating female. The last one I would like to give you much detail because my topic is GIT. These 3 you have to see mainly in skin. I hope you got the approach. Now coming on the food poisoning. Okay. Food poisoning. You have to tell me 4 things in all food poisoning. Number 1, incubation period. What I am teaching you, Staph aureus, now where it is coming. You know this classification now. So Staph aureus is coming over here. So incubation period is 1 to 6 hours. No need to learn. It's 1 to 6 hours. 2 to 6 hours. After eating the food, within 6 hours, person have the symptoms. Okay. Which food? What is the food predilection? It is dairy. Dairy product. You know dairy product. Milk and paneer and cheese and pastry cream. The dairy products. Okay. Apart from that, meat and fish. Meat, fish and dairy. Dairy means milk. Milk and milk products. Okay. Symptom. Diarrhea ki vomiting. Both. Now see these 2 images. I will show either both or one of them. Okay. In all the 13 bacteria. In all 13 bacteria, either both will occur or only vomiting occur or only diarrhea occur. So with the help of images or something, you just try to retain that which bacteria is causing both, which is causing only vomiting, which is causing only diarrhea. So this one causes both vomiting as well as diarrhea. Staph aureus. Staph aureus cause both diarrhea as well as vomiting. What is the mechanism? Now please understand the mechanism. See this diagram. Please see this diagram. Diagram is crystal clear. No need of labeling. That's why I have not done the labeling. Even a blind person can say what is their interpretation of this diagram. You tell me the name of this bacteria. See 3 things. See 3 things. Color, shape, and appearance. What is the shape? Cocci. What is the color? Violet. And what is the arrangement? What is the arrangement? Clusters. So this bacteria is Staph aureus. No need to say. Just a second. Where it is gone. This bacteria is Staph aureus. Okay, and see the food. You can see the meat, you can see the fish, you can see the dairy product, you see the food. Now what this person is eating? Person is eating the food. Does the food contain bacteria? No. Food don't contain bacteria. Food contain the toxin formed by the bacteria. So bacteria is forming the toxin. It is known as preformed toxin. The food contains preformed toxin. You have not asked me the question. You have not asked me a question, ma'am, how it can be so fast? How these 2 can be so fast? I am eating something. Within 1 or 2 hours, I am having diarrhea and vomiting. Within 1 hour. Within 2 hours after eating. How it can be so fast? Because I am not eating bacteria. If I am eating bacteria along with the food, bacteria will go inside. In the intestine, it will make the toxin. It takes time. It takes time. But I am eating the ready-made toxin. Since I am eating the ready-made, preformed toxin, that's why it's so fast. So in both of them, it's preformed toxin. It's your question and you have to highlight it. It's preformed toxin. Got it? So this person is not eating bacteria. It is not eating the bacteria. Okay, it is eating the preformed toxin. This is preformed toxin. Got it? So preformed toxin is going inside, reaching the stomach and intestine. From the stomach and intestine, this preformed toxin go in the blood. Okay. From the blood, it go to the brain. In the brain, it does two things. Please understand mechanism. It stimulates the vomiting center, it stimulates the vagus center. Vomiting center, vagus center. Since it stimulates vomiting center, person have vomiting. Since it stimulates vagus center, vagus increases parasympathetic system. And that's why the person have diarrhea. Person have both vomiting and diarrhea. So it is vagus stimulation and vomiting center stimulation. So you can write down both vagus and vomiting center. Okay, so that is a mechanism of action, the food poisoning. I hope we are done with the clinical picture. Lab diagnosis. What sample you will take? Either we can collect the vomitus or we can collect the stool sample in a container. So we can take the vomitus, we can take the feces, or we can take the food, what these persons have eaten. Okay, so that these are having these symptoms. If the food is pending, I mean it is ruminant. Okay, so that is the sample. And we can detect the enterotoxin, the preformed toxin in these. The preformed toxin with the help of ELISA or latex agglutination test. Or we can do PCR. So basically we are not testing bacteria. In both of these, tests, I want to highlight we are testing toxin. Common sense, है ना? इन्होंने toxin खाया है, इन्होंने bacteria नहीं खाया है. So the vomitus contain toxin, the feces contain toxin. So we collect vomit, we collect feces, and we look for toxin. Now we can look for toxin with the help of PCR, with the help of ELISA, with the help of latex agglutination. I hope I'm crystal clear. Got it? So in today's session, most of the infections out of 30, simple treatment, symptomatic treatment, supportive treatment, oral rehydration solution. For most of the gastroenteritis and food poisoning, we don't have any specific antibiotic or drug of choice. So here we simply give ORS. Okay, in most of them today we will give ORS. Prevention, we don't have any vaccine available. We are done with the first bacteria. People respond. Can we go on the next one? Yes. So let's move to the second bacteria, Bacillus cereus. This was the biggest one. Others are small. So let's move to the second one, Bacillus cereus. Okay, I assume we can fill this column completely. Let me finish these 4, then we will fill it completely. Because one by one I will fill it, it will take time. Let me finish these 4, then we will try to fill. You can have a look on my pre-filled version. I have already filled it. The empty one, the filled one. Whatever images I have shown you, all the images are over given here. Each and every information which is there in the PPT, which is there in the lecture, it is already there. Okay, so first you read the organism like this, the 4 organism like this, then you revise row wise. First column wise, then row wise. Follow this pattern and get back to me with 100% strike rate in all the exams. First read the organism column wise, all the organisms, and then read them row wise. All the introduction at one place, all the culture media at one place, all the clinical feature at one place, all the lab diagnosis at one place, all the drug of choice at one place, like that. And you see how it fit in your mind. Got it? Shall. So let's move. Where is my pen? So let's move to the next bacteria. The next bacteria is Bacillus cereus. People, Bacillus cereus. So Bacillus cereus is anthracoid bacilli. What is anthracoid bacilli? The main hero in Bacillus is Bacillus anthracis. You see the classification now. Where is Bacillus? See, this is Bacillus. This is Bacillus. So main hero over Bacillus is Bacillus anthrax or anthracis. All other Bacillus apart from Bacillus anthracis, there are many Bacillus. They all are known as anthracoid bacilli. So by the term anthracoid bacilli, I mean the Bacillus apart from Bacillus anthracis. Okay, the one which causes anthrax. Usko chhod ke jo bhi bach gaye, that is anthracoid bacilli. So it is one of the anthracoid bacilli. Got it? Morphology, no need to learn. You tell me where it is coming. It is one of the gram-positive bacilli. Bacillus. ABCDLM ka B hai. ABCDLM ka it is B. Okay, so that is gram-positive bacilli. Capsules for motility. You tell me. I am talking about Bacillus cereus. Is it coming in this mnemonic? No, I don't think so. Is it coming in this mnemonic? Yes. All Bacillus come in this mnemonic. Yes. So it is spore forming. It is spore forming. What about motility? What about motility? All Bacillus are motile except anthrax. I am not teaching you anthrax. I'm teaching you Bacillus cereus. So it is motile. So motility is also present. So see the summary. No need to learn. Non-capsulated, but spore forming and motile. Motile by Peritragus flagella or polar flagella. You can see it is motile by Peritragus flagella. Learn these three mnemonic people if you want to learn the morphology. So no capsule, spore present and motility present. Motility is present with Peritragus flagella. Okay, so that is a morphology. That is a morphology. Okay, after morphology, the selective media. Take nutrient agar. You will see my nutrient agar with the complete world will grow. I will add something. So take this nutrient agar and add four things. Add four things. Add mannitol in it. Add egg yolk in it. Add phenol red in it. And add polymyxin in it. Add four things. Okay, it is known as MYPA. Okay, mannitol, egg yolk, Y for yolk. Mannitol, egg yolk, phenol red, polymyxin agar. Okay, so if you add four things, now try growing any other bacteria. No other bacteria will grow except Bacillus cereus. Only Bacillus cereus will grow, others will not grow. What was the selective media of Staph aureus? Staph aureus, three selective media: Luteolum, mannitol salt, and the third one was Columbia, containing cholesterol and nalidixic acid. Okay, what is the selective media of Bacillus cereus? MYPA. Learn the full form. So here the selective media is MYPA. Okay, biochemical reaction is something I cannot help, but you have to learn it. Okay, so most important is catalase positive, like Staph aureus only. It is MR and BP also positive, urea is also positive, and nitrate reduction is also positive. If you want to learn, although not very important, but if you want to know, all these reactions are positive in Bacillus cereus. Bacillus cereus. What is the virulence factor? Virulence factor. The virulence factor, it is also having toxin. Only enterotoxin. There we have four type of toxin. Cytotoxin, toxic shock syndrome toxin, okay, epidermolytic toxin, and the fourth was entero. Here we have only entero. We don't have other three, but entero is of two types. There we have entero only one type. Here we have two type of entero, the emetic and the diarrheogenic. The emetic one is heat stable, the one which causes vomiting. Emetic means vomiting. Okay, the one which causes vomit is heat stable. The one which causes diarrhea is heat labile. So vomiting is heat stable. Diarrhea is heat labile. Make some mnemonic and learn it. Make some trick and learn it. Mode of transmission is same. Ingestion of contaminated food and water. Clinical feature. It causes two type of food poisoning. People, please understand. It causes. I'm teaching you Bacillus cereus. Pehli baat to. First of all, I'm teaching you Bacillus cereus. It causes two type of food poisoning. What two type of food poisoning it is causing? People, what two type of food poisoning it is causing? See this person. See this person. This person is eating fried rice. Chinese fried rice, to be specific. Fried rice. Okay, you like fried rice? Beware of it. So it can cause the food poisoning, Bacillus cereus food poisoning. It is a food predilection. So here the food predilection is Chinese fried rice. And this person is eating the raw vegetables and meat. So raw vegetables and meat. Okay, here the Chinese fried rice contain preformed toxin. Preformed toxin. Okay. And the meat and vegetables do not contain preformed toxin. It contains the bacteria. It contains the bacteria, Bacillus cereus. This is the bacteria. Okay, so basically this person is eating toxin and this person is eating bacteria. So I'm drawing the toxin with the red color. So here the red toxin is going inside. Red toxin is going inside. Okay, and here the bacteria is going inside. The bacteria is going inside. I'm drawing the bacteria with blue color. So here bacteria is going inside. So see here toxin and here bacteria. So that's why this one is fast. This is preformed, na? So toxin act immediately. Okay, here bacteria after going inside, it will also form toxin, but it takes time. It takes time. It takes time because it will also form toxin, but it takes some time. This one is preformed. This one is preformed. So this person within 1 to 6 hours, vomit. Only vomit, no diarrhea. No diarrhea. That is known as emetogenic food poisoning. And this person after 6 to 16 hours, because it takes time, toxin is not preformed, it is formed in intestine later on. It takes 6 to 16 hours. It is having only diarrhea. It don't vomit. And this one is known as diarrheogenic food poisoning. How many of you got it? You get question on that. So see the two type of food poisoning caused by Bacillus cereus. Okay, what are the two type of food poisoning? The two type, you get this question, Bacillus cereus, the two type of food poisoning. Emetogenic type and diarrheogenic type. Emetogenic type, the food is Chinese fried rice. Here the food is meat and vegetable. Here person only vomit, no diarrhea. Here person only diarrhea, no vomit. Here incubation period is 1 to 6 hours because the toxin is preformed. That's why it is fast. Here the incubation period is 6 to 16 hours because toxin is formed in the intestine. It takes time. This toxin is heat stable, the one which is in vomit. And this toxin is heat labile, the one which is in diarrhea. Howsoever you learn, but you have to learn it. Okay, I have drawn the diagram also. So you can see the person who is eating fried rice is vomiting, only vomiting, not diarrhea. The person who is eating the meat and vegetable is having only diarrhea. That person is having only diarrhea, no vomiting. So this is amitogenic type, this is diarrheogenic type. Here the incubation period is 1 to 6 hours, here the incubation period is 6 to 16 hours. The food you can see, the symptom you can see. Here the toxin is preformed. And here the toxin is formed in the intestine. Okay, this toxin is heat labile. This toxin is heat stable. So please learn the same table in the form of the diagram. That's why Bacillus cereus was coming here also, here also. Now you understood. Okay, Staph aureus was coming only here, but Bacillus cereus coming in this category, this category. So here it is the amitogenic food poisoning, which is within 6 hours. And here it is diarrheogenic food poisoning within 6 to 16 hours. I hope it's crystal clear. Can we revise it quickly? Can you help me in revising this table? Say amitogenic type and diarrheogenic type. What is the food predilection? Here the food is Chinese, fried rice. Here the food is meat and vegetables. Symptom only vomiting, no diarrhea. Only diarrhea, no vomiting. Incubation period 1 to 6 hours, 6 to 16 hours. Why it is so fast? Because the toxin is pre-formed. Why it is slow? It is taking time because toxin is formed in intestine. Okay, heat, labile, heat, stable. Done, 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 done. You get many MCQs also and short question also based on that. Lab diagnosis is standard. Learn once for all. All bacteria. Yesterday also I taught you. We do direct microscopy, we do culture, we do biochemical reaction if necessary, they are important, and we do molecular. Molecular mein PCR. For bacteria it's PCR. For virus it can be PCR or RT-PCR. So use your common sense for writing lab diagnosis. Don't try to mug it up from organism to organism. In direct microscopy you will get gram-positive bacilli. Okay. Culture mein you will do on MYPA agar. You know the MYPA agar. You can do the PCR. Okay. Treatment. No specific treatment. We don't have drug of choice. We will do ORS. We will give ORS. But sometimes the food poisoning is very severe. In that case only, we give clindamycin, erythromycin or vancomycin. Otherwise, we don't give. It is not DOC. I have not written DOC. Only in severe case we try. So we are done. Prevention, no vaccine available. We are done with 2. Can we move on the third one? Huh? Can we move on the third one? Yes, Harshal, absolutely right. So let's move on the third bacteria quickly. Clostridium perfringens. Now I will teach you 2 Clostridium back to back. I will teach you Clostridium perfringens followed by Clostridium botulinum. 2 Clostridium. So let's start the third bacteria, Clostridium perfringens. How many of you know the old name of Clostridium perfringens? In the introduction, you write down the old name. It's also known as Clostridium welchii. Initially, it's the old name. Okay, nowadays it is known as Clostridium perfringens. So Clostridium welchii. Clostridium welchii is the old name, but you should know that sometimes they can ask you. It is saprophytic. Okay, it is saprophytic. It is present in soil. The spores are present in soil. It is invasive as well as toxigenic. It produces toxin. Okay, how many of you know this mnemonic? Have you attended my first session? Can't breathe air. I'm talking about obligate anaerobe. I am talking about obligate anaerobe. How many of you know this mnemonic? I guess everyone. So C, can't ka C is Clostridium. So I am teaching you Clostridium, both the Clostridium. Actually all Clostridium. Okay, I will teach you one more difficult today only. I will teach you 3 Clostridium today. So all the Clostridium are obligate anaerobe. You know that. Can't breathe air ka C hai ye. So tell me the culture media in which we grow obligate anaerobes. I taught you culture media, na? Tell me the culture media in which we grow these obligate anaerobes. Robertson cooked meat agar. If you remember, I taught you in the first session. Watch my sessions in a sequence. Okay. So Robertson cooked meat agar we use for anaerobic bacteria. Yes or no? So before I start the detail of this, and the detail of the next Clostridium difficile, 3 Clostridium, you should know the classification of Clostridium. Okay, in the introduction itself. How many type of Clostridium? There are many type of Clostridium. But I will teach you 4. 3 today and 1 in CNS. Clostridium tetani. Tetani I will teach you in CNS, which causes tetany. Tetany is a disease of central nervous system. So I will teach you that in CNS. Okay, but 3 I will teach you today, which cause GIT infection. I will teach you perfringens, I will teach you botulinum, and I will teach you difficile also. Perfringens and botulinum causes food poisoning. Difficile do not cause food poisoning. It causes antibiotic associated colitis. Okay. And tetany I will teach you later on. But you should know the complete classification. So classification of the Clostridium, you can see there are many type of Clostridium. The classification is based on RCM. You know what is RCM? I taught you Robertson cooked meat agar. What is Robertson cooked meat agar? What is RCM? You tell me. What is RCM? In RCM, we take a test tube. Can you see? Here we are taking a test tube. We are filling it with the nutrient broth, and we are taking the meat pieces, ox heart meat pieces. We take a ox, we kill the ox, we take the heart of the ox, and we cut the heart of the ox into multiple pieces and put it here. So these are the meat piece. What does meat contain? You eat meat. So what does the meat contain? Meat contain carbohydrate as well as fat. Protein. Carbohydrate as well as protein. So in meat piece, carb and protein both are present. I guess everyone know it's a rich source of carb, rich source of protein. What you like in your diet? What you like in your diet? Everyone have their preference. You like carb more or you like protein more? Or you like both equally? Some people like sweets, they like carb lover. Some people know muscle building, okay, body building, they like protein. So which category you belong? So similarly, Clostridium are of many types, based on which category they are coming. So if I'm putting one drop of Clostridium over here, one drop of Clostridium over here. So what are the possibilities? This Clostridium will come inside the meat piece and eat only carb, not protein. So the meat piece will convert into pink color, and or else it will eat only protein but not the carb. So meat piece will convert into black color. Okay. So this one is known as saccharolytic Clostridium, and this one is known as proteolytic Clostridium. So Clostridium are of two types, saccharolytic Clostridium and proteolytic Clostridium. Based on the color of the meat piece, we will decide. See, so if I am putting a one drop of Clostridium over here, after going inside, meat piece become pink in color, so it is saccharolytic Clostridium. Meat piece become black in color, it is proteolytic Clostridium. So based on the color of the meat piece, we classify them. So this is the classification. I will not read it, you just have a look. What are the categories in the classification? You can see the categories in the classification. This is the Clostridium which eat only protein and no saccharolytic. This eat only saccharolytic but not protein. So this one is only proteolytic. This one is only saccharolytic. This is neither proteolytic nor saccharolytic. It don't like protein or carbohydrate. It like fat. No protein, no carbohydrate. This one. Okay, and these one like both. These one like both proteolytic as well as saccharolytic. These one predominantly proteolytic, these one predominantly saccharolytic. So there are many Clostridium in our syllabus, but you should know this classification, you should have an overview, and you should understand how we are classifying. So I taught you the classification of all bacteria till now, whatever I taught you yesterday also, today also. I'm not skipping even classifications. You can see the real images. You can see the meat piece converted pink color, this is saccharolytic. You can see the meat piece converted black color, this is proteolytic. I hope you got it. Okay. Currently, I am teaching you Clostridium perfringens. Currently, I am teaching you Clostridium perfringens. Where is Clostridium perfringens? It is coming over here. You can see Clostridium perfringens. So it is mainly saccharolytic. It eats both, but mainly it is saccharolytic. So focus on perfringens now. No need to learn the gram staining. I guess everyone know where in the gram staining Clostridium perfringens is coming. So here is Clostridium. All Clostridium are gram positive bacilli. A, B, C, D का C है ये. A, B, C, D, L, M का C. So they are gram positive bacilli. No need to learn that. What about spore, capsule and motility? It's confusing. I'm teaching you Clostridium perfringens. People, Clostridium perfringens. Is it coming in this mnemonic? Have a look. Have a look. Don't rush. Have a look. Yes. Only perfringens is capsulated. Other Clostridia is not capsulated. Perfringens and Deutericum are capsulated. Others are not capsulated. So yes, it is capsulated. Capsule is present. What about spore? All Clostridia have spore. All, without any exception. All of them are spore forming. So this is also positive. This is also positive. This is also positive. What about motility? What about motility? So here is Clostridia. All Clostridia are motile except the Raja, except the king. I told you this in my first session. Clostridium perfringens is not motile. Rest all Clostridia are motile. Currently, I am teaching you the Raja only. I am teaching you the king only. So it is non-motile. It is non-motile. Okay, it is non-motile. You can remove titany over here. Titany is motile. Okay, I am correcting. Titany is motile. So you can see it is capsulated. It is spore forming. Capsule positive, spore positive. But motility negative. It's coming in exception. So same thing I have written over here. Okay, capsule positive, spore positive, motility negative. And it's the obligate anaerobe. It is coming in, can't breathe here. I guess you know that. Okay, see the three Clostridium I'm going to teach you today. See their morphology. The first one I'm teaching you perfringens, capsule present, spore present, but motility absent. For Clostridium, they will keep on changing the things. That's why I have compared them. Okay, now coming on culture. The culture of the Clostridium. Quickly, we will see the culture of the Clostridium. Yes, culture of the Clostridium. I will teach you four culture. Blood agar, RCM, egg yolk and reverse camp. Let me explain. On blood agar, either it is alpha or beta or gamma. Yes or no? Till now, On blood agar मैंने बताया है, I taught you either it is alpha or it is beta or it is gamma. It's a unique bacteria which shows double hemolysis. So see, this is the colony. Around the colony there is alpha, and around that there is beta. So double hemolysis. Around the colony, you can see this is the colony. Around the colony there is alpha hemolysis, and around that there is beta hemolysis. So it is known as double hemolysis, bull's eye, okay, target hemolysis. So on blood agar, a unique one and only bacteria, Clostridium perfringens, which show double hemolysis or target hemolysis. It show double hemolysis or target hemolysis. The second is RCM. RCM पे pink color, it's saccharolytic. We have already seen RCM पे, it's saccharolytic. Take egg yolk. This is egg yolk, okay? This is egg yolk. It is having many toxins. One of the toxin is alpha toxin. Okay, I will tell you in virulence factor. The alpha toxin produce a zone of opalescence around the colony. Zone of opalescence around the colony. Okay, so there is a colony, and around the colony there is clearance, zone of opalescence or zone of clearance, which is produced by alpha toxin, that is lecithinase. This is known as Niegler reaction. So on egg yolk, it gives Niegler reaction positivity. Niegler reaction positivity. And it is the only bacteria, one and only bacteria, which is reverse CAM positive. You will say, what is reverse CAM? You can see this is reverse CAM. Let me explain. In camp or reverse camp, we take blood agar. This is blood agar. Okay, imagine this is blood agar. What we are doing on blood agar? What we are doing on blood agar? Please see. Here we are taking a perpendicular strip of group B streptococcus. That is Streptococcus agalacti. Streptococcus agalacti. That is group B, not group A. Group A is Streptococcus pyogenes. Group B, beta hemolytic streptococcus, Streptococcus agalacti. It shows beta hemolysis. Okay, so around these strict, there is beta hemolysis. Perpendicular to this, I am taking my bacteria right now. Which bacteria? Clostridium perfringens. So perpendicular to this, take Clostridium perfringens. This also show beta hemolysis. This also show beta. This also show beta. So let me write down beta hemolysis here. Let me write down beta hemolysis. This is blood agar. Both show beta. So hemolysis start from the center. Please understand. Hemolysis start from the center. You can see hemolysis start from the center. But it is not equal on both the axes. If hemolysis is equal on both the axes, the zone of hemolysis should be circular. But it is not like that. The hemolysis is more on perfringens. Perfringens wala axis. The hemolysis is more on perfringens. Clostridium perfringens ka axis as compared to Streptococcus agalacti. And a butterfly shape. You can see a butterfly shape or arrowhead. Arrowhead shape, zone of hemolysis is formed, and this is known as reverse CAM. How many of you got it? It comes in your exam, university exam. What is reverse CAM? So take blood agar, take a streak of Streptococcus agalacti, that is group B beta hemolysis. Perpendicular to this, take take Clostridium perfringens. Now both show beta. This also show beta hemolysis, this also show beta. But the beta is not equal. We are comparing the beta hemolysis of the two. So we got more beta hemolysis on this axis, the Clostridium perfringens bacteria, which I am teaching you right now, as compared to the test bacteria. I will call this bacteria as test bacteria. I will call this bacteria as reference bacteria. And I will call this bacteria as test bacteria. Okay. So hemolysis on the test bacteria is more as compared to reference bacteria. And this is known as reverse CAM positive. So this is reverse CAM positive. You can appreciate the same diagram which I have drawn for you over here. Appreciate the same diagram. See, this is Streptococcus agalacti. Okay, and see, this is Clostridium perfringens. So appreciate the zone of hemolysis. I guess you can see the zone of hemolysis is more over here. Appreciate the butterfly shape or arrowhead shape, the zone of hemolysis. So this is reverse CAM. Now coming on biochemical reaction. Biochemical reaction is very unique here. A unique biochemical reaction that is known as Stormy clot or litmus milk reaction. Let me explain. You know, Clostridium perfringens is a gas forming bacteria. I guess everyone knows it. From which thing it formed the gas? Everyone know perfringens form gas. It's a gas producing bacteria. That's why it causes gas gangrene. But from which thing it formed gas? It ferment the sugar and form the gas. It ferment the sugar and it form acid and gas. All other bacteria ferment the sugar and form only acid. They do not form gas. But Clostridium perfringens is a unique bacteria which ferment the sugar and form acid and gas. Prove it. Can you prove it with the help of an experiment? Can you prove this thing? It is forming acid and gas both. Yes. We will prove it with the help of an experiment. Take a test tube. I'm having a small test tube. Suppose this is the test tube. Fill milk in that white color milk. Can you see this is white color milk? Fill this test tube with delicious milk. Okay. So fill it with white color milk. litmus paper. In your childhood, you may have read litmus. Huh? There are two types of litmus paper, blue and red. So have you read the mnemonic bar and rub? BAR bar and RBB rub. We used to learn in our 5th standard. So in case of acid, blue litmus convert to red. And in case of base, red convert to blue. Yes or no? So here acid will form. Na? We are saying acid will form. So if acid is formed, the blue litmus convert into red. Common sense. So litmus drops are available. So take milk and put blue litmus drops in that. Put few blue litmus drops in that. So it will become blue color. Appreciate the blue color. The milk become blue color. This is the milk which become blue color. Now put 1 drop of Clostridium perfringens in this. Put 1 drop of Clostridium perfringens in that. Okay, you know milk contains sugar. The name of the sugar is lactose. Yes? So lactose get fermented. Clostridium perfringens ferment lactose and convert the lactose into two things. Acid and gas. Acid and gas. As soon as the acid is formed, the blue litmus convert to red. Because of acid. In presence of acid, the blue litmus convert into red. You can see. You know, and second thing, acid clot the milk. Have you put lemon in milk? Dahi jam jata hai. The milk convert into curd. I mean, it get clotted. So here the curd is formed. The red curd. It was blue milk converted into red color curd. Hai na? Because it got clotted in presence of acid. So you can see the solidification occur. The curd is formed. And the gas is the disturbing the curd and forming the bubbles over here and coming it out. It is looking like inside the test tube a storm have arrived. Tufan aa gaya iske andar. So it is known as stormy clot experiment or litmus milk experiment, with the help of which we have proven that Clostridium perfringens is a gas forming bacteria. It forms acid and gas from the sugar. So it's the main biochemical property. Coming on antigenic structure. Can I come on antigenic structure? See. So antigenic structure में toxins. How many type of toxins? 4 major, 8 minor. 4 major toxin, 8 minor toxin. Currently, I'm not interested in the major. Currently, I'm not interested in minor. I'm interested in enterotoxin, which cause food poisoning, because my topic today is GIT. Okay, minor toxins are not important. Okay, major में 4 major toxin. Alpha, beta, eta and epsilon. Okay, epsilon and eta. Alpha, beta, eta and epsilon. These are the 4 major. Among them, alpha is most important. Alpha toxin is the main toxin, which is known as lecithinase. It's an enzyme, lecithinase, which is responsible for Niegler reaction, I told you, and it is responsible for gas gangrene. I will teach you this bacteria again in the skin. Skin mein many bacteria are from GIT only, because it causes gas gangrene. Gas gangrene is a disease of skin. Gas gangrene is not a disease of GIT. Currently, I am interested in enterotoxin, which cause food poisoning. That's why I am teaching you this here. Like Staph aureus only. Staph aureus also form 4 toxin. One of them is enterotoxin. This also forms 3 type of toxin. One of them is enterotoxin. Enterotoxin cause food poisoning. The route of entry is eating contaminated food for enterotoxin, but for other toxins, major and minor, the route of entry is wound contamination. Wound contamination, I mean this person was traveling on a bike, fell down, got an accident, and the soil contaminating the wound. In the soil, I told you that this is saprophytic. In the soil, the spores of Clostridium perfringens may be present and they can penetrate. So this is how other toxins enter. But enterotoxin is entering via this way. Currently, I am interested in enterotoxin. So Clostridium perfringens causes 3 diseases. I'm teaching you Clostridium perfringens, people. I'm teaching you. I wish these master charts are in your hand right now. They are already filled. You don't have to write even a single word. Whatever I'm teaching you, it is already written. Already 100% coverage with images. You have to just mark it. I wish it was in your hand. So you don't forget which bacteria you are reading right now. Okay. In the flow, sometimes you forget. Now, currently I'm teaching you Clostridium perfringens. It causes 3 diseases. 2 of them is skin. Okay. And 1 of them is GIT. Currently I'm teaching you GIT. So I'm interested in food poisoning caused by Clostridium perfringens. But you should know that it is causing gas gangrene and necrotizing enteritis also. I will teach you these 2 again in the skin. Skin wale day pe we will revise. So this is gas gangrene. This is necrotizing enterocolitis. Okay, but currently I am interested in food poisoning. Tell me the same 4 things for the food poisoning. Whenever question on food poisoning, you have to tell me 4 things. So which toxin causes food poisoning? Is it major toxin? Alpha, beta, eta, theta? No. Is it minor toxin? No. It is enterotoxin, which may be type A. It's your question. Enterotoxin is causing common sense. Enterotoxin causing food poisoning. What is the food predilection here? Meat. What was the food predilection for Staph aureus? Dairy, meat, and fish. Food predilection for Bacillus cereus, for Emetic type, it's Chinese fried rice. And for diarrheogenic type, it was vegetables and meat. And what's the food predilection for Clostridium perfringens? Again, meat. Okay, where it is coming, incubation period kya hai? What's the incubation period? Here, it is 6 to 16 hours. It is 6 to 16 hours. No need to laugh. It's 6 to 16. Is it causing diarrhea or vomiting? Only diarrhea. See my image, people. It's only diarrhea. No vomiting. Vomiting is absent. Vomiting is absent. It causes only diarrhea. That's what you have to learn. What's the mechanism of action? How does it cause diarrhea? How does it cause diarrhea? So this toxin, I mean the person is eating the meat. This is meat. Okay. Meat contains bacteria. Meat contains bacteria. So the bacteria is going inside. After going in the intestine, the bacteria form the toxin. This is the toxin. Okay. And these are the cells lining the intestine. Okay. This toxin causes pores in these cells. The toxin causes pores. It causes pores in the intestinal mucosal cells. And that's why the person had diarrhea. Okay, so this is the mechanism. It is not going in the brain, stimulating the vagus like Staph aureus. No. It is causing pores. Pores in the intestinal mucosa here only. That's why it is causing only diarrhea, no vomiting. I hope you got it. So that is the mechanism of action of food poisoning. Lab diagnosis, we test the bacteria. So collect the stool sample. Because it is causing diarrhea, no vomiting. You have to common sense, collect stool. We can't collect vomiters. So collect the stool sample and look for the toxin. You can look toxin with the help of ELISA or PCR. Look for toxin in the stool. Okay. Treatment, ORS. No specific treatment, symptomatic treatment. Prevention, no vaccine available. Coming on the last bacteria in this category, Clostridium botulinum. Clostridium botulinum. Can I start? The last bacteria in this category? Yes. Last bacteria in this category is Clostridium botulinum. It produces most potent toxin among all bacteria. The name of the toxin is botulinum toxin. Botulinum toxin, BT. Botulinum toxin. Among all the bacteria, 60 bacteria, most of them produce toxin. The most potent toxin is botulinum toxin. Botulinum toxin is the most potent toxin of the bacterial toxin in the world. So that you have to write down in the introduction. Got it? Morphology, no need to learn. It's also gram-positive bacilli. You can see this is Clostridium botulinum. It is gram-positive bacilli. What about capsule spore motility? Clostridium botulinum. So it is not coming in capsule. You can see. Clostridium perfringens and Butyricum is coming. This is Butyricum, not botulinum. So it is non-capsulated, but it is spore forming. All Clostridia form the spore. So spores are present. Spores are present. What about motility? What about motility? All Clostridia are motile except perfringens. I'm not teaching you perfringens. I'm teaching you botulinum. So it is motile. So no capsule, but spore is there and motility is there. So don't confuse. Learn the mnemonic and compare it with the perfringens. I taught you perfringens and now I'm teaching you botulinum. Okay, spore is present in both of them, but the other two things are reverse. This was capsulated, this is non-capsulated. This was non-motile, this one is motile. So don't confuse. If you try to mug up, you will get mad. Okay, so learn the mnemonics. The best ways to learn the mnemonic. So that is the morphology. It's an obligate anaerobe, can't breathe air. I guess everyone knows that. Okay, so morpho is done. Coming on culture. On culture, it's beta, and on RCM, it's both 50-50. Sometimes some serotypes are saccharolytic and some serotypes are proteolytic. It is both. Okay, biochemical reaction, the unique biochemical reaction, it produces H2S gas. It produces H2S gas, H2S production. Okay, that is a unique biochemical reaction. There in Clostridium perfringens, the unique biochemical reaction was Stormy clot experiment, litmus milk experiment. This one do not produce gas. It does not produce gas producing bacteria. Perfringens was gas producing bacteria. Botulinum do not produce gas. So here the unique reaction is H2S production. It don't produce gas. Okay. Virulence factor में only one toxin. Okay. Botulinum toxin which is of 8 types. What are the 8 types? A, B, C1, C2, D, E, F, G. You have to learn, people. You have to learn. Because among them, C2 cause food poisoning. C2, I'm interested in C2. It's your question. Which serotype of botulinum toxin causes food poisoning? In today's session, in purple GIT, I'm interested in C2. C2 causes food poisoning. It is enterotoxin. It causes. The remaining 7 are neurotoxin. The remaining 7 cause paralysis. The remaining 7. I will teach you this again in CNS. You are getting my point. The remaining 7 toxins are neurotoxin. You are getting my point. Many bacteria are overlap, people. In the next 3 sessions, many of them are going to repeat. So next time when I will teach now, I will not say the complete story. I will just flip the pages so that you can revise, and I will hold on the disease and diagnosis and treatment portion. That will differ. But morphology, biochemical friction, that's going to be same every time. You got my point? Sure. So here you can see total 8 serotype: A, B, C1, C2, D, E, F, G. C2 is enterotoxin causing food poisoning. The remaining all are neurotoxin. They cause flaccid paralysis. Cause flaccid paralysis. Okay, so I'm not interested in paralysis. The paralysis portion I will teach you in CNS. Okay, I am interested in food poisoning today. I'm interested in food poisoning. So it causes paralysis. In the paralysis, there is flaccid paralysis, there is diplopia, ptosis, the complete tone is gone. The baby become like this. It's flaccid paralysis. But in food poisoning, today my topic is food poisoning. In food poisoning, it causes only vomiting. It causes only vomiting. No diarrhea. No diarrhea, only vomiting. Purpharingus was causing only diarrhea, no vomiting. This one causes only vomiting, not diarrhea. The first two bacteria were causing both. Okay, so this one causes only. And where it is coming? No need to learn the incubation period. You learn this. Where it is coming? Clostridium botulinum. So Clostridium botulinum is coming here. So it is coming in more than 16 hours. So here you can see the incubation period is 16 to 36 hours. More than 16 hours. So that is the incubation period. Okay, so that is the clinical features we are done. Lab diagnosis, we will collect the vomitus and look for the toxin in that. The same. Okay, toxin demonstration. And treatment is antitoxin. Antitoxin for, paralysis. The main treatment. For GIT point of view, for food poisoning, we give ORS. For food poisoning, we give ORS. But if paralysis is there, the treatment of choice is antitoxin. The treatment of choice is antitoxin. Prevention, none of them have the vaccine. We are done with the 4 bacteria. People, we are done with the 4. Can I start the next 4? How many of you are interested in filling it quickly with me? How many of you are interested in filling it quickly? People, anyone who is alive, interested? The most compact version I'm telling you. From the book, we have covered 20-30 pages, people. If you start reading these 4 bacteria, you will get confused. These are very big bacteria given in the book. Here we have crushed them very easily. Yes? Chalo. So can we fill it quickly? Let me open my master chart. Okay, so here I'm having my master chart in front of me. You just remember what to write in each of them. So let me hang it in front of me. Okay, chalo. We will revise it. We will revise it quickly. Tell me the introduction. Here the introduction, Alexander and Rosenberg were the name of the scientists. And here I taught you the classification of the step completely. The 3 tests, catalase test, coagulase test, and novobusiness susceptibility test, based on which how we are classifying Staph aureus. Okay, coming on the introduction of the Bacillus cereus, I told you it is an anthracoid bacilli. It's an anthracoid bacilli. The bacilli apart from Bacillus anthracis. In Clostridium perfringens, I taught you it is saprophytic, and I taught you the classification based on RCM. Some of them are saccharolytic, some of them are proteolytic, some of them are none, some of them are both. So where Clostridium perfringens is coming, you have to see it. Botulinum, I told you it is the most potent toxin it is forming. Botulinum toxin is most potent toxin. That is the introduction. Morphology, I'm not saying. You just apply whether the gram stain may. This is GPC, this is GPB, this is also GPB, and this is also GPB. Okay, for capsule, spore, and motility, you just apply the mnemonic. I do not learn it on the spot. I apply the mnemonic and I come on a conclusion. Coming on the culture media. Okay, the selective culture media. Here I taught you 6 media, but if you can't learn all, learn the selective media. The selective media are 3 here. Ludalam, which contain lithium chloride and telluride. Okay, the second media was mannitol salt, and the third media was Columbia. Columbia containing colistin and aldehydic acid. Bacillus cereus में MYPA, mannitol, egg yolk, phenol red, phenoxylin agar. Okay, MYPA agar. Clostridium perfringens में RCM, and RCM में which colony? RCM में it is pink, saccharolytic. Apart from that, on egg yolk, it gives Nigler positivity. Apart from that, it gives reverse CAM positivity. And apart from that, on the blood agar, it gives double hemolysis or target hemolysis. Okay, Clostridium botulinum. In the botulinum, I told you only RCM. On the RCM, it shows both. And on blood agar, it shows beta. We don't have selective media. Selective media is RCM only in both of them. So we are done with the culture media of all of them. If you are confused, learn 3 selective here, learn 1 selective here, learn 1 selective RCM over here, one selective RCM over here. Learn the selective media. If you can learn all, it's very good. If you can't learn all, at least learn the selective media. Biochemical reaction, here it is catalase positive and coagulase positive. We know that. Okay. Bacillus cereus में MR positive, VP positive, urease positive, catalase positive, everything was positive over here. Clostridium perfringens में it is stormy clot experiment. Stormy clot experiment, and here it is H2S gas production. So if you see like column wise now, if you see like row wise, column wise as well as row, it will fit in your mind. So there is a biochemical reaction. Antigenic structure, virulence factors में we have to tell the toxin. Here 4 type of toxin, but I am interested in enterotoxin. Okay, here only enterotoxin, but it is of 2 type, emetic type and diarrheogenic type. Remembering here 4 major, 8 minor and enterotoxin. So I am interested in enterotoxin. And here 8 type of toxins are there. Okay, A, B, C1, C2, D, E, F, G. I am interested in C2. C2 is the enterotoxin here. So search the enterotoxin everywhere, because in GIT it is the enterotoxin who is causing the disease. Coming on the mode of transmission in all of them is ingestion of food and water. Clinical feature, this one causes diarrhea plus vomiting both. Okay, here the emetic type causes vomiting and the diarrheogenic type causes the diarrhea. The 2 type of food poisoning. Here only vomiting, here only diarrhea. Here only diarrhea occur and here only vomiting. So that's what you have to insist and you have to learn and you have to understand its mechanism also. Lab diagnosis is standard. Treatment, no specific treatment. ORS and all of them. In some of them, in severe cases, we have given the antibiotic. Prevention with no vaccine available. I hope in short we are able to revise it. People, can we go ahead? So let's start the next bacteria. This is how you have to fill these charts and learn it. So coming on the next 4 bacteria. All of them belong to Enterobacteriaceae. The next 4, E. coli, Shigella, non-typhoidal salmonella, and Yersinia. Okay, Yersinia enterocolitica and pseudotuberculosis. One by one we will take it. So the first one is the E. coli. Introduction, the scientist who discovered E. coli. What is the full form of E? Why it is known as E. coli? Who is E? E is Escherichia. Escherichia is the scientist who discovered it. So the full form of E. coli is Escherichia coli. Okay, E for Escherichia. So in the introduction, you write down Escherichia is the scientist who discovered it. So that is the introduction. Okay. Now this is a classification of Enterobacteriaceae. I'm saying these all 4 are Enterobacteriaceae. So before I start these, you should know how many bacteria come in Enterobacteriaceae. Among them, I'm teaching you 4, but these all are the bacteria of Enterobacteriaceae. So we divide Enterobacteriaceae. We classify Enterobacteriaceae based on lactose fermentation. The one which are lactose fermentation, they form pink colonies on Mekong agar. The one which are non-lactose fermenter, they form yellow colonies on Mekong agar. Okay, and the third is late lactose fermenter. They are pink from the beginning till end. They are yellow from the beginning till end. They are initially yellow, but after 24 hours, they change their color to pink. That's why they are known as dhokebaz, or they are known as late lactose fermenter. So we have LF, we have NLF, we have LLF. Okay, so LLF is only one. Shigella Sony. Sony is late. You can learn Sony. Sony is late, or Sony is dhokebaz, whatever you learn. One of the Shigella. Okay, and pink one are 4. I'm having a mnemonic. You know, cheeks are pink. Chubby cheeks, dimple chin. Have you read the poem? So cheeks are pink in color. You can learn a mnemonic. So cheek में H भी आता है, but neglect H. Ignore H. So these are the 4 bacteria which are pink in color, which form pink colonies on Mekong agar. So C stand for Citrobacter. The 2 E, one is E. coli and one is Enterobacter. Currently I'm teaching you E. coli. And K for Klebsiella. K for Klebsiella, which I already taught you yesterday. Okay. So these form pink colony. Remembering Klebsiella, mucoid, sticky colonies, pink color. And remaining all will come in non-lactose fermenter. I will teach you Salmonella today. Okay, I will teach you Shigella today. I will teach you Yersinia today. They all are yellow. And I will teach you E. coli, which is pink. So I'm teaching you 4 now. So this one is forming pink and the remaining 3 is forming yellow. Because this one is pink, I mean lactose fermenter, and remaining 3 are non-lactose fermenter. Among the Shigella, one of the Shigella is late lactose fermenter, so it will convert from yellow to pink later. Shigella Sony. Remaining all Shigella are yellow. So that is the classification, and that is the Mekong agar ka summary in front of you before I start the food. Okay, yes. So introduction is done, classification is done, morphology. I'm teaching you the first one, E. coli. Don't forget that. मैं E. coli पढ़ा रही हूँ. I'm teaching you the first one. Okay, tell me the morphology. It's a gram-negative bacilli. It's a member of Enterobacteriaceae. Okay, you can see it here. E. coli. It's gram-negative bacilli. It's a member of Enterobacteriaceae. In all 4, this line is same. The next four I'm teaching you. This line is same. Okay. Capsule, spore and motility. See E. coli. Write down E. coli over here. Is it coming in this mnemonic? No. Is it coming in this mnemonic? No. Is it coming in this mnemonic? Yes. Search where? Here it is E. coli. So yes, motility is positive with the peritracheous flagella. Learn it once, people. Pura kaam ho jayega. So yes, it is motile. So no capsule, no spore, but motility present with the help of peritracheous flagella, not polar flagella. You can see here. Okay. Culture, I already taught you. Lactose fermenter. So since it is lactose fermenter, it is forming pink colony. Cheek, CEEK, one of the E. E. coli. Other E is Enterobacter. So it is forming pink colony. Okay, biochemical reaction. Now all these four have a biochemical reaction in this sequence. Yesterday also I taught you this sequence. So my sequence is COICU. COICU. C for catalase, O for oxidase, I for indole, the second C for citrate, first for catalase, second for citrate, and urease. You have to learn the sequence for all five, all four bacteria. I'm teaching you four bacteria now. So learn the sequence for the four bacteria. Here the first is positive, then negative, then positive, then two negative. I know it's difficult. Okay, it is known as COICU test. COICU test. Okay, so you have to learn basically here in biochemical reaction COICU ka sequence in all four. You have to fix one sequence and you have to learn the positive, negative, because individually you can't learn. You can learn a set कि भाई यहाँ पे alternate है, यहाँ पे दो positive, दो negative, फिर positive, like that. So you have to learn the set for all four. So make one set. Make one set. The set is COICU. COICU. Make this set. Okay, so that is a biochemical reaction. I know it's more of factual. It's difficult, but you have to learn. Antigenic structure. Let me draw the antigenic structure of E. coli in front of you. I'm drawing E. coli. Okay, let me draw E. coli in front of you. Okay, so this is the bacteria. E. coli. This is the cell wall of the bacteria. Okay, in the cell wall of the bacteria, there is the antigen, O antigen. O for somatic. The first antigen is O antigen, O for somatic. Okay, second antigen is present. Second antigen, O. The second antigen is present. It is an envelope on the bacteria. This is the envelope. This was the cell wall. This is the envelope. Okay, it's not capsule. It's a non-capsulated bacteria, but it is having a thin envelope. On the envelope, K antigen is present. K is present on the envelope. The second antigen is K. K is present on envelope. O is present on cell wall. This is somatic antigen. This is known as capsular antigen. Capsular antigen. It is not capsule, but it is envelope. Okay. Same हम capsule का spelling तो C से चालू होता है. I know. But Greek language में capsule start with K. Greek language में. That's why it is K. K for capsule. Okay. The third antigen is present on the flagella. These are the flagella. On the flagella. It is H antigen. And the fourth and the last antigen is present on the fimbriae. It is F antigen. Okay, so C, O antigen present in the cell wall, K antigen present on the envelope, H present on the flagella, and F is not important, which is present on the fimbriae. Okay, so you can see somatic O antigen, capsular K antigen, flagellar H antigen, and fimbrial F antigen. All four are shown over here. Okay, so there are many type of E. coli. We write down O137, H7, K wala types. So there are many types, serotypes of E. coli based on this. What is the route of transmission? The route of transmission is again feco-oral, because E. coli is the normal common cell in the intestine. In our intestine, in our large intestine, all healthy individuals have E. coli. So the E. coli is passed in the feces. It's a common cell. Suppose this girl is passing E. coli in the feces. She is passing E. coli in the feces. Okay. And after using toilet, she is not washing the hands nicely. And with the same hand, she is eating the food. So that is known as feco-oral. Feco-oral, okay, from the feces to oral, to mouth. This is the infection that is known as endogenous infection. The own bacteria are entering into the mouth, okay, and that can cause the GIT infection. The second is ascending and catheter associated. It is causing UTI over here, okay, ascending and catheter associated. How should I explain to you? Listen. Male, female. In male, two openings are there. Okay, this is urethra, this is anal opening. In female, three openings are there. This is urethra, this is vaginal, and this is anal opening. Okay, I'm saying E. coli is present in intestine. It's common cell. It's present in normal large intestine. So anal opening, here E. coli is present. It is passed in the feces. Anal opening, E. coli is present. So once it is present in the large intestine, it do not cause any disease. It's a common cell, okay? But from here, due to contamination of the urethra with the feces, because the location is closed, it enters in the urethra and urinary tract and cause UTI. In urinary tract, it's not common cell. Okay, in large intestine, it's common cell. But once it enters the urethra, it causes UTI. Once it enters the urethra, it causes UTI. So second route, this is known as ascending route. From urethra, it will ascend upward to the kidney and cause UTI. So this is known as ascending route. You can see here, the urethra get contaminated with the feces. Okay, and it ascends upward and cause UTI. This is known as ascending route, and it can be catheter associated. So E. coli causes two diseases in human. What are the two diseases caused by E. coli? UTI. E. coli is the most common cause of UTI. I will teach you E. coli again in the last day 7 session in urogenital. Okay, E. coli causes UTI. E. coli causes diarrhea also. Currently, I am interested in diarrhea. So it causes UTI and it causes diarrhea. E. coli comes in two systems. E. coli. Okay, so currently I am interested in diarrhea. Here the infection is endogenous because of not cleaning the hands, fingers properly after using the toilets. So that is the endogenous infection. And once it enters via mouth, it can cause the infection. Okay. There are 6 types of diarrhea. How many types of diarrhea E. coli causes? E. coli causes 6 types of diarrhea. How many types of diarrhea? It is causing 6 types of diarrhea. What are the 6 types of diarrhea? Anthropathogenic E. coli, anthrotoxigenic E. coli, anthroinvasive E. coli, anthrohemorrhagic E. coli, anthroaggregative E. coli, and diffusely adherent E. coli. I guess everyone knows the full form: EPC, ETEC, EIEC, EHEC, EAEC, and DAEC. I guess everyone know. Now the most important, they all cause diarrhea. Currently, I'm interested in diarrhea. It don't cause vomiting, by the way. It cause only diarrhea. So they all cause diarrhea. I agree. They all cause diarrhea. All 6. The mechanism of all 6 is different. So in your exam, they ask you the mechanism of action of all 6. All 6 cause diarrhea, but the mechanism of 6 is different. So tell me the 6th mechanism for 6 type of diarrhea. Okay, so in one master diagram, you can see my master diagram. In one master diagram, I will show you all 6 mechanisms. So have a look. All 6 mechanisms are shown to you in one diagram. The first one is EPEC. I have shown the bacteria with red color. So can you see EPEC? Enteropathogenic E. coli. P for pathogenic. So suppose I am eating food. Okay, I am eating food which is contaminated with EPC. EPC will go inside. It will go in the lumen of my intestine. Okay, so this is the lumen of my intestine. You can see the enterocytes and this is the lumen of my intestine. So here is the EPC. Okay, via contaminated food it reach here. After reaching in the intestine, enteropathogenic E. coli destroy the villi. It causes flattening of the villi, effacement of the villi of the intestinal cell. What is the purpose of villi? Why God has given us villi? For absorption. So absorption will not occur and person have diarrhea. So EPEC causes diarrhea by destroying villi. This is the mechanism of action, number 1. The second is ETEC. Suppose I am eating anything which contains ETEC. So ETEC will reach in my intestine. You can see this is ETEC. It is reaching in my intestine. Okay, naam mein hi hai, enterotoxigenic. It will not destroy villi. It will secrete 2 toxins, ST and LT. It will secrete 2 toxins, ST, LT. ST is stable toxin, LT is labile toxin. Heat stable and heat labile, ST and LT. What does they do? After entering, their toxin enter inside the cell. Bacteria do not enter inside the cell, mind my words. This is not entering inside the cell. It is sending its toxin inside the cell. So ST and LT. LT increases cyclic AMP and ST increases cyclic GMP. Okay, you can learn the mnemonic. Local anesthesia, Sunil Gavaskar. Okay, LT increases AMP. Okay, local anesthesia. Okay, and ST increases GMP. ST increases Sunil Gavaskar. You can make some mnemonic. Okay, so they are increased inside the cell. What does they do? What does cyclic AMP and cyclic GMP will do? On the intestinal cell, there are sodium transporter. They absorb sodium. They are blocked by cyclic AMP and GMP, both of them. So sodium will not absorb. Since sodium will not absorb, water will also not absorb. Water follows sodium. So sodium and water in the lumen of the intestine, that is passed in the feces and the person have diarrhea. So this is mechanism of action. You should be able to draw the complete flow chart. Okay, all these details are given in my main course, PBL course, in which I have taken the entire microbiology in 90 hours, in which I have given the detail of each and every organism. Here is a quick revision. Okay, so all the flow charts are given over if you want the details. So this is the mechanism of action of enterotoxigenic E. coli. Got it? The third type of E. coli is entero-invasive E. coli. Let's come on the third one. The third one is entero-invasive. I for invasive. Entero-invasive E. coli. नाम में ही है. What do you mean by the term invasion? Invasion यानी घुसना, invade करना, penetrate करना. So the entero-invasive E. coli itself enter inside the cell and rupture the cell. Itself, himself enter. It don't secrete toxin. It itself, the bacteria itself enter inside the intestinal cell of human and rupture the cell. That's why here diarrhea nahi, dysentery will occur. Since the cell is rupturing, bleeding will occur and blood will come in the feces. That's why this one leads to dysentery. Okay, not the others are leading diarrhea. This one and this one leads to dysentery. The fourth one is enterohemorrhagic E. coli. What does enterohemorrhagic E. coli do? Enterohemorrhagic E. coli. It also secretes a toxin. The name of the toxin is virotoxin. VT, virotoxin. Virotoxin enters inside the cell and inhibit the protein synthesis inside the cell. Since this cell cannot form the protein, it will rupture, and again it will lead to dysentery. The fifth one is enteroaggregative E. coli. A for aggregative. Enteroaggregative E. coli. It also secretes a toxin, yeast toxin, but mechanism is unknown. By some unknown mechanism, it causes diarrhea. It is not mentioned in any of the books. Okay. And last one is diffusely adherent. See how I have drawn all these bacteria with the red dot. See how I have drawn this one. I have drawn this one differently. Okay. There is an intention. It is having many fimbriae on its surface. It is having abundant of fimbriae, pili on its surface. And that's why it will go and adhere with this cell. Adhere, chipak jayega. It will adhere. Diffusely adherent. It will adhere with this cell and make the cell non-functional. And this is how it causes diarrhea. So you should know the mechanism of action of all six. Not like this. You should be able to draw the flowchart of all. That is the mechanism of action of all six. You got it or you didn't got it? The mechanism of action of all six. I'm sorry. You can see. So what's the mechanism of action? Enteropathogenic, disturbing villi. Enterotoxigenic, secrete two toxin, ST, LT. Local anesthesia, Sunil Gavaskar. They increase cyclic AMP, cyclic GMP, which inhibit the sodium transporter, and that's why increase the sodium and water in the feces, and this is how it is causing the diarrhea. Entero-invasive, invade the cell and rupture the cell. Okay, enterohemorrhagic, secrete VT, virotoxin. Virotoxin inhibit protein synthesis and rupture the cell. So these two, you can see these two causes dysentery and other causes diarrhea. The next is enteroaggregative. It secretes yeast toxin with some unknown mechanism. It causes diarrhea. And the last one, diffusely adherent, have many fimbriae on its surface which adhere with the cell and make it non-functional. People keep responding, say something. Okay, so these are the 6 type of diarrhea caused by 6 type of E. coli with their mechanism of action. You get many MCQs also and university exam questions, short question, long question of that. So I assume you got it. Now some additional points. First you tell me their mechanism of action, then I will tell you the additional point. Enteropathogenic, pathogenic destroy villi. Okay, enterotoxigenic, toxigenic secrete toxin. It secrete two toxin, ST, LT. ST increase cyclic GMP and LT increase cyclic AMP, which inhibit the sodium transporter, and that's how they cause diarrhea. Enteroinvasive cause invasiveness. Hemorrhagic secrete virotoxin, and virotoxin inhibit the protein synthesis inside the cell and rupture the cell. Okay, enteroaggregative secrete yeast toxin. We don't know the mechanism, unknown mechanism. And diffusely adherent secrete fimbriae. It is having many fimbriae on its surface with which it will adhere with the cell and make it non-functional. Now some additional point on some of them. You tell me among the 6, which is the most common cause of traveler's diarrhea? You tell me, which is the most common cause of traveler's diarrhea? Learn T40. Most common cause of traveler's diarrhea. It's an MCQ. Traveler's diarrhea, T for P, is enterotoxigenic. T40. Enterotoxigenic is the most common cause of traveler's diarrhea. Okay. Tell me, Serini test is applied for whom? Serini test. Serini test. I will explain what is Serini test. Serini test is applied for enteroinvasive. Let me explain what is Serini test. Suppose I am having a patient in my clinic. What is the complaint of my patient? My patient is saying, doctor, I am having diarrhea and dysentery. My stool contains blood. So I am having suspicion that it can be enteroinvasive E. coli, because enteroinvasive E. coli causes dysentery. Okay, I will collect the stool sample in a sterile container and I want to know whether it is enteroinvasive, yes or no. So what I will do, I will take a guinea pig. I will take the stool sample. I will take the saline suspension. The saline suspension of the stool sample in a dropper and put few drops in the eyes of the guinea pig. Yes, the stool sample of the patient in the eyes of the guinea pig. Okay, and now there are two possibilities. Either it contain antro-invasive or it do not contain. If it contain antro-invasive, it invade everywhere. It don't only invade in intestine, it invade everywhere. Hindi में कहूँ तो हर जगह घुसता है. Everywhere it penetrate. Okay, so after you are just installing it with a dropper superficially. So if it contain antro-invasive E. coli, if the stool sample, so it will invade in the cornea and conjunctiva and it will cause keratoconjunctivitis in the eye. So after 2-3 days, the guinea pig will have keratoconjunctivitis and the test is positive. It is known as Serine test. It is known as Serine test. I hope you got it. Okay, so that is the additional finding over here. Antrohemorrhagic E. coli. Antrohemorrhagic. Hemorrhagic E. coli causes 2 additional diseases apart from diarrhea. It causes 2 additional diseases apart from diarrhea. Learn H for H for H. It causes hemorrhagic colitis and hemorrhagic uremic syndrome. H. Apart from diarrhea, it causes 2 additional disease with some other mechanism. Okay, and not all serotype. It is O157H7. O157H7 serotype of antrohemorrhagic E. coli causes these 2 diseases. So it's a very important MCQ. So these are the additional findings. You can see hemorrhagic colitis and hemorrhagic uremic syndrome caused by O157H7. So that's all. Okay, lab diagnosis. You will do the stool sample collection and the standard lab diagnosis. Treatment may, standard treatment is ORS. But if the diarrhea is very severe, like antro-invasive or antro-hemorrhagic, then give azithromycin. It's not drug of choice. In severe cases, you give azithromycin, otherwise give ORS. So that is E. coli. Prevention, no vaccine available. We are done with E. coli. Can I come on Shigella? Let's come on the next bacteria quickly. The next bacteria is Shigella. Okay, Shigella. Why it is known as Shigella? Why this was known as E? E is Escherichia, the scientist. Okay, Shigella was discovered by Shiga. The Shigella, the scientist who discovered it, Shiga. Kiyoshi Shiga, Japanese scientist. Okay, that isolated the Shigella first time and that's why it is known as Shigella. It is discovered by Shiga, Kiyoshi Shiga. Okay, so that is the importance. It is of 4 types. How many type of Shigella we have? Shigella is of 4 types. Type A, type B, type C, type D, or type 1, 2, 3, 4. The first one is Shigella dysentery. Second is Shigella flexneri. The third is Shigella boydii. And fourth is Sony. Sony is dhokebaz. Sony is late. So all the Shigella form yellow colony. The last one, initial form yellow, later on changed to pink. Okay, dhokebaz on McConkie agar. Which is most common among the 4? Most common in the world is dysentery. But in India, most common is flexneri. Most common is flexneri. Which is most virulent? Most virulent is also dysentery. Least virulent is Sony. Sony is subtle, cold, least virulent, late, like that. So you get question on most common, least common, most virulent, most common in the world, most common in the India. This is for your MCQ purpose. The 4 type of the Shigella. Coming on morphology, no need to learn. Okay, you can see it is coming in gram-negative bacilli. It's a member of Enterobacteriaceae. And if you apply Shigella here, it's negative, it's negative, it's negative. It's not coming in capsule, spore or motility. So it's triple negative. Okay, so that is the morphology. Culture media. It is one of the delicate darling. Have you watched my first session? I taught you what are delicate darlings. The delicate darlings are the delicate bacteria which need transport media. Okay, so here the transport media is buffered glycerol saline. I told you in my first session. It is buffered glycerol saline. Okay. The next is McConkie. On McConkie, all of them are yellow. Okay, non-lactose fermented except Sony, which is LLF. That is, it changed from yellow to pink. So on McConkie, we already know. What is enrichment media? The liquid media. And what is selective media? The solid media. Enrichment media is selenite F growth and gram-negative growth. This I taught you in my first session. Okay, what is the selective media? The selective media is DCA and XLD. On DCA, it is forming yellow colony. On XLD, it forms red colony. So DCA and XLD are the selective media on which Shigella grow. Only Shigella grow, others do not grow. So DCA and XLD. Shigella and Salmonella grow on DCA and XLD. So learn DCA, yellow colony, and XLD, red colony. So DCA and XLD are the selective media. Biochemical reaction, please learn the sequence. Okay, the sequence I have not changed in all 4 bacteria. It is COICU, but you have to learn this sequence. What is positive, what is negative. I know people, it's difficult. I will provide you a table. In which this sequence is filled. COICU, it is filled for the complete Enterobacteriaceae. So you can make this table and send it to me now. You can make this table. Okay. So here it is for E. coli. What is the sequence I told you? Now I'm telling you for Shigella. Then the next I will tell you for Salmonella. The next I will tell you for Yersinia. So fill this table for all these bacteria. Don't change the sequence. COICU. It is given in my master charts also. You can see that table. So you have to learn this in a sequence. There is no way, no easier way, no trick I should tell you. The only thing in the form of the table or in the form of the master chart, you can see COICO for all of them and you can compare. So that is a biochemical reaction. The complete enterobacteriaceae, COICU. Okay, antigenic structure. Now please understand, how does Shigella causes dysentery? By the way, Shigella causes dysentery like EIEC and EHEC. It causes blood in the stool. How? There are two mechanisms. See, this person is eating the food. Okay, this person is eating the food or drinking the water which is contaminated with the bacteria Shigella. Red color. I draw bacteria with red color. Okay, or I am drawing. Chalo, it is red color. So it is going inside. It is going inside. It is going inside. It is going inside. It is reaching in the intestine. It bypass acidity and reach the intestine. Now in intestine it do two things. Please understand, it do two things. This bacteria. Number one, it enter itself inside the cell and rupture the cell like EIEC. Invasion. Number one, it is doing invasion like EIEC. It invade and rupture the cell, and that's why since the cell got ruptured, it is bleeding and causing the dysentery. Number one, invasion. Number two, it secretes a toxin, Shiga toxin, and the toxin is entering inside the cell. Okay, it itself also enters the cell and standing its toxin also inside the cell. So since the toxin enters inside the cell, the Shiga toxin, this is known as Shiga toxin. Shiga toxin inhibit protein synthesis inside the cell. Since the protein synthesis is inhibited, okay, like antrohemorrhagic EHEC, again the cell will rupture and again it will bleed. So there are two mechanisms. Number one, it itself go, and number two, it will send its toxin. Okay, if it itself go, it is rupturing the cell. It is rupturing the cell. And if it is sending its toxin, the toxin is inhibiting the protein synthesis. It is inhibiting the 60S ribosome and it is inhibiting the protein synthesis, and that's why it is bleeding. So via both mechanism it causes dysentery. Double mechanism, it's a very important question. How does Shigella cause dysentery? How does Shigella cause dysentery? So learn the bacteria which cause dysentery. Okay, diarrhea may be special dysentery. The root is same. Okay, fecal-oral root. Okay, it's very powerful bacteria. Even 10 bacteria can cause the dysentery. 10 to 100 is the minimum infectiosis. It's also an MCQ. The name of the disease is Shigellosis, in which patient have diarrhea and dysentery. Patient have diarrhea and dysentery, blood and stool. That is important. Okay, patient have vomiting also. And patient have complications. Complication में GIT में toxic megacolon can occur. Metabolic में hypoglycemia and hyponatremia, because most of the sugar and most of the sodium is lost in the feces. So hypoglycemia and hyponatremia can occur. CNS में Ikiri syndrome, due to the metabolic changes in the blood. CNS में convulsions and seizures can occur, and this is known as Ikiri syndrome. So that is a complication. Shigellosis. Lab diagnostics is standard for all bacteria. I use the same. Don't write this in your exam if you're a second Prof student. Describe them. Direct microscopy में write down its gram negative bacteria, no capsule, no spore, no motility. Draw the diagram. Culture में write down transport media, buffered glycerol saline. Write down the enrichment media, okay, selenite F growth, and write down the selective media, DCA, XLD. Draw their diagrams. DCA में yellow color column, XLD में red color. So draw it, write it. Biochemical reaction में COICU का complete sequence. This is how you have to write with the diagram. Don't write just these three words. For explaining, I am saying so. Okay. Treatment में here we have drug of choice. First time I'm telling you we have drug of choice. Okay, and I guess the last time. For Shigellosis, we don't take chance. It is very severe. It's dysentery. We directly give ciprofloxacin. Ciprofloxacin, third generation cephalosporin, is the drug of choice. Otherwise, everywhere it was ORS. Here also we will give ORS. But drug of choice is ceftriaxone. Prevention, no vaccine available. Okay, so E. coli is done. Shigella is done. Now, next two are very minor. Okay, very minor. They don't ask you usually, but we will complete it quickly. Give me 1 minute. Are you getting it everything? Is this way useful for you? First, you give me your feedback. Meanwhile, I will drink water. It will take 1 minute. Okay, you write down your feedback in the comment box that how you are getting the system-wide microbiology. Huh? Yes or no? Is it a positive experience or a negative experience? You are getting them in comparative columns without skipping any information in just compact version. Yes or no? Write down your genuine feedback. I do not want the manipulation. Just to make me happy, don't write the positive thing. Whatever you feel, write down that. You are open to write. Chalo, you people are understanding. That's nice. That's good. So we are done with E. coli. We are done with Shigella. Now we are starting non-typhoidal salmonella, the third bacteria in this category, non-typhoidal salmonella. Okay. So I taught you salmonella already, typhoidal salmonella in my first lecture in CVS. My first bacteria was typhoidal salmonella only. Yes or no? Yes or no? So salmonella, I taught you there. So the same introduction. Why salmonella is known as salmonella? Because of the scientist who discovered it. The scientist who discovered it is Salmonella and Smith. To honor the salmonella, we named it salmonella. So there are two types of salmonella: typhoidal salmonella and non-typhoidal salmonella. Okay, non-typhoidal salmonella include these 5 variants. Okay, so today I am teaching you these 5. On that day, on CVS, I taught you typhoidal salmonella, which includes 4 salmonella. That is Salmonella typhi, Salmonella paratyphi A, Salmonella paratyphi B, and Salmonella paratyphi C. So these 4 are known as typhoidal salmonella. I already taught you in CVS. Watch if you have not watched that. That is more important, ultra important, make 5-star. But these one are not very important. So typhoidal salmonella cause typhoid fever or enteric fever, which we have covered in CVS. Non-typhoidal salmonella cause GIT infection. It includes Salmonella typhi murium. It's not Salmonella typhi. It's typhi murium, Salmonella enteritis, Salmonella Newport, Salmonella Heidelberg, and Salmonella doubli. Salmonella doubli. So these are less important, but for the sake of completion, quickly we will review it. Of course, it is also gram-negative bacilli, and if you see, it's also gram-negative bacilli. You can see this is non-typhoidal salmonella coming in gram-negative bacilli. It's the member of Enterobacteriaceae, and Salmonella. It's non-capsulated, non-spore forming, but see in the motility. In the motility, here is salmonella. I told you all salmonella are motile except Gallorium and Pullorum. Currently, I am not teaching you Gallorium and Pullorum. You can see in the classification there is no Gallorium and Pullorum. So these all are motile. So motility is present. No capsule, no spore, but motility is present. Motility is present with peritracheous flagella. So with the help of this mnemonic, you can make it out. McConkey you already know where is salmonella. Here is salmonella. Salmonella form yellow colonies. It is non-lactose fermenter without any exception. So they form yellow colonies. Appreciate the yellow colonies. And the selective media is XLD. Okay, XLD, like Shigella only. Shigella was forming red colony on XLD, if you remember. Here, this one is forming red colony with black center. Red colony with black center. People, I'm not only covering your university exam, I'm covering your all MCQs of NEET, PG, FMG, INI-CT. I'm challenging 100% strike rate in the MCQs also, in the university also. So I'm showing you all the diagrams. It's a subject. Microbiology is a subject which is having high load of images. Okay, that's why in my master charts I have included high definition images. In the columns, these images are small, but there is a QR code given on every page. You just scan this QR code. You will get all these images, the zoom version. Okay, the high definition version. Here the images are very small for your reference. Okay, you just scan this QR code and you will get a quiz also based on these organisms to analyze this page. Okay, so it's a perfect solution to complete the entire microbiology without touching the book and getting high rank in the microbiology. Yes, so here in XLD, in Shigella, red colony, but in Salmonella, red colony with black center. Okay, and in McConkey, of course, it's yellow. Both of them are yellow, Shigella, Salmonella, except Salmonella soni, which is late lactose fermenter. Learn the sequence and make a chart. You have to learn the sequence. Sequence is COICU. You have to learn the sequence. COICU is different for all. Antigenic structure. It is having O antigen, H antigen, but it don't have VI antigen. Have you attended my first session? Have you attended the Salmonella typhi? Huh? I taught you 3 antigens. It is having O antigen that is present on the cell wall. It is having H antigen present on the flagella, but it is having a VI antigen also that is present on the envelope. Here VI is absent. The remaining two are same as that of typhoidal salmonella. Root is same, fecal-oral. Root is same, fecal-oral. But fecal-oral mein what is the food prediction? The food prediction is poultry. Poultry yani egg. Poultry and milk. So poultry and milk is important. Okay, this is your clue in your questions, MCQ questions. So this person is eating the poultry. Root is same, fecal-oral. Okay. Clinical features mein it causes gastroenteritis in which diarrhea, vomiting both occur. You can appreciate diarrhea, vomiting both occur. After causing gastroenteritis, it goes in the blood, causing septicemia. And after that, it involves other organs also, like meningitis, osteomyelitis, arthritis. So first, it caused diarrhea and vomiting both. I'm talking about non-typhoidal salmonella, which include 5 types of salmonella. You know the names. Most important among them is salmonella typhimurium. If you can't learn all 5. Okay, I'm talking about salmonella typhimurium, not salmonella typhi. It causes gastroenteritis, in which diarrhea and vomiting both occur. After that, it goes on blood, causing bacteremia. And after that, it involves focal, different organs, meningitis, osteoarthritis, osteomyelitis. Lab diagnosis is standard. Okay, and treatment is supportive. Supportive may we give ORS, but in severe cases, we give ciprofloxacin. So that is the treatment. Prevention, no medicine available. Coming on the last bacteria of this category. After that, we have 4 more. Okay, last 2 together we will take. So 5 bacteria will be done. Okay. So we will take Yersinia enterocolytica and Yersinia pseudotuberculosis together. Okay, because everything is same in them except one of the biochemical reaction. Rest everything is same. So I will count them 2 bacteria out of 13, but we will take them together. So let's start the Yersinia. You know how many type of Yersinia is there? You know how many type of Yersinia is there? Huh? There are 3 type of Yersinia. Yersinia pestis, Yersinia pseudotuberculosis, Yersinia enterocolytica. Yes or no? Yersinia pestis causes plague. It causes plague. That I will teach you in some other system in the next 3 sessions. Yersinia pestis, the main Yersinia. I'm not teaching you that. I'm not teaching you. I'm teaching you these 2. They causes gastroenteritis, which is known as yersiniosis. So Yersinia are of 3 type. The Yersinia pestis causes plague. The remaining 2 Yersinia, that is pseudotuberculosis and enterocolytica, they cause a disease known as yersiniosis in which gastroenteritis occurs. So patient have gastroenteritis over here. Okay, morphology is same. You can see here is Yersinia, my 2 Yersinia. People, my classification is very judicious. You can count. Total I say 60. Actually, it's not 60. 58 bacteria are there to be exact. Okay, because count by count, I am sure in my count. Okay, so 58 bacteria in my syllabus. And I have written all of them. You can count if you wish. I have covered all of them in one of the 8 systems. Okay, some bacteria will come multiple times because they involve multiple systems. You can see the 2 Yersinia. These 2 Yersinia I am taking in GIT, my purple color. Okay, these 2 Yersinia enterocolytica and Yersinia pseudotuberculosis. They are gram-negative bacilli, member of Enterobacteriaceae. You can see all the members of Enterobacteriaceae. Yersinia is one of them. Got it? So, and applying the mnemonic, yes, mnemonic is tedious. Yersinia, Yersinia enterocolytica and Yersinia pseudotuberculosis. ठीक है, in the capsule here is Yersinia. Yes का Y is Yersinia. All Yersinia are capsulated. All 3 Yersinia are capsulated. So capsule present. Okay, spore, no way, no spore. But motility, people, in the motility I have not mentioned Yersinia. I have not mentioned. But if you wish, you can make it a column here and write down Yersinia pseudotuberculosis and Yersinia enterocolytica. You can elaborate this mnemonic. Okay, Yersinia are motile. Yersinia, these 2 Yersinia, not pestis. These 2 Yersinia are motile. So see the morphology. It's gram-negative bacteria. You already know that. It's capsulated. You already know that. Yes, very killer bacteria ka Y is Yersinia only. Spore, not spore, but motility present. Now you may claim कि ma'am आपके mnemonic में नहीं आ रहा. It's not coming in your mnemonic. So I am modifying myself. Okay, so kindly you can elaborate the mnemonic if you wish. But motility, why I have not included? Say ma'am, why you have not included in your mnemonic initially only? I have not included because it's not completely motile. It's partially motile. What do you mean by partially? I mean it's differential motility. It is motile at 22 degrees Celsius, but become non-motile at 37. So that's why I have not included it in the mnemonic, but it's partially motile, you can say. Differential motility. What is human body temperature? Human body temperature is 37. So inside human body, they get upward and sit quietly. They are non-motile. You can learn like that. So at 37, no motility. They are filled. But outside the human body, at 22, they are motile. So actually they are motile or not motile, you decide yourself. If you think they are partially motile, kindly include in this mnemonic. Coming on the culture, McConkey, they are non-lactose fermenter. I told you, na? They are forming yellow colonies. The selective media, take nutrient agar. Take nutrient agar and add three things in the nutrient agar. It becomes selective. What three things? Add CIN. CIN. What is CIN? C for cefsilutin. Cefsilutin, it's a chemical. Cefsilutin. I for igrasan. I for igrasan. And N for novobas. CIN. If you can't learn the full form, it's okay. You can say CIN only. In your exam, if you are writing CIN, you will get the marks in university exam. And in competitive, you can identify in the option. Okay, so CIN. These are the three chemicals you have to learn. If you add these three chemicals, not try growing any other bacteria. No bacteria will grow except the 2-year senior. Which 2-year senior? Enterocolitical and pseudotuberculosis. Okay, there is a unique feature. They grow better at 4 degree. So once you inoculate on the plate, don't keep the plate at 37. Usually we keep the plate at incubator at 37 human body temperature, so growth occur. But here the growth occur better if you keep this plate, culture media ka plate, agar plate, CIN plate into the fridge at 4 degree. So growth occur better at 4 degree, not at 37. This is known as cold enrichment, a unique property of these year senior. Give me a minute. Feeling cold. So a unique property of these 2-year senior. Got my point? So that is known as cold enrichment. So please learn cold enrichment. That is a unique that can come in the MCQ. Biochemical reaction. One biochemical reaction can differentiate the two. The name of that biochemical reaction is ornithine decarboxylase test. Ornithine decarboxylase test is positive in enterocolitica and negative in pseudotuberculosis. Make some trick and learn it. It is positive in enterocolitica and negative in pseudotuberculosis. What's the name of this test? Ornithine decarboxylase test. Okay, very important MCQ. Antigenic structure, nothing important. Root, same fecal-oral. Clinical feature, gastroenteritis. The name of the gastroenteritis is yersiniosis. It is known as yersiniosis, in which patients have only diarrhea, no vomiting. People, please learn in which of them only diarrhea, in which of them only vomiting, in which of them both. In the end, I will enumerate for all 13, but please you learn. So here only diarrhea. It is causing some complication. Enterocolitica causing terminal ileitis. It's a PYQ of INICT. Pseudotuberculosis cause mesenteric lymph node enlargement, that is mesenteric adenitis, and both of them cause pseudoappendicitis. So these three complications are ultra important for your MCQs. It is causing diarrhea, wo to easy hai. Very easy. Everyone knows it causes diarrhea. It don't cause vomiting. It causes only diarrhea. But learn the complication. You get MCQs on complication. What is the complication of enterocolitica? Enterocolitica causes terminal ileitis, infection in the terminal ileum. Okay, pseudotuberculosis causes lymph node enlargement. It don't affect ileum. It causes lymph node enlargement in the mesentery, mesenteric adenitis, and both of them cause pseudoappendicitis. So that's what you have to focus. You have to focus these three complications for MCQ, NEET, PG, FMG, like that. Lab diagnosis, standard, treatment, no specific treatment. Okay, severe cases, as usual, ciprofloxacin. So in most of them, 90% of these bacteria I am teaching you, all of them have symptomatic treatment ORS. In severe cases, we are giving ciprofloxacin in all of them. That's it. Prevention, no medicine available. So we are done with 9 bacteria. The last 4 bacteria. Okay, in these last 4 bacteria, let's start the last 4 bacteria. Okay, in these last 4 bacteria, Vibrio cholerae is the most important. Remaining 3 are very small. Okay, so let's start with Vibrio cholerae. Make a 5-star over it. The important bacteria, kindly make 5-star over it. So this is Vibrio cholerae. Okay, make a 5-star over it. So let's start Vibrio cholerae. Who discovered Vibrio cholerae? In the introduction, write down the name of the scientist, Robert Koch. Yesterday I taught you Robert Koch in some other bacteria also. TB, Mycobacterium tuberculosis. So Mycobacterium tuberculosis also discovered by Robert Koch, and Vibrio cholerae also discovered by Robert Koch. Okay, Robert Koch discovered 3 bacteria. The name of the 3 bacteria is CAT. C-A-T, cat. C-A-T, cat. Okay, it discovered TB, it discovered cholera and anthrax. Cholera, TB and anthrax. All 3 are discovered by Robert Koch. So Robert Koch is a scientist who discovered it. Now see the classification. Before I start Vibrio cholerae, you should know complete classification of Vibrio. Complete classification of Vibrio. So we divide the Vibrio into 2 categories. This classification is known as Venkataraman Ramakrishnan classification. It is known as VR classification. The scientists who have done this classification is South Indian scientist. VR classification, Venkataramana Ramakrishnan classification. Okay. We divide Vibrio into two categories based on the H antigen. Now see this diagram. The Vibrio have O antigen on the cell wall. Suppose this is a cell wall. On the cell wall, there is O antigen. Yes or no? It is having O antigen on the cell wall. On the flagella, it is having H antigen. So first, based on H antigen, based on this H antigen, we are dividing them into two categories. Based on the H antigen, we are dividing them into groups. There are two groups. Okay, the one which cause cholera are known as Vibrio cholerae. The one which do not cause cholera are known as others. I am interested in Vibrio cholerae, so I will further classify it. I will not further classify others, but you should know there are two groups based on the H antigen. Learn that based on H antigen, we are dividing into two groups, Vibrio cholerae and others. Okay, now Vibrio cholerae is further divided into serogroup. They are group, ye group hai. Now we will further divide based on O antigen. Based on O antigen into serogroup. So H, based on H, we are dividing into two types, Vibrio cholerae and others. Based on O, we are dividing into 139 types. 200 types. From O1 to O200. 200 type of O is there. How many type of O is there? O1 to O200. Out of this 200 types, serogroups, these are known as serogroups. Okay. Out of this 200 type, only two causes cholera. O1 and O139. Out of 200. The remaining are known as non-O1, non-O39. Non-O1, non-O39, whatever left from O1 to 200. Okay, the remaining 198. So these are the three serogroups. One is O1, one is O39. Main is O1. Okay, but O139 also caused some epidemics and some cases of the cholera. And remaining all are non-O1, non-O139. Remaining all out of the 200 will come here. So this serogroup is based on O antigen. The groups are based on H antigen. Serogroups are based on O antigen. Okay, here we divide Vibrio cholerae and others. Here we divide O1, O139 and non-O1, non-O139. Now here, based on the biochemical reaction, we divide into biotypes. O1 is divided into two biotypes, classical and Altor. Classical and Altor are the biotypes because they are based on, see the basis. Groups are divided based on H antigen. Serogroups are divided based on O antigen. Biotypes are based on biochemical reaction. Okay, so there are two biotypes, classical and Altor. You will say, ma'am, what is the difference between classical and Altor? This is the difference. See the biochemical reaction. Okay, you take sheep ke RBC. You know sheep, bhed, sheep is an animal. Sheep ke RBC. And grow both, classical and Altor. Classical will not rupture it. Classical causes no hemolysis. But Altor will cause rupture of sheep RBC. Take chick ka RBC. Take chick ka RBC. This also same. Classical do not rupture it, but Altor will rupture it. Altor will cause a rupture or hemolysis of both RBC. Sheep RBC and chick RBC. Okay. Now take polymyxin B. It's an antibiotic. Polymyxin B, say this one will die. It is susceptible. It will die. But this one is resistant. Altor will not die. Okay. Take phage 4. Group phage 4. It's a virus. Again, this virus will kill it. Kill the classical one. Classical one will get killed with phage 4 virus. And this one is resistant. But take phage 5. Phage 5 said this one is resistant. And this one is susceptible. Okay, these all are your MCQs, people. I'm helpless. I have to tell. Huh? And VP. This one is VP negative, VP positive. So these are the biochemical reaction. They're easy, not very difficult if you have understood it. Based on these biochemical reaction, we are dividing into classical and Altor. Can we revise biochemical reaction? Sheep ka RBC. This one do not cause lysis, this causes lysis. Chick ka RBC. This do not cause lysis, this causes lysis. Okay, polymyxin B, phage 4. This one is susceptible, this one is resistant. But phage 5, this one is resistant, this one is susceptible. And VP, this one is negative, this one is positive. Like that. Okay, so that is the biotypes. Okay, now both of them are divided into 3-3 serotype. The last one is the serotype. Both of them divided into 3-3 serotype. Ogawa, Inaba, Hikkojima. You will say, ma'am, what is this? Ogawa, Igawa, Hikkojima. Listen. All of them are O1, na? So they all have O1 on them. O1 is the major antigen. O1. Okay, they all have O1 on that. These are the types of O1. Serotypes of O1. This is major antigen. Now there are three minor antigen A, B, C. If it is having A and B, it is known as Ogawa. it is known as A and C. If it is having A and C, it is known as Inaba. And it is having all ABC. A, B, C, all three. It is known as Kojima. You got my point? This is what I am saying. If it is having AB, it is known as Ogawa. It is having AC, it is Inaba. It is having ABC, all three, it is known as Kojima. So this is based on the minor antigen. So see the classification last time. Complete classification. We are dividing Vibrio into groups, into serogroups, into biotypes, into serotypes. First you learn this, the 4 categories. See my yellow color over here. Groups are based on H antigen. Okay. Serogroups are based on O antigen. Biotypes are based on biochemical properties, biochemical reaction. And serotypes are based on minor ABC antigen. Now you see this classification, you get many questions and you get a big long question in your university exam. Write Venkataramana Ramakrishnan classification in your university exam. VR classification. This is what you are supposed to draw. Got it? So we are done with the classification of the Vibrio. Okay. After that, let's come on the morphology. I guess everyone knows the morphology. I will tell you the extra points. Morphology mein first you tell me. See where it is coming. It's a gram-negative bacilli. Where is Vibrio? Here is Vibrio. So of course everyone knows it's a gram-negative bacilli. Apply it here. Vibrio cholerae. Vibrio cholerae. Is it coming in capsule? I don't think so. Vibrio parahemolyticus come. Mind my words, huh? Yes, very killer bacteria ka V is Vibrio parahemolyticus. It is Vibrio parahemolyticus. So Vibrio parahemolyticus is coming, but cholerae is not coming. Currently I am teaching you cholerae, not parahemolyticus. Yes. So it is negative for capsule. Negative for spores also. What about motility? Oh my God. What about motility? It is motile. It is motile. Motile by polar flagella. Motile by polar flagella. Not only motile. I told you out of these 12, 4 of them have unique motility if you have attended my first session. It is having darting motility. Shooting star. Darting. Have I taught you? So same thing is written over here. You can see. It is a gram-negative bacteria. But it is coma shape. English mein how we apply comma? Comma. We apply comma na in English. Comma. So you can appreciate it as coma. It is coma shape. Appreciate the coma shape. Okay. Capsule negative, spore negative, motility positive. Motility by polar flagella and it's darting. Darting means shooting star. See this video. You cannot trace it. You cannot trace it. Shooting star, toota tara. It's seen, it's disappear. It's shooting star, darting mortality. Have you seen fishes? How does fish travel? In a stream, in a lake, fish lake. Hai kabhi? Fish travels in group. So in the blood, in human blood, they travel in small groups. Small groups. So it is known as fish in stream appearance. What it is known as? Fish in stream appearance. And it is also known as swarm of genets. Genets are birds. They also travel in groups, small groups. So since it is in small groups, it is known as swarm of genet or fish in stream appearance. So this is the extra findings you can write down in the morphology. So see the complete morphology in front of you. Appreciate the morphology of Vibrio cholerae. We have seen the morphology. After that, come on the culture. Again, it's a delicate darling. Remember my first session. It's a delicate darling. Yes or no? It's a delicate darling. So transport media is required like Shigella. In Shigella, it was buffered glycerol saline. Here it is Venkataraman Ramakrishnan media, VR media and CB, Carrie Blair media. VR, CB. Watch my first session, people. Watch it. Okay. Enrichment media is liquid and selective media is solid. Yes or no? So liquid media is alkaline, peptone, water. I taught you in the first session. And selective media are 3. What are the 3 selective media? BSA, Gutta and TCBS. See the full form? What is BSA? Bile salt agar. BSA. What is Gutta? Gutta is gelatin, telluride, trypticase telluride agar. Gelatin taurocolate. Gelatin taurocolate, trypticase telluride agar. And TCBS is thiosulfate citrate bile salt agar. If you are not learning the full form, it's okay. We are adding all these things in the nutrient agar. Take nutrient agar, add bile and salt. No other bacteria will grow, only Vibrio grow. Again, take nutrient agar, add Gutta, add gelatin, taurocolate, trypticase, telluride. No other bacteria will grow, only Vibrio will grow. And again, take nutrient agar and add thiosulfate, citrate, bile salt. So no other bacteria will grow, only Vibrio will grow. So we are taking nutrient agar and adding all these bluff. Okay, all these things. So that only Vibrio will grow, others will not grow and it becomes selective. So learn the 3 selective media, BSA, Gutta and TCBS. BSA, Gutta and TCBS. If you're not learning the full form, you can make an assumption in the MCQs. And in the university exam, it's okay if you're writing these names only. BSA, Gutta, TCBS. At least you will get good marks, if not full. Okay, so this is BSA, translucent colony. This is Gatta, in which colony with a center, black center. And this is TCBS, TCBS with yellow colony. I always show you the images so that image-based question also should be correct. Okay, biochemical reaction, you have to learn. So C, it is catalase. Catalase को अलग छोड़ दो. You can arrange in the same sequence. It is catalase positive, pergolase positive, indole positive, citrate variable, urease negative. But the unique feature here is string test. It's sticky like Klebsiella. So this is the colony. This is the colony. Take a loop. Suppose this is a loop. I am touching the colony and going above. So it is forming a string. Okay, like a Fevicol. Okay, so the colony is very sticky. If I touch it, it will form a string. String test positive like the Klebsiella pneumoniae. In Klebsiella also string test is positive. I taught you yesterday. Yes, antigenic or virulence factor. Virulence factor are two. One is toxin. Both are toxin. The main toxin is cholera toxin. And the minor toxin is zona occludens toxin. But there is a question in INICT, recent INICT on this. So you should know both the toxin. Main toxin is cholera toxin. Suppose I am a human. Actually, not suppose. I am actually a human. But suppose I am eating something which is contaminated with the Vibrio cholerae. Suppose. Okay. So I am eating something which is contaminated with Vibrio cholerae. The food or water. Okay. So Vibrio cholerae will go inside. The Vibrio cholerae will go inside. It will go inside. It will go inside. Here is Vibrio cholerae. Okay, it will secrete two toxin in my intestine. Name the two toxin. These are the two toxin it is secreting. See the two toxin. What is the name of these two toxins? Say. So it is secreting two toxin. One is cholera toxin and other is the zona occludens toxin. Okay, zona occludens is very simple. What does zona occludens toxin will do? Zona occludens toxin disturb the junctions between the intestinal cell. So it disturbs the junctions between the intestinal cells. The junctions between the intestinal cells will be disturbed. That was an MCQ. And that's why the person had diarrhea. No dysentery, only diarrhea. That is zona occludens. Okay, what does cholera toxin will do? Cholera toxin enter inside the cell and increase cyclic AMP. It increase cyclic AMP. It increase cyclic AMP like the LT toxin of E. coli. LT toxin of ETEC. Yaad aaya? It increases the cyclic AMP. The same will occur. Cyclic AMP, once it is increased, it will block the sodium transporter. Sodium will remain in the lumen, water will remain in the lumen, and it will cause watery diarrhea. It will cause typical watery diarrhea. You got my point? So same mechanism is shown. You can see cholera toxin. Cholera toxin enter inside the human cell, stimulating adenyl cyclase. It is converting ATP to cyclic AMP. So cyclic AMP is increased. Cyclic AMP is inhibiting the sodium and chloride channels. So sodium and chloride will remain in the lumen. Water will remain in the lumen, and that will lead to watery diarrhea. I hope you got it. Mode of transmission is same. It is ingestion of contaminated food and water. You can learn the minimum dose. It's the PYQ. The minimum dose of Shigella was 10 to 100 bacteria. Here the minimum dose is 10 to the power 8 bacteria. Okay. Clinical feature, it causes a disease. The name of the disease is cholera. In cholera, patient have only diarrhea. Only diarrhea. It is known as watery diarrhea. No vomiting. Only diarrhea. Only diarrhea. Okay. What's the color of the stools? Collect the stools in a container. Collect the stool of this person in a container. See the color of stool. Is it yellow? No. Is it white? No. Is it transparent? No. Because it's buff color. Buff color. It's a bad simile. Have you cooked rice? Have you cooked rice in your kitchen somewhere, or have you seen how rice are cooked? Before we cook rice, we wash the rice in water and then we cook it, obviously. So the water in which we wash the rice, the color is looking like that. It's a bad simile. That's why it is known as rice water stool. Typically, cholera have rice water stool. It is having only diarrhea, no vomiting, no fever. Fever is also absent. So that is cholera. Lab diagnostics is standard, nothing important. And treatment, supportive में ORS, severe to azithromycin. Okay, macrolide. Severe में not third generation cephalosporin. You have to learn in severe gastroenteritis in which disease what we are giving. In some of them we are giving third generation cephalosporin. In some of them we will give macrolide. So here we will prefer macrolide. Now first time I am telling you vaccine is available. Here vaccine is available for cholera. Okay, first of all, for chemoprophylaxis, tetracycline can be given to the close contact. Suppose one of the child is having cholera and the mother is in close contact. Now mother is afraid that I will also have it. So we can give tetracycline to the mother. Okay, we can give tetracycline to the mother. So for chemoprophylaxis to the close contact who are already exposed, we can give tetracycline. Now two type of vaccines are available for vaccine. One is killed whole vaccine and one is live vaccine. One is killed vaccine, one is live vaccine. The killed vaccine brands are Shanchol and Uvitol. And live vaccine is Vaxcora. Vaxcora. This is the brand. CDV 103 HGR. This is a strain. Learn all strains of vaccine at one place. Here 2 doses are given. Here single doses given. Both are oral. Oral drops like polio boon zindagi ki. So both are oral. Both are oral, not injectable. So that is the vaccine, the simplest way I can tell you. So the biggest bacteria is done. Now next 3 are very small. They are minor bacteria. So quickly we will flip it. Okay, the last 3 bacteria out of the 13, 10 are done. Next 3 I will quickly finish it. So let's come on the next bacteria, H. pylori. Okay, now H. pylori and this is Helicobacter pylori and this is Campylobacter jejuni. These are twin brothers. Both of them are twin brothers. Okay, I mean most of the things of both of them are same. So Helicobacter, Campylobacter. Quickly we will finish them. Helicobacter followed by Campylobacter. So coming on Helicobacter. Helicobacter colonize only stomach. It affects only stomach. One organ in human body, that organ is stomach. So it affects only stomach. Okay, coming on the morphology, you can see here is Helicobacter. Okay, so it's also gram-negative bacilla, but not a part of a member of Enterobacteriaceae. Okay, the shape is very typical. The shape of Vibrio was coma shape. The shape of Vibrio cholerae is coma shape. Okay, Helicobacter pylori and Campylobacter jejuni are like flying birds. Have you seen a flying bird? So this shape is there. Vibrio cholerae is coma shape. These 2 brothers, okay, they are flying birds. So it is known as seagull shape. You can appreciate the seagull shape over here. Appreciate the seagull shape. Appreciate. It's like this. If you can see, it is slightly like this. It is known as seagull shape. That can be the MCQ. Now see, you can apply Helicobacter pylori. No capsule, okay, no spore, but motility. Yes, both the brothers have motility. Where are both the brothers? Helicobacter, Campylobacter. Both, this one and the next one. Okay, both of them are motile with polar flagella. Both of them are motile with polar flagella. So they are motile with polar flagella. Okay, and have I taught you the mnemonic of microaerophilic? The bacteria which require just 5% oxygen. Bataya hai kya maine? Have I taught you in my first session? Who are obligate aerobe, who are obligate anaerobe, who are microaerophilic? If you remember my first session, the mnemonic is hell. The camp is boring. So what is hell? Helicobacter. What is camp? Campylobacter. Both brothers are microaerophilic. That's why I'm saying they are exactly same. Both of them same shape, same capsule, same spore, same motility. Both of them are microaerophilic. In Campylobacter also same page will come. Okay. So morphology is done. Okay. After morphology, culture. Selective media is Q media. I don't know the composition. Kindly check and let me know also. You should know the composition. What we are adding in the blood agar or nutrient agar. We take blood agar or we take nutrient agar. We add something in that. So that other bacteria do not grow. Only this bacteria will grow. So the name of the media is Q. Screw media. But actually I do not know the composition. I will not bluff. So kindly check it and write it down. That is the culture. Biochemical reaction. Both are catalase positive. Both are oxidase positive. Don't learn it. How to differentiate the 2 brothers, Helicobacter from Campylobacter. So Helicobacter is urease positive, hippurate hydrolysis negative. Campylobacter is urease negative, but hippurate positive. So please, in comparative chart, it's very easy for you to compare the 2. Got my point? So this one is urease positive, hippurate negative. This one is urease negative, hippurate positive. So that is the biochemical reaction. This is how you can differentiate the 2 brothers. Now, antigenic or virulence factor, H. pylori. I am teaching you H. pylori, Helicobacter right now. It is having 2 genes, VAC and CAG. VAC A, CAG A. Very important for short questions and MCQs. It is having 2 genes, VAC A and CAG A. So it forms 2 proteins. Suppose my stomach, this is my stomach. This is the stomach of a human. And it contains the H. pylori. Can you see the yellow color mucus lining? You know our stomach is having mucus or bicarbonate lining. This mucus and bicarbonate lining is having the H. pylori. It is having the H. pylori. Okay, it is impacted with H. pylori. You can see the red color is the H. pylori. Now it will form 2 proteins. It will form VAC and CAG. What does VAC will do? VAC protein will form vacuoles in the cell. VAC for vacuole formation. So in the human stomach cell, vacuoles are formed. Vacuoles are formed in the cytoplasm. VAC for vacuole. Okay, and what does the second one? CAG will do. This is VAC. CAG will produce inflammatory cytokines and cause the inflammation of these cells. It will produce pro-inflammatory cytokines and cause the inflammation. Not only this, it will cause expression of proto-oncogene. It's a oncogenic bacteria. Yesterday I taught you an oncogenic virus. Which virus I taught you? Oncogenic, which cause cancer in human. I taught you EBV, if you remember. Epstein-Barr virus. Yesterday I taught you. It was an oncogenic virus which caused cancer in human. Burkitt lymphoma. This one is oncogenic bacteria. It caused cancer in stomach. Because of the CAG, it caused cancer. Because of VAC, it is causing vacuoles. Because of CAG, it is proto-oncogene expression. And it caused cancer. Mode of transmission is fecal-oral for all of them. Clinical feature, it caused 5 disease. 4 of them are benign. You can see. 1 of them is malignant. H. pylori. It caused 4 diseases which are benign. 1 is malignant. All the 5 diseases in stomach. All 5 diseases in stomach. You can see H. pylori in stomach. It affects only one organ, I told you. It colonized stomach. In the introduction only I told you. It colonized stomach. It colonized stomach. Okay. It causes acute gastritis, chronic gastritis, autoimmune gastritis. So 3 to gastritis हो गए. 3 type of gastritis. Acute, chronic and autoimmune. And it causes peptic ulcer. It causes peptic ulcer. So that is the gastritis and the peptic ulcer. Okay. Now it causes cancer in the stomach because of the CAG gene. CAG-A gene. Okay, it causes 2 cancers in the stomach. It can cause adenocarcinoma in the stomach and it can cause lymphoma of the stomach. The lymphoma of the stomach is known as maltoma. So it is causing 2 type of malignancies in the stomach. Please learn that. Okay, lab diagnosis. What sample you will take? What sample? It is not causing diarrhea or vomiting. Mind it. H. pylori, no diarrhea, no vomiting. It is causing gastritis. The first bacteria I am teaching you, which is not neither causing diarrhea nor causing vomiting. It is causing something else. It is a gastrointestinal infection only, but it is not diarrhea. It is not vomiting. Huh? It is not both of them. It is causing gastritis. So what sample you will take? You cannot take stool. You cannot take vomiters. You have to take stomach biopsy. Okay, so there are 2 type of tests. Invasive, non-invasive. In invasive, do the stomach biopsy. Okay, using a endoscope, take a part of stomach, a chunk of stomach biopsy. After taking the biopsy out, you do the gram stain. Okay, you do the screw culture on the screw medium culture. Okay, you do gram stain, you do culture, and there is a special stain for H. pylori, which is known as Warden-Starry. Very important MCQ. You can do Warden-Starry stain on the biopsy that will highlight the bacteria with the brown color. Warden-Starry stain. But person do not like this test. Patient do not like because it is invasive, na? It's painful, costly. Person have to admit for cutting a portion of the stomach. It's invasive. Patient do not like it unless until it's malignancy. For benign thing for gastritis, patient do not want invasive test. So we have a non-invasive test also. Name of the non-invasive test is urea breath test. It is urea breath test. What is urea breath test? Quickly, I will tell you here. Here the patient do not want to do the biopsy. Okay. Suppose this patient coming to me. I'm a doctor and patient is saying, doctor, I'm having dyspepsia. I'm having burning sensation in the stomach, loss of appetite. Whenever I eat anything spicy, I'm having burning and dyspepsia. So I'm having suspicion the patient is having gastritis. Okay. And maybe it is due to H. pylori. Gastritis ke alag reasons. I mean, without H. pylori also gastritis can happen. But suppose it is due to H. pylori, I want to get sure. So I will ask the person, let's biopsy your stomach. A portion I will take it out, a small chunk, and we will do the culture, gram staining and Warden-Starry stain, and diagnosis will be confirmed. Whether it is H. pylori or something else. Patient say, no, doctor, some invasive test, I'm getting afraid. It's costly, I have to give anesthesia and all. No, some non-invasive test. So I will offer, okay, we will do urea breath test. Okay, what we will do in urea breath test? In urea breath test, I will give you a medicine to drink. Will you drink that medicine? No injection. Only medicine. Patient say, okay, I will drink the medicine. What medicine you will give? You will give urea to breathe. Urea, commercially available urea. Urea contains carbon. You know the chemical formula of urea? I don't remember, but it is organic compound. It contains carbon. Okay. Normal carbon is C12, but this contains C13 carbon. It's a special urea. C13 urea. Ask the person to drink it. So person is drinking the C13 urea. So C13 urea is going in the stomach. C13 urea is going in the stomach. Yes or no? Yes, it is going in the stomach. So the C13 urea, which contains C13 carbon, is in the stomach. Now there are 2 possibilities. Either the stomach have H. pylori or stomach do not have H. pylori. That's what I want to know that now. Either the stomach have H. pylori or do not have H. pylori. That's what I want to know. Suppose my suspicion is right and the stomach have H. pylori. You can see. So H. pylori is urease positive. H. pylori secrete urease enzyme. Urease enzyme that will degrade the urea. That will degrade the urea into water and carbon dioxide. So here water and carbon dioxide is formed. Water and carbon dioxide. This carbon dioxide ka carbon is C13 carbon because it's a metabolite of urea. This water and carbon dioxide will go in the blood. Water also go in the blood and CO2, C13 wala CO2 also go in blood. Water excreted out of the kidney. Okay, CO2 is not excreted in urine. Okay, CO2 is a gas. It is excreted via lungs. It is exhaled in lungs. We inhale and we exhale. So carbon dioxide is exhaled. So here C13 CO2 will come. Okay, and we have a detector, a small instrument, in which we will ask the patient to exhale in that instrument. So if it contains C13 carbon, the instrument will give us a flickering. It will give a buzzer. Yes, the breath contains C13. Currently, if you test my breath contains CO2. We all exhale CO2, you, me, everything. But it's not C13, it's C12. It's C12 CO2. Okay, normal CO2. But if I drink urea and my stomach contain H. pylori, then my breath can contain C13 CO2. So this is how we detect it. So that's why it is known as urea breath test. See, the person is exhaling in the instrument and it will give a buzzer if it contains C13 CO2. Okay, so that's why urea. Drink the urea and breathe out after 10 minutes. Drink the urea, after 10 minutes, breathe in the instrument. Like that, exhale in the instrument. So that is urea breath test. You should write down the steps. Okay, urea breath test. What's the treatment? We have drug of choice. No ORS, it is gastritis. It is gastritis. For malignancy, we give chemotherapy, radiotherapy, surgery and all. But for gastritis, we give triple therapy. The drug of choice is triple therapy. I teach you this in pharmacology also. Yeah? Triple therapy. So OCM or OC, one of the two triple therapy. O in both of them stand for omeprazole. It is one of the PPI. I guess you know what is PPI. C is clarithromycin. It is one of the macrolide. Okay. And either it is M, that is metronidazole, or it is A, that is amoxicillin. So either OCM or OCA. It is very important in pharmacology also. Learn the triple therapy. Prevention, no vaccine available. So H. pylori is done. Coming on the next, uska bhai, twin brother. Helicobacter is done. Coming on Campylobacter. Let's move on the Campylobacter. Okay, Campylobacter, it causes diarrhea, but it causes systemic disease also. Okay, morpho is same as H. pylori. C, it's gram-negative bacteria. It's also seagull. Okay, no capsule, no spore, but motility with polar flagella. It also darting like the Vibrio cholerae. It also microaerophilic. You can see, hell, the camp. Camp is boring. Camp ka C is Campylobacter. So you understood the morphology. Culture media is same. Screw media. Culture is same. Biochemical with two differences are there. That was urease positive, hippurate negative. This is urease negative, hippurate positive. Okay, catalase oxidase is same in both of them. Please highlight urease and hippurate. H. pylori is urease positive, and that is the basis of urea breath test. Urea breath test positive in H. pylori, not here, because this one is urease negative. This one is hippurate positive. Okay, so please learn these two. Antigenic structure, it secrete toxin, enterotoxin and cytotoxin, two type of toxin. Okay, clinical feature may it cause only diarrhea. Okay, H. pylori was causing gastritis. This one causes diarrhea. This one causes diarrhea. After that, it goes in blood, and after blood, it goes in brain also. It don't only stop in GID like the Yersinia. I told you, no, they also cause systemic infection. So it is causing systemic infection also. Lab diagnosis standard, treatment ORS. If it is severe, it's macrolide. You got the pattern. Prevention may no vaccine available. Coming on the last bacteria. 13th bacteria, the last bacteria, Clostridium difficile. I already taught you two Clostridium. I taught you Clostridium perfringens and Clostridium botulinum. Okay, in food poisoning, my first master chart. Okay, here I'm teaching you Clostridium difficile. It's not a food poisoning, it's a unique bacteria which cause a unique infection. It is associated with prolonged use of antibiotic. If the person is taking antibiotics since many days, maybe since 15, 20 days, so you are doctor now, you give antibiotic only if it is necessary. I will tell you the reason. First, you tell me the morphology. Where is Clostridium? Here is Clostridium. All Clostridium are gram-positive bacilli, ABC ka C. Okay, Clostridium difficile. See its morphology. Clostridium difficile. Capsule negative, spore present. You can see all Clostridia are spore forming, and motility also present. All Clostridia are motile except perfringens. This is not perfringens, this is difficile. So you can see capsule negative, spore present, motility present, and it's the obligate anaerobe. No, can't breathe air ka C hai. It's obligate anaerobe. I guess everyone knows. It's obligate anaerobe. Culture media, the selective media for Clostridium difficile. Okay, you take two nutrient agar. nutrient agar. You got the pattern now. Add something so that only this bacteria can grow. Only Clostridium difficile grow, others do not grow. So you add these three things: cefoxitin, okay, cyclocerin, and fructose agar. So it is known as CCF agar. Okay, CCF agar. Cefoxitin, cyclocerin, and fructose. Here the two things are same. Cefoxitin, cyclocerin, but instead of fructose, here I am adding egg yolk. Egg yolk. So either CCF or CCY. Okay, F is fructose and Y is yolk, egg yolk. CC is same. Okay, so CCF, CCY. In short, you can learn the selective media for Clostridium difficile. Not try growing any other bacteria, it will not grow, only Clostridium difficile will grow. Biochemical reaction, gelatin hydrolysis. It is having an enzyme gelatinase. That's why it hydrolyzes the gelatin. Okay, now the antigenic structure. Listen, very important. The root is same. Most important thing, please understand. We all have colon. You understand? Yes. Just a second. See, we all have colon. Of course, we all have colon. In the colon, we have good bacteria. You know, in the colon, we have good bacteria. The good bacteria are known as commensal. The good bacteria are known as commensals. The good bacteria are known as commensals. They are good bacteria. What good they do for us? Why we call them good bacteria? They are like gods. They inhibit the entry of harmful bacteria. If some harmful bacteria try to enter, they are like gods. Thankfully, God has given these commensals in us. Now, suppose there is a patient coming in your clinic and saying, doctor, I'm having some bacterial infection, maybe pneumonia, maybe abscess, maybe any bacterial infection, any bacterial infection. What you will do? You will give antibiotic. So you give antibiotic only if necessary. What is antibiotic? Antibiotic is a drug that kills bacteria. Antibiotic. It's a drug that kills the bacteria. Okay, it kills the bacteria. It don't differentiate good or bad. So bad का तो क्या हुआ मुझे नहीं पता. I don't know whether it is killed the bad or not. But if you give a prolonged course of antibiotic, the good bacteria will also get killed. The commensals will be killed. So if you are giving a prolonged course of antibiotic to any patient, the commensals in the intestine, the large intestine, are dead. Okay, now there is no one to inhibit the entry of harmful bacteria. In that case, Clostridium difficile will enter. Otherwise, Clostridium difficile cannot cause infection. In me, I'm not an antibiotic right now. Okay, Clostridium difficile cannot enter. But if I take the antibiotic for 15, 20, 30 days, a prolonged course, my all commensals are dead, no guards, that Clostridium difficile can enter inside me very easily. After entering inside me, it secretes two toxins in my intestine, enterotoxin and cytotoxin, that cause necrosis of the colon. That cause necrosis of the colon, the membrane of the colon. And here a pseudomembrane is formed. A green color pseudomembrane is formed. A green color pseudomembrane is formed. It is known as pseudomembrane colitis. What it is known as? Pseudomembrane. Pseudomembrane in the tonsil, diphtheria. I told you gray color pseudomembrane in the tonsil yesterday. Pseudomembrane in the colon, Clostridium difficile. So that is a disease. It is known as pseudomembrane colitis, also known as antibiotic associated colitis, in which the colon ke andar pseudomembrane is formed. Patient have abdominal pain and diarrhea. Appreciate the endoscopy. In the endoscopy, appreciate the green color pseudomembrane in the colon. Okay. Lab diagnosis, collect the stool sample. Okay, collect the stool sample. In the stool, look for the toxin. Look for the toxin either with ELISA or with PCR. Or do colonoscopy. In the colonoscopy, you will get the green color membrane. So that is a lab diagnosis. Treatment, we have a drug of choice. Don't give ORS over here. We have a drug of choice. The drug of choice, very important PYQ, repeated many times recently. It is fidaxomicin. It's a newer antibiotic discovered. It is fidaxomicin or vancomycin. If fidaxomicin given in the option, go with that. If not given, go with vancomycin. So that is a treatment. Prevention may antibiotic stewardship. That is, give antibiotic only if necessary for the minimum duration at the minimum dose. Okay, give minimum dose, minimum duration only if necessary. Don't do unnecessary use of antibiotic. It can lead to pseudomembrane colitis. People, I know it took time. It took 3 hours. I assume 2 or 2.5 hours. But it took 3 hours. But I am satisfied. I taught you 13 bacteria without skimming even a single information. You can see all 13 bacteria in 3 master chart. First master chart, food poisoning. Second master chart, Enterobacteriaceae family bacteria. And third master chart, other miscellaneous infections. Okay, so I hope, I wish if I could have more time, I fill these 3 master charts in front of you, because since the time is limited and it's a rapid revision, I cannot do so. But I can show you. I can show you over here. So in my master chart, I have already done my homework. Okay, you can see this is my first master chart containing the 4 bacteria which cause food poisoning. This one, the empty one here, the filled one is here. You can see the same 4 bacteria. You can see this is the next master chart containing the bacteria of Enterobacteriaceae. Appreciate these 4 with the images and everything. So you can compare the drug of choice, compare the lab diagnosis, compare the clinical feature, which causing only diarrhea, which causing only vomiting, which causing both, which causing gastritis, like that. Okay, so it's very easy. First read column wise, then read row wise. Follow it. Okay, and these are the miscellaneous 4. Okay, the remaining 4 bacteria which are there here. You can see the remaining 4 bacteria. Okay, I have given the numbers A, B, C, so you can understand the bacterial infection of GIT A, B, C. This is the C, and there is a QR code everywhere for seeing the high definition images. Now let's start the viruses. Only single virus we are having, rotavirus. It's a small virus. It's a small virus we are having, rotavirus. Quickly in 5 minutes we will finish it. Okay, after that I will take a break of 5 minutes and we will finish parasites after that. Chalo. So can we come here? Okay, chalo. So come on the viral infection of GIT. We are done with bacterial. Viral infection of GIT, only one virus. The name of the virus is rotavirus. Okay, you can see here. I cannot start directly. First I will go with the classification. You know the classification of viruses? Of course you do. Okay, there are DNA viruses, 6 families. RNA viruses, 14 families. So there are 38 viruses in our syllabus. I will teach you all. Don't worry. Some are already done in CVS and RISP. Some will be done ahead. But today I will teach you one. Only one out of 38. Where is my one? Where is my purple one? The GIT one. Where is it? Here it is. So is it DNA or RNA? So you can see it is RNA. It is rotavirus belonging to Rheoviridae. See the family Rheo. So one of the R. A, B, C, D, F, O, R, T, P, H ka one of the R. Rheoviridae. Okay. So it is belonging to Rheoviridae. So see my classification. Look for purple. You will get only one purple. I'm sure you will get only one. I think so. You will get only one purple. So appreciate. You can see this or you can see this. The same. Appreciate only one purple. Can you see? This one. This one. I don't think colors are very good visible to you, but these are on the glossy paper. The colors are like brilliant shiny. Okay, so we choose paper, we choose print, everything of the best quality so that you have the best experiences of these master charts, the color coded master chart, and it will bring a revolution in the subject microbiology in the next few months or years, I believe. Anyways, so you can see this is rotavirus. This is rotavirus. You can see the purple one. So let's start. You know my fixed set of headings in the virology. A small virus, rotavirus. In the introduction, we always write down its family. What is the family? I already taught you it's Rheoviridae. It is the most common cause of diarrhea in children. Most common cause. Most. Whenever it's most common anywhere, highlight it. So Rheovirus, rotavirus. Okay. It is the most common cause of diarrhea in children. Morphology में my fixed set of heading for virus. For bacteria, it was gram stain, capsule, spore, motility. Here, it's nucleic acid, envelope, symmetry, shape, and something extra. So you know my fixed set of heading. Everything is fixed. Okay. So here, nucleic acid, it's RNA. All RNA. All RNA are single-stranded. Come on the classification again. All DNA are double-stranded. Have I taught you this? Except parvo, which is single-stranded. I taught you this on day 1. All RNA are single-stranded except one dhokebaz. Who's that? Rota, which is double-stranded RNA. So that is a unique feature here I have written. Highlight the word double-stranded RNA. You will say, RNA भी double-stranded होता है क्या? Yes, it can happen in Rheovirus, rotavirus. Double-stranded RNA. Okay, not only this, it is coming in Bora also. I taught you on day 1. 4 of the RNA viruses having fragmented RNA. So it is Bunia, B-O-R-A. Bunia, orthomixo, Rheo, and Arena. You can see here 11 pieces. 3, 8, 3 plus 8 is equal to 11. And 1 plus 1 is equal to 2. So you can count the number of pieces. It will be 10 to 12 or 11. You can count it. If you wish, you can count it. This is the diagram. Image-based question is very frequently asked on this. Because of this fragmentation, it is looking like a wheel. These are looking like spokes of wheel. It is looking like a wheel. So the shape is wheel. Symmetry is icosahedral. It is having 6 structural and 6 non-structural proteins. The structural proteins are VP1 to VP7. VP5 don't exist. You can count VP1 to VP7. And non-structural proteins are NSP1 to 6. So VP1 to VP7, no VP5, and NSP1 to NSP6. You can appreciate the diagram. See, it's double-stranded RNA. See, it is 11 fragmented RNA. Two things are unique. 11 fragments and double-stranded. Highlight it for your MCQs also, for universities also. And wheel shape, highlight it. And the proteins, highlighted. So morphology is very important. You are asked to draw the diagram of rotavirus. Okay, you can see. Mode of transmission, fecal-oral for all of them. Pathogenesis. Suppose I am eating something, the food and the water, which is contaminated with rotavirus. Rotavirus go inside, reach in the intestine. It enter inside the intestinal cell. Okay, because viruses enter only in living cell. You know this rule? Day one, you know this rule? So viruses enter inside the human cell, intestinal cell, and multiply. They multiply in the cytoplasm of human intestinal cell. They are multiplying over here. They are multiplying. When they multiply, they inhibit the transport mechanism or the transport mechanism of the cell. The transport mechanism is gone. So these cells cannot absorb anything. These cells cannot absorb anything and the person will have diarrhea. Person will have watery diarrhea, secretory diarrhea because absorption is not there. They inhibit the transport mechanism of these enterocytes. So this is the mechanism of action or pathogenesis, you can say. Okay, clinical feature, acute gastroenteritis. Person have diarrhea and vomiting both. Diarrhea, vomiting, abdomen both. So please mind where it is diarrhea, where it is vomiting, where it is both. Okay, so please see. I have used these two images only in all my master chart. So you can compare. Here only this image, here only this image, here both the images. You can compare like that. So this is the clinical features. Okay, lab diagnosis. Standard lab diagnosis for virus. For virus, I told you, no, we do serology, antigen detection, and we do PCR or RT-PCR. Since it is a RNA virus, we will do RT-PCR. For DNA virus, we do PCR. On electron microscopy, we see the wheel shape. Appreciate the real image. Appreciate the wheel shape because of 11 fragments, 10 to 12 fragments. It is looking like a wheel shape, spokes, the spokes of a wheel. Appreciate. Treatment, no specific treatment. ORS. Prevention with 2 vaccines are available for the rotavirus. What are the 2 vaccines available for rotavirus? It is Rotavac and Rotarix. Rotavac. Appreciate the suffix. Rotavac, bolo, Rotavac and Rotarix. Okay, both are live attenuated, live. Whenever any vaccine comes up, people first you see is it killed or live. So both are live. Okay, live attenuated, live. But the strains are different. You have to know the strains. Here the strain is Exosola 116E and here the strain is G1P8. So these are the strains names you have to learn at least for your MCQs. Here 3 doses required, here 2 doses required. Here the 3 doses given at 6, 10, 14 week. Here the 2 doses given 6, 10. Both are oral. You can see this is Rotavac, appreciate. And this is Rotarix, appreciate. Both are given oral. In National Immunization Schedule, this one is included. Rotavac is included. In National Immunization Schedule, not the Rotarix. Okay. In 2020, recently only it is included in NIS. Please learn complete National Immunization Schedule. So that is done. So we are done with the virus also. So summary of the virus in front of you. It is causing both. We are done with virus. Now the last thing, I want to teach you 16 parasites. Actually 14. 2 of them are in the same column. So 14 more columns we have to go. If I give even 5 minutes to each of them, it will take another 2 hours. Okay, but we will finish it. We will try to finish it maximum we can do and hopefully the complete. So let's see the 16 parasites of GIT, but you know my way. I can't start it directly. Okay, so first, before I start, I would like to have the classification. So this is the classification of the parasites in front of you. I guess everyone knows the classification of parasite. We divide parasite into 2 categories. What are the 2 categories we are having? We are having protozoa, we are having metazoa, the unicellular, the multicellular. Okay. The unicellular, the multicellular, protozoa and metazoa. Protozoa also divided into 3 categories. Amoebas, flagellates and sporozoans. Metazoa also divided into 3 categories. You can see cystodes, trematodes and nematodes. So there are nearly 45 parasites in our syllabus. I will teach you all of them. But currently, not today. Today I will teach you 16 out of them. So out of all the parasites, today we will cover 16 out of them. So where are my 16? Where are the today's 16? Where are the purple 16 parasites? Let's highlight them. So you can see this is the highlighted version. You can count there are total 16 solar parasites which cause infection in the GIT. So we will divide them. Which of them are protozoa? We will take them separately. In the metazoa, we will take cystodes separately, trematodes separately, and nematodes separately. Okay, so total 4 master charts. 1 master chart for protozoa. In protozoa, we will take all these, the protozoa. The protozoa mein amoeba bhi, flagellate bhi, sporozoa bhi. We will take them in 1 master chart. Metazoa mein we will take 3 master charts. Metazoa are helminths. I guess you know metazoa are helminths. So in the metazoa, we will make 3 master charts. Which 3 master chart? Cystode ka master chart, trematode ka master chart, and nematode. So have a look. So see this classification and see this classification. You can see it's same. This is non-color coded. This is color coded. 8 colors for 8 system. So you can appreciate where it is purple. So appreciate the purple color everywhere. Can you quickly appreciate it? Yes. So here you can appreciate it. So whatever given here, it is given here. Appreciate the 16 purple. Appreciate the 16 purple. So in GIT, we are having many parasites, but they are very easy. Once you get the pattern in 2, 3, you will run in the next. Okay. So you can see the purple parasites, the 16 parasites. So first thing you should learn, which are protozoa, which are causing GIT infection. Among the metazoa, which are causing GIT infection. Among the cystodes, trematodes, nematodes. Okay. So let's start it. First, I will teach you the protozoa. There are 4 master charts. 1 for protozoa, 3 for metazoa. In the metazoa, 1 for cystode, 1 for trematode, 1 for nematode. So let's start with the protozoa. In the protozoa, you can appreciate the 3 purples. Appreciate. One is Entamoeba coli, Entamoeba histolytica, sorry. Entamoeba histolytica, the first one. The second is Giardia lamblia, the second one. And third is sporas, the 3 sporas. Cryptospora, cyclospora, isospora. Okay. So let's start with the protozoa. Let's start the 3 protozoa. You can see the 3 purple, the 3 protozoa. Entamoeba histolytica, which is an amoeba. Giardia lamblia, which is a flagellate. And coccidian parasites, which is a sporozoa. So we have 3 type of protozoa. One from each category. Protozoa are of 3 type. So one we have taken from each category. Let's start with the first one. Are you people there? No one is confirming. Harshal, Kushal. Let me ask the names. Anyone who is Arun, anyone who is live, kindly confirm that I am clearly visible, audible, and you are there. Okay, chalo. So let's start the protozoa. In the protozoa, the first protozoa I am starting Entamoeba histolytica. Let's start Entamoeba histolytica. Entamoeba histolytica is a type of amoeba. The first thing people learn the classification. Without classification, you cannot proceed. If I say the name of any parasite, you should know where it is coming. See the Entamoeba histolytica, where it is coming. Entamoeba histolytica, first thing it is a protozoa. In the protozoa, we have 3 options. Is it amoeba, flagellate, or sporozoan? So it is amoeba. It is amoeba. Can you see? It is amoeba. So Entamoeba histolytica. Introduction may write down. It's a protozoa. It's the only pathogenic amoeba. See all the amoebas in front of you. Only one amoeba causes disease in human. That is Entamoeba histolytica. There are many amoebas. You can see there are many amoebas. Okay, but only one amoeba is pathogenic, Entamoeba histolytica. So that's why I have written it is the only pathogenic amoeba. There are many amoebas which are commensal. They enter in human body, they don't cause anything. They are non-pathogenic, they are commensal. And 3 of them are free living, which affect the central nervous system. I will teach you all these 3 in CNS. So in CNS, you will see the remaining 3 amoeba. But currently, the only pathogenic amoeba is Entamoeba histolytica. That is the introduction. Morphology. There are 3 morphological forms. Protozoa. Okay, it's a protozoa. There are 3 morphological forms. Trophozoite, precyst, and cyst. Okay, in the metazoa, we have adults, egg, and larva. In the protozoa, we have trophozoite, precyst, and cyst because protozoa are unicellular and the metazoa are multicellular. Whenever it's multicellular, we have adult, egg, and larva. But here we have trophozoite, precyst, and cyst because it's a protozoa. Okay, so trophozoite, precyst, and cyst. Among them, the trophozoite is most important. Draw the diagram of trophozoite. It's a question in your university exam and MCQ very frequently. So here you can see this is the trophozoite. Let me tell you the important features in the trophozoite of Entamoeba histolytica. So in the trophozoite, first thing is shape. What is the shape? The shape is having a pseudopodia. One or multiple. What is pseudopodia? Pseudopodia is cytoplasmic extension. So it can be one or multiple. Okay, so any single unicellular organism having pseudopodia is amoeba. Amoeba mane pseudopodia, having cytoplasmic extension. Okay, so it is having a pseudopodia. It is having 2 types of cytoplasm, ectoplasm and endoplasm. So outer is ectoplasm, inner is endoplasm. Ectoplasm is clear, endoplasm is granular. Okay, in the endoplasm, there is granularity. It contains cell. It contains RBC and WBC and food vacuole. That's why it is granular. You will say, well, RBC, WBC, what you are talking about? It's a unicellular organism. From where it will bring RBC and WBC? Does it have the bone marrow to synthesize RBC-WBC? No, no, no. This trophozoite is present in human intestine. Okay, and it is attaching with human intestine and sucking the blood from human intestine. So this RBC and this WBC is human RBC and WBC, and it's a very important feature of Entamoeba histolytica. I'm teaching you Entamoeba histolytica. It's a very important feature of Entamoeba histolytica to suck the blood. The trophozoites suck the blood, and this is known as erythrophagocytosis. Erythrophagocytosis. It is known as erythrophagocytosis. RBC inside the cytoplasm of the trophozoite. Endoplasm. Okay, so this is erythrophagocytosis. It have RBC, WBC, and food vacuole. Now come on the nucleus. Nucleus is single, one nucleus. In the nucleus, there is a central dot. The central dot is known as karyosome. What it is known as? Label it as karyosome. And the chromatin in the nucleus is arranged in spoke-like fashion. The chromatin in the nucleus is arranged in spoke-like fashion. And the karyosome is central. It's not eccentric. It's central in location. So this is the nucleus. So this is a diagram of trophozoites. See it, learn it, label it. Now see here. Whatever I have told you, it's visible here. See the pseudopodia, cytoplasmic extension. See the ectoplasm, which is clear. Okay. See the endoplasm, which is granular. See, endoplasm contain RBC, food vacuole, WBC. It is known as erythrophagocytosis. See the nucleus. See the karyosome in the nucleus. See the spoke-like pattern of the chromatin. You have to draw it. You have to learn for MCQs. So this is trophozoite. Appreciate all the things I told you. Pre-cyst same. Pre-cyst same. Only thing RBC, WBC are not there. RBC, WBC are not there. Rest everything is same. Pre-cyst is same. Cyst is spherical, no pseudopodia. Cysts are always spherical or oval. Okay, they don't have pseudopodia. Double walled. Cysts are double walled. They are spherical. They are double walled, having 4 nucleus. Here 1 nucleus. Here 1 nucleus. Here 4 nucleus. 1, 2, 3, 4. So cyst is quadrinucleate. Quadrinucleate. It is known as quadrinucleate cyst. So that is the morphological form. Now, what is the mode of transmission? Mode of transmission of these 16 parasites is very unique. Okay. This one is entering via ingestion. Simple. Ingestion of contaminated food and water. So this person, this human is eating food and water which is contaminated with what? And what is the infective form? Out of the 3 options, does the food contain trophozoite? Does the food contain pre-cyst? Or does the food contain cyst? Out of the 3 things, what is present in the plate? What is present? Is it trophozoite, pre-cyst, or cyst? Which is the infective form? It's cyst, quadrinucleate cyst. If this person eats trophozoite, nothing will happen because trophozoites are destroyed by human acidity. If this person eat pre-cyst, nothing will happen because again it is destroyed by human acidity. But if this human is eating the quadrinucleate cyst, infection occur. Amoebiasis will occur. Okay, so the infective form is quadrinucleate cyst. So among the 3, whatever is the infective form, mark it here. So among the 3, this is the infective form. You always learn infective form, very important. Okay, now life cycle, I will explain. Only one host is there, man. No intermediate host, only one definitive host is there. See the life cycle. I will explain life cycle also, host also, disease also, diagnosis also, and treatment also. All 5 things in one diagram. Not only for this parasite, for all other parasites in my one diagram, everything will be clear. So everyone here, more than 50 MCQs are hidden on this one page. So mind it. Okay, everyone here. Now see this human. This is the same girl. Imagine the same girl. Imagine this is the same girl. Suppose, okay, she is eating or this human is eating the food and water which is contaminated with quadrinucleate cyst. Okay, see I have drawn the quadrinucleate cyst. Appreciate. Okay, quadrinucleate cyst is the infective form. And the route of entry is ingestion, eating. So the cyst go inside, bypass acidity. Trophozoite and pre-cyst are destroyed in acidity. Cyst bypass acidity. Okay, it goes in small intestine. Okay, it goes in duodenum, it goes in jejunum, it goes in ileum, it reach cecum. In the cecum, it rupture. I am talking about the cyst. Excistation occur. What it is known as? Excistation occur. When excistation occur? Where it occur? Cecum. So in the cecum, excistation occur. When excistation occur, from one cyst, 4 trophozoites will come. From one cyst, 4 trophozoites will come. From one cyst, 4 trophozoites. Cysts are very seedha sada. They don't do anything. They are just infective, but they don't cause the disease. Trophozoites are notorious. They cause the disease. What does they cause? These trophozoites, number one, okay, they penetrate in the wall of the cecum and produce flask-shaped ulcers over there. Okay, in the mucosa, submucosa, musculus propria, serosa, sometimes through and through perfo can occur. So flask-shaped ulcers, trophozoites are causing flask-shaped ulcers in the cecum. I will show you the diagram. Not only this is known as intestinal amoebiasis. The name of the disease is intestinal amoebiasis, in which person have flask-shaped ulcer in the cecum, pain in the abdomen, and ulcers are bleeding. Since the ulcers are bleeding, person have diarrhea and dysentery. Not only diarrhea, dysentery also. All ulcers bleed. So ulcers are bleeding over here. Got it? After that, these trophozoites go in the portal vein. You know, from the intestine, the blood goes in the portal vein. You know, first pass metabolism. Yes. So it is going in the portal vein. It is going in the portal vein. Who is going? Trophozoite. Cyst is not going. Mind my words, people. Mind my words. So it is trophozoite who is going. Trophozoite going. Trophozoite reach in the liver. Once the trophozoite reach in the liver, they will form an abscess in the liver, which contain pus. If you see the pus is brown color, and it is known as anchovy sauce abscess. Anchovy sauce. Have you seen chocolate sauce? Brown color sauce. It's looking like a chocolate sauce. Anchovy is not chocolate. Anchovy is fish. Fish sauce. Okay, fish sauce is famous in abroad, not in India. So fish sauce is brown in color, like the chocolate sauce. So anchovy sauce abscess on the right lobe of liver on the posterior superior surface, to be specific. Right lobe of liver, posterior superior surface, anchovy sauce abscess. Abhi bhi shanti nahi mili. Trophozoites are not done yet. Okay. After that, they will go in the hepatic vein. Okay, hepatic vein. You know, all veins drain in SVC, IVC. So hepatic vein is draining in IVC, inferior vena cava. So it is going in inferior vena cava and going in the right auricle, going in the right ventricle, that is right side of the heart, impure blood. Then it is going in pulmonary artery, and via pulmonary artery they reach the lung. The next abscess they form in the lung, again with the brown pus. The next abscess they are forming in the lung. From the lung they are going in the pulmonary vein. From the pulmonary vein they are coming to the left side of the heart. From the left side of the heart, they go to the aorta and they can cause abscess in multiple organs. So first organ involved is intestinal amoebiasis, and rest all is extraintestinal amoebiasis. Rest all together known as extraintestinal amoebiasis. So two diseases it is causing. It is causing intestinal amoebiasis and extraintestinal amoebiasis. Intestinal amoebiasis में flask-shaped ulcer in the cecum. Person have diarrhea, dysentery, and abdominal pain. Okay, extraintestinal amoebiasis में the first organ involved is liver, followed by lung, followed by all other organs. So this is the complete life cycle. Only man is involved. No other host is involved. Definitive host is man. Intermediate host is no one. I hope it's crystal clear. So you got the two diseases. What is intestinal amoebiasis? What is extraintestinal amoebiasis? Intestinal amoebiasis is the first disease. Extraintestinal amoebiasis is the second disease. So pathogenesis is done. Life cycle is done. Man is the definitive host. No intermediate host. Now last thing is lab diagnosis. Do the lab diagnosis. What sample you will take? For lab diagnosis, what sample you will take? What sample? So say sample separately for intestinal amoebiasis and say sample separately for extraintestinal amoebiasis. For intestinal amoebiasis, obviously the sample is 2. Okay. And for extraintestinal amoebiasis, the sample is pus aspiration. Aspirate the pus in a test tube doing sonography. Okay. Guided pus aspiration. So guided pus aspiration from the liver. Okay. So take 2 samples. One stool sample for examining intestinal amoebiasis and one guided pus aspiration for examining extraintestinal amoebiasis. I am giving you 3 options. Here what you will get, trophozoite, cyst or both. Here what you will get, use your common sense, trophozoite, cyst or both. You tell me what you will get. What you will get. So here we will get both trophozoite and cyst. See now, intestine mein cyst is converting into trophozoite. Maybe some cysts are excreting, some trophozoite are excreting. So here we get both. Here we get both. In the stool, we get both. But here in the pus, we get only trophozoite because it is the trophozoite who is traveling there. Cyst is not traveling. So here we get only trophozoite. We don't get both. So that is a diagnosis. And we can do imaging. On the imaging, we can see the cyst. Okay, we can see the abscess. Okay, you don't have to draw this in your exam. People, this is for your understanding. The simplified diagrams I have drawn for you. Okay, I have drawn for you. So this you don't have to draw. You have to draw like this. Okay, draw in the form of a flow chart, the circular flow chart or straight flow chart. Your starting point should be your end point. Okay, so draw. Whatever I have explained you, you draw in the form of the flow chart. So this is the life cycle. Okay, now disease, I already explained you the disease. It is causing 2 diseases, intestinal amoebiasis, extraintestinal amoebiasis. Intestinal amoebiasis में flask-shaped ulcer. Appreciate the flask-shaped ulcer. Appreciate the margin. Appreciate the margins. It is looking like a flask-shaped ulcer. Okay, extraintestinal MBBS इसमें see the liver abscess. Appreciate the brown color first. Appreciate it is known as anchovy sauce abscess on right lobe of liver posterior superior surface. So after that, lung abscess and other organs can be involved. Lab diagnosis is standard. Learn the standard lab diagnosis of all bacteria, all virus, all fungus, all parasite. You have to fix that unless until something extra is there. So here what you will do, of course, you will take the stool sample and you will do stool microscopy. I told you in stool you will get both. You get trophozoite as well as cyst. You can do the culture. Here for amoeba and amoeba, culture is also available. The name of the culture is polygenic and azanic. Polygenic, azanic culture, in which the colonies can be grown. We can do stool antigen detection, we can do blood antibody detection, we do PCR. And here we can do radiology, CT, MRI, we can do the abscess in the liver. Okay, we can see. So common sense. Okay, this is the standard till here. It is the standard lab diagnosis of all intestinal parasites. Today I am teaching you intestinal parasites. Drug of choice. What is the drug of choice? We have two drug of choice. Please understand. Have you seen my pharmacology videos? If any one of you is already a subscriber and you have studied pharmacology from me, I have taught you very well anti-protozoal drugs. In anti-protozoal drugs, I taught you very well the luminal agent and tissue agent. You may be understanding. So there are two type of drugs, luminal agent and tissue agent. What do you mean by luminal agent? This person is taking the drug with mouth. Okay, oral. So oral tablet go inside, 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 and here in the intestine. So if it is not absorbing, if it is remaining in the intestine only, it is known as luminal agent. And it will kill the trophozoite and cyst present in the intestine. Okay, so one of them are luminal agent. That will act in intestine. And that kill the trophozoite and cyst present in the intestine. That is lumen of the intestine. Now if it get absorbed and go in the blood, if it get absorbed and go in the blood, okay, via blood it reach the liver and it kill the trophozoite in the liver. So that is known as tissue agent. Liver is a tissue, liver, lung, and other organs in which cyst is there, abscess is there. So tissue agent get absorbed. Okay, luminal agent are not absorbed. They act on the lumen of the intestine, but the tissue agent get absorbed, go in the blood. Via blood they reach in the liver, lung, and other organs where the trophozoite is present. So they kill only trophozoite. So this one kill trophozoite, and this one is killing both trophozoite and cyst. This one acting in intestinal lumen, not absorbing in the blood. This one absorbing in the blood and reaching the tissue. So first give tissue agent, that is metronidazole, followed by luminal agent, that is paromomycin. So please learn the standard drug of choice for Entamoeba stolidea. The first is done. Can I move on the second one? How many of you with me? Can I move on the second one? Okay, parasito is very easy and it's my one of favorite. Okay, so Entamoeba stolidea is done. Coming on Giardia lamblia. This one was amoeba, this one is flagellate. This one is the only pathogenic amoeba. But how many type of flagellates we have? See the type of flagellates. I am teaching you Giardia lamblia, my second parasite. Okay, so you can see how many type of flagellates we have. We have three type of flagellates. The flagellates which remain in the intestine of human are intestinal flagellate. The flagellate which remains in the genitals of human are genital flagellates. And the flagellates which remain in the blood of the human are hemoflagellates. Okay, hemoflagellates. Got it? So, intestinal flagellates में Giardia lamblia. I am teaching you Giardia lamblia, which is intestinal flagellate. This will be covered on the last day. Okay, Trichomonas vaginalis in urogenital system. This should be covered in CVS, but I have not yet uploaded it. That day I couldn't complete na CVS. So it is not yet uploaded, but tomorrow is a break day. I will try to upload it tomorrow if possible. Okay, so I will try. Okay, so these I will upload tomorrow. You can watch it tomorrow. So these are in CVS, hemoflagellates. Currently, I am interested in Giardia lamblia, intestinal flagellate. Okay, so it's an intestinal flagellate. It don't have precyst. It have only trophozoite and cyst, two form. In contrast to Entamoeba histolytica, which was having three form. This one is having only two form. One trophozoite, one cyst. It don't have precyst. One trophozoite and one cyst. Question come on trophozoite. Trophozoite is important. So you can see, rather than seeing it here, we should draw. So let's draw the trophozoite. I will draw. What is the shape? This is the shape. What is this shape? Some people say, ki, ma'am, it's looking like a badminton racket. Okay. Some say ki it's looking like tennis racket. Okay. Some say it's looking like teardrop. And some say it's looking like monkey face because I will draw the nucleus. Okay. Some say it's looking like monkey face. So all four shape are correct. Badminton racket, write down. Okay, tennis racket, write down. Monkey face or teardrop. Okay, so that is the various type of shape. It is a flagellate now. It is having flagella. How many flagella? How many flagella? It is having four pairs. 4 pair. I'm not saying 4. I'm saying 4 pair. 4 pair means 8 flagella. Where are the 8 flagella? Draw it. Draw it. So this is pair number 1. This is pair number 2. This is pair number 3. I will draw the 3 pairs. The 4th pair is coming intracellular. The 4th pair is coming intracellular and then coming outside. The 4th pair is coming intracellular, dividing the parasite into 2 halves. Okay. The intracellular portion of the flagella is known as axoneme. Axoneme. You should be able to label it. So this portion is known as axoneme. The intracellular portion of the flagella. And total 4 pairs. And this one is dividing the parasite into 2 halves. Now how many nucleus? Everything is double. Everything is double. It is bilaterally symmetrical. I would say it's bilateral. If you fold it, it's bilaterally symmetrical. It is having 2 nucleus. It is having 2 paravasal disc. And it is having 2 sucking disc. 2 paravasal disc, 2 sucking disc, 2 nucleus. It's looking like a monkey face. Monkey face. Okay, monkey face, teardrop. So this is a diagram. You can appreciate. You can appreciate everything, whatever I told you. Appreciate the 4 pairs. Because of 4 pairs of flagella, it moves very fast. And the motility is known as falling leaf motility. Have you seen a falling leaf from the tree downwards? It is known as falling leaf. Falling leaf motility. Please learn it. Falling leaf motility. So that is regarding the trophozoite. See everything over here. Huh? Sister, same. Quadrinucleate. People, here 2 nucleus. 1, 2. Here 4 nucleus. Sisters always spherical or oval. 1, 2, 3, 4. Sisters same like entamoeba. Quadrinucleate. Okay. Mode of transmission is same. Ingestion. Okay. Now you have to tell me what is the infective form among the 2. So the plate. This plate contains what? Does it contain trophozoite or does it contain cyst? I'm giving you 2 options. Trophozoite or cyst? Cyst. Again quadrinucleate cyst. See, I have drawn. The infective form is quadrinucleate cyst. So the plate contains quadrinucleate cyst. This person is eating quadrinucleate cyst, not the trophozoite. If this person eats trophozoite, that get destroyed by the acidity. Okay, this is quadrinucleate cyst which is present. Man is the only host here. No intermediate host, only one host, man. Okay, now I will draw the complete life cycle with clinical feature with lab diagnosis over here. Okay, so kindly focus. One diagram, more than 40 questions. Everyone here, Giardia lamblia. Never forget the parasite I'm teaching you. Giardia lamblia. So see Giardia lamblia. Okay. Now this person is eating the food or drinking the water which is contaminated with quadrinucleate cyst of Giardia lamblia. Got it? See. So the route of entry is eating, ingestion. It's same as that of entamoeba. You will say where is the difference? You tell me where is the difference. Got it? So it is like entamoeba, it is going inside. It will go inside, bypass acidity. Go inside. But in duodenum, not in cecum. In duodenum, accestation takes place. So accestation takes place in duodenum. Got my point? So when accestation takes place in duodenum, in contrast to cecum in entamoeba histolytica, again 4 trophozoite will come. Here also 4 trophozoite. 1, 2, 3, 4. 4 trophozoite will come. Say yes. Huh? These 4 trophozoite, what they will do? They are very Siddha Sadha. Huh? Here sisters also see the trophozoite. In contrast to entamoeba trophozoite were invasive. They were invading in the wall and causing ulcers. They are non-invasive. Giardia ke trophozoite non-invasive. They don't invade. They don't cause ulcers. Simam, then what they do? You are saying neither the cyst is harmful nor the trophozoite is harmful. What does they do? The trophozoite, they don't invade. There is no ulcer, So that's why there is no dysentery here. In entamoeba, we have dysentery. Here we have diarrhea. Contrast, compare and contrast. Got it? So here the trophozoite are destroying the villi superficially, like EPEC. Yaad aaya, the bacteria today I taught you. Enteropathogenic E. coli. Superficially destroying the villi. Since the villi are destroyed, okay, nothing else, no ulcer. Now villi is destroyed. Why? God has given us villi? For absorption. So absorption will not occur. Since the absorption will not occur, the name of the disease is malabsorption syndrome. They cause malabsorption syndrome. The name of the disease was, there is amoebiasis. Intestinal amoebiasis, extraintestinal amoebiasis. Here we have only one disease, malabsorption syndrome. Nothing will be absorbed. Okay, what all things are absorbed? Carbohydrate, protein, fat, vitamin, mineral. Here nothing will absorb. If carbohydrate is not absorbed, it will cause flatulence. Okay, carbohydrate converted to gases. If protein is not absorbed, PAM, protein energy malnutrition, kwashiorkor, marasmus. Okay, if fat is not absorbed, Fat excreted in stools. Stools become bulky. Stool containing fats, foul smelling, steatorrhea. It is known as steatorrhea. Vitamins are not absorbed. Vitamin D deficiency, vitamin A deficiency. Minerals are not absorbed. Iron deficiency, anemia. So that is the symptoms of malabsorption syndrome. The name of the disease is one, malabsorption, and these are the symptoms of that. So we are done with the disease also. Now, the last thing, diagnosis. You know the life cycle? Only one host man. Diagnosis. You first tell me the sample for diagnosis. Who will tell me? Who will tell me? Not flattening, the destruction of the villi. Harshal. Okay, here destruction of the villi occurs. There flattening, destruction both occur in APC. Okay, listen here. What sample you will take? Who will tell me? The specimen, the sample. I think most of the students will say, ki, ma'am, stool sample. I think stool sample is a correct answer, but not a good one. Because you know it is duodenum. After duodenum, we have jejunum, we have ileum, we have ascending colon, transverse colon, descending colon, then we have stool. So, you know, these trophozoite and cyst reaching till stool, they all will be dead. Because the distance is much. There we have cecum. Okay, so yield was good. There were more chances we will get them in stool sample from the cecum till anus. Here from duodenum to anus, you know how many meters, huh? So duodenum to anus, you know, they will be dead on the way. So there is hardly any chance you will get something in the stool, trophozoite or cyst. If you are lucky, you can get it in your patient sample. Trophozoite also, stool also. Now, trophozoite also, cyst also in the stool sample. Stool sample is a good option, but not a very good option. I need the best option. Tell me the sample. See, ma'am, then maximum chances are there in the duodenum only. Okay, so take duodenal aspirate in a container. Take duodenal aspirate in a container. That will be the best sample you will have. I agree, but that is invasive, na? Now, Giardia lamblia affect children, small children, 3-year, 2-year. Okay, and what is the complaint? The child is not growing weight. The child is having foul-smelling feces. And you ask the mother, we will do a test in which we will do a sonography. We will take a syringe. We will aspirate something from the duodenum. Mother will say, no, doctor. My child, 1-year, 2-year-old child, no invasive test. Tell me some non-invasive test. So what you will do? I want duodenal aspirate only, but some non-invasive method. So there is a method. The name of the method is antero test. See, what is antero test? I will take a capsule. It's an empty capsule, empty gelatin capsule. I will tie it with a thread. See, this is the thread. The length of the thread is same as that of the distance from the incisor till duodenum. We know the nasogastric tube, the distance from the incisor till duodenum. We know the distance in children. So it's exactly same length thread we will take. And we will ask the child to take, we will ask the person to take the capsule with mouth. The capsule will go inside. Keep the other end of the thread in hand. Okay, this capsule along with the thread. This capsule along with the thread will go inside. Here it will stop because the length is that only. Here it will stop. Once it will stop, the capsule will dissolve. It's a gelatin capsule, it will dissolve. Okay, now the other end of the thread, you just stick it with the sellotape on the cheek. And this end of the thread remain there for 2 hours. 2 hours. After 2 hours, now if it contains trophozoite and cyst, if the duodenum, that's what we want to know now. If the duodenum contains trophozoite and cyst, this trophozoite and cyst will stick on the lower end of the thread. Now take the thread out. No anesthesia is required, it's not painful. Just take the thread out. Once you take the thread out, take a test tube, fill it with normal saline and put the thread there. Put this thread over there. If it contains trophozoite or cyst, it will dissolve in the saline. Centrifuge it, make a button, make a slide. You will get trophozoite and cyst. This test is known as antero test or string test. It is a way to collect the specimen. I hope you got it. So mode of transmission, infective form, host, life cycle you have to draw like this, not like this. This is for your understanding. Okay, you read it, you will understand it. These life cycles are given. Once you scan the QR codes in my master chart, these life cycles are given. These are not given in the master chart. Here I am explaining you in the lecture, but you will understand everything. You can correlate with my diagram. Clinical feature, only one disease, malabsorption syndrome. You know the symptoms of malabsorption syndrome. For test, we will collect duodenal aspirate with the help of antero test or string test. Okay, so that is lab diagnosis. Else everything is same as that of Entamoeba stolidea. Drug of choice here is tinidazole. There the drug of choice was metronidazole followed by paromomycin. That is tissue agent followed by luminal agent. Here we give only tinidazole. The drug of choice is tinidazole. No vaccine available. Can I come on the third protozoa? So protozoa will be done. Quickly we will finish cystode, nematode. They are very small. People, can I come on the next one? Let me show you my master chart of the parasite. I have not yet shown you. So let me come on the protozoa. Beautiful protozoas. I have given all the morphological forms. Appreciate the trophozoite precyst and cyst of Entamoeba. Appreciate the trophozoite and cyst of Giardia. Appreciate their mode of transmission. Mode of transmission of all of them is ingestion. Okay. Appreciate the clinical features. You can appreciate once it will be in your hand. Here, magnification is not very good. You can't see the original copy, but they are very good. And once you scan it, now you will get all these images high definition. You can make of your own also. I mean, I'm not saying that you should use these only, but it is the best way to remember the microbiology. Okay, many students make the master chart with their own handwritten. Daily, I take the class. After the class, once I check my WhatsApp, I get multiple messages from the students taking the snapshot with their own handwritten master chart during my lecture. And I feel so happy that my strategy is benefiting thousands of students. Okay. And it will benefit the patients in future once you will start the practice. I'm very happy. Anyways, come back. Come back. Coming on the third protozoa, the last protozoa. Can I come on the last protozoa? Coming on the third and the last protozoa. Coccidian parasites. Okay. Here by coccidian parasite, I mean the three sporas. Which are the three spora? Cryptospora, Cyclospora, Isospora. Cryptocycloisospora, together known as coccidian parasite. These are sporozoite. So basically, protozoa are of three types. Protozoa are of three types. One I have taken from amoeba, Entamoeba staudica. One I have taken from flagellate, Giardia lamblia, and one I have taken from sporozoan, coccidian parasite. So three categories, one member from each category causing intestinal infection. Here we have amoebiasis. Here we have malabsorption syndrome. And let me finish this, then I will fill it for you. So coming on the coccidian parasites. Okay. Coccidian parasite do not cause infection in healthy individual. It causes opportunistic infection only in HIV patient. It do not cause infection in healthy individual. It causes infection in HIV patients. Okay. Morphology, 5 form. Okay. Here we were having three form, trophozoite, precyst, cyst. Okay. Here we have 2 form, trophozoite and cyst. Here we have 5 form. Which 5 form? What are the 5 form? We have oocyst, most important form. See the oocyst of 3. See the oocyst of cryptospora. See the oocyst of cyclospora. And see the oocyst of isospora. Isospora also known as cytoisospora. Iso nowadays is known as cystoisospora. This is crypto. This is cyclo. This is cystoiso or iso. In some books it's written iso. In some books it is written with a prefix cystoiso. So don't get confused. This is the oocyst. Okay. After oocyst, trophozoite is there, scythoid is there, merozoite is there, and gametes is there. So you have to learn. Okay. Oocyst image-based question come in NEET PG very frequently. You have to identify oocyst of which one is which. You don't change the sequence. Crypto, cyclo, iso. Crypto. Learn with crypto. Then cyclo. You know we run cycle. So this is crypto. Crypto, cyclo, iso. Make some mnemonic. CCI or make some mnemonic. Okay, now here there are 4 sporozoite. Okay, simply inside it. Here 4 sporozoite, but inside 2 sporocyst. Okay, and here 8 sporozoite. So this is how we identify them. Image-based questions, mind map words common in NEET PG. How to identify the oocyst of the 3. So 100% strike rate, if I'm commenting now, it's a big challenge and I'm ready to accept the challenge. So each and every MCQ I'm covering in these master charts in this final gear. Okay, chalo. So that is the morphology. Mode of transmission is same. The person is eating the food contaminated with oocyst. So among the 5, what is present in the food? What is present in the plate? In the plate, oocyst is present. One of the oocyst of the 3. So infective form is oocyst, not all these. If all these are present, they get destroyed with acidity. But oocyst resists acidity. So oocyst is the infective form. Learn the infective form. The infective form is oocyst. Okay, of all of 3. Life cycle only one host man. Let me explain you here. So suppose this is the person who is eating the oocyst of one of them. This is the oocyst of cryptospora. This is the oocyst of cyclospora. This is the oocyst of isospora. Okay. You can identify the image. Now this person is eating the food and water which is contaminated with one of the oocysts, not all 3. One of them. And this person is HIV person. Because it enters, it causes infection only in HIV person. Okay. Now this oocyst goes inside in the intestine. Oocyst in the intestine. Excision occur. Okay. This oocyst goes inside. Excision occur. When excision occur, all of them contain sporozoite. So 4 sporozoite or 8 sporozoite. Okay. So sporozoite come out. Oocyst में से sporozoite come out. Okay, either 4 or 8. You can see, common sense. The oocyst will rupture and the sporozoite will come out. Now these sporozoite convert into trophozoite, scyzont, merozoite and gamete. So these sporozoite convert trophozoite, scyzont, merozoite and finally gamete. In the sequence only it convert. Okay, it's very easy. So oocyst will rupture, sporozoite will come out, and sporozoite convert first into trophozoite, then into scyzont, then into merozoite and finally gamete. Gametes are of two types, male and female. So male gamete, female gamete, they will fuse with each other. They will fuse with each other, male, female gamete, and zygote is formed. And zygote again convert into oocyst. So oocyst will be excreted. So the person is eating the oocyst, person is excreting the oocyst. Got it? Person is eating the oocyst, oocyst will rupture, it will convert into this. Gametes will fuse and again oocyst is formed. So life cycle is very simple. Okay. So you can read it. I have drawn the flowchart. Okay. Clinical feature, only in immunocompromised host. HIV host. It causes chronic diarrhea. It causes chronic diarrhea in the intestine. Sometimes they go in the blood, and in the blood they cause extraintestinal symptoms. May sclerosing cholangitis in the bile duct and pancreatitis. So it involves bile duct and pancreas also. These three sporas, and they cause infection only in HIV persons. Lab diagnosis standard. You collect the stool sample. What you will get? You collect the stool sample. Collect the stool sample. You will get the oocyst, and based on the oocyst, you can identify which one of this. Okay, they are acid fast, partial acid fast. CMAP first session, they are partial acid fast. Okay, so in the lab diagnosis, you will get you do Zn stain. On Zn stain, you will get the oocyst, which are red color. And based on the oocyst, you can identify them. The remaining lab diagnosis is standard. Stool antigen detection, serology, PCR in all parasites. Okay, drug of choice. It's different. For crypto, it's different. For the remaining two, that is cyclo and iso, it's different. For the first one, it's different. Crypto. For crypto, it is nitazoxanide. Nitazoxanide. And for cyclo and iso, it's cotrimoxazole. These all are your MCQs, people. I cannot help more than that. Yes, I'm doing the best. I mean, I can challenge. It cannot be easier than this. So that is the treatment. Okay, so we are done with the 3 protozoa. Now, who will help me in filling it quickly? I know it will take 2 to 3 minutes, maybe 4 minutes, but it's easy. Okay, this chart we will fill. Maybe the next 3 charts, no, but this is the first chart I will fill with you. So kindly help me in filling, people. I will keep it in front of me. Here. On my wall. Chalo, quickly I will fill it for you. So, Anamoeba histolytica, Giardia lamblia, and coccidian parasite. Here I will write introduction में only pathogenic amoeba. It's only pathogenic amoeba. Here I will write it's an intestinal flagellate. Okay, and here I will write it infects only HIV person. Introduction. Introduction is not very important, but morphology you can't skip. Morphological form. Here 3 form: trophozoite, pre-cyst, cyst. You know the diagram of all 3. Okay, please. Here only 2 form: trophozoite and cyst. Okay, remember. And here we have oocyst, then trophozoite, oocyst, trophozoite, scyzont, merozoite and gamete. So these are the 5 forms we are having. So 3 form, 2 form, 5 form. Now after that, mode of transmission. Mode of transmission is ingestion of contaminated food and water. You know the image I provide here. Now infective form. Among the 3, here the infective form is cyst, quadrinucleate cyst. Okay, among the 3, here 2 infective form is cyst again. And here the infective form is oocyst. This is how I write. I circle also there. So this is the infective form. After infective form, tell me, man is the definitive host in all of that, and intermediate host is none. Only one host, man. Intermediate host is none in all of that. Okay. Clinical feature. Here the name of the disease. Here the name of the disease is amoebiasis. It's amoebiasis. Intestinal amoebiasis, extra-intestinal amoebiasis. Intestinal amoebiasis में flash-shaped ulcers in the cecum. Extra-intestinal में anchovy sauce abscess in the liver, followed by lung, followed by other organs. Okay. Here the name of the disease is malabsorption syndrome. Nothing will absorb: protein, carbohydrate, fat, vitamin, mineral. Okay. And here the name of disease in immunocompromised person, intestinal and extra-intestinal. Intestinal में chronic diarrhea and extra-intestinal में sclerosing cholangitis with pancreatitis. So that is the clinical feature. Lab diagnosis you will do. Treatment में here metronidazole followed by paromomycin. Here tinidazole, and here it is different. For cryptospora, it is nitazoxanide, and for the other 2 spora, it's cotrimoxazole. Okay, got it. So this is how you read, you learn the parasites life cycle. You have to draw it completely. I have just written it. You have to draw it completely with a flowchart. So this is the summary. See the 3 morphological form here. See the 2 morphological form and see the oocyst of it. You should be able to draw it. Say, people say. Can we go ahead? Can we start the sisters? Enjoying learning? Huh? Show some interest. Can we go on the Hellman's sisters? Protozoa is done. Metazoa. I'm sorry what I have done. Just a second. Wait a while. I'm sorry. So protozoa is done. Okay, coming on the metazoa. Protozoa is done. Coming on metazoa. 3 categories: cystode, trematode, nematode. So let's start with the cystodes. Okay, cystodes. Before I start metazoa, have an overall look. What are cystode, what are trematode, and what are nematode? Quickly we will have a look. So you can see this is cystode, this is trematode, this is nematode. Quickly have a look. Cystodes are fragmented. Can you see? There is a scolix. Scolix means head. Head means scolix. This is scolix. This is neck, and this is the body. The body is known as strobila, and the body is fragmented. Multiple fragments are present. Multiple segments are present. You can see the number differs from parasite to parasite. In each fragment, there is a ovary, there is a testy. Ovary में से ova will be released. Testes में से sperm will be released. They will fuse with each other, and eggs are formed. Eggs are formed. In the last segments, they will rupture. They will rupture, and eggs is secreted. Okay, so length will shorten. The neck will form the new fragments. Okay, the neck will form these fragments are known as proglottids. Okay, so neck is forming the new one. These are immature one. They don't have ovary and testes. Then they mature. They have the ovary testes. In the ovary testes, they fertilize. Once fertilization takes place, eggs will come. They become gravid. Gravid means pregnant. So this is monoecious, male, female in the same organ. Cystodes are monoecious, male, female in the same organ. Okay, same organism. Next are trematodes. Trematodes are leaf shape. Leaf. That's why they are known as fluke. They are flattened and elongated like a sellotape, inch tape. Have you seen sellotape? Have you seen inch tape? They are flattened, lambe, flattened. They are known as tapeworm. That's why they are known as tapeworm. These cystodes. They are known as tapeworm because they are flattened. Okay, they don't have cavity. These are known as fluke. Fluke means leaf. They are known as flukes. Flukes means leaf. They are leaf shape. Here also ovaries and testes both are present. Ovary and testes in the same organism. Ovaries say ova, testes say sperm. They will fuse with each other. The organism become gravid, and they have a vaginal cavity from which it comes out. Okay, from which it comes out. It don't rupture. So eggs come out. They are also monoecious. They are also monoecious. They are also monoecious. The last one are the nematodes, the most advanced one. These are cystodes, these are trematodes, these are nematodes. The nematodes are the most advanced one. There is a male, there is a female. Okay, they are separate. The sexes are separate, the genders are separate. Male is separate, female is separate. Male, female come closer, do the physical intercourse. Okay, sexual intercourse. Okay, here male, female in the same organism. Here male, female in the same organism. But here they come closer and do a physical contact. In the male, spicules are present. In the female, vulva is present. So spicule is inserted in the vulva. Via spicule, the sperms are inserted in the vulva. After going inside, the sperm fuse with the ova and eggs are formed. Once the eggs are formed, this female becomes gravid and she lay the eggs. So this is the overall diagram. Once for all I told you. Okay, so this is adult, this is adult, this is adult. Adult of any three do the fertilization. If it is cystode, it is in the same organism. If it is trematode, it is in the same organism. But if it is a nematode, two different organisms. Ultimately, the adults, they do fertilization. Once the adult do fertilization, eggs are formed. Eggs inside this, eggs inside this, eggs inside the female, not inside the male. Okay, eggs are formed. They lay the eggs. They lay eggs. Okay, once eggs are laid, this is the egg. After few weeks, egg will rupture. From the egg, larva will come, and larva will again grow into adult. Okay, so this is the life cycle of overall. Now, what questions come in your parasitology? You should learn the eggs. You should learn the eggs over here. Okay, whenever I tell you any except I will tell you adult also, I will tell you larva also. For all these worms, whatever I will teach you. Okay, till now I taught you 3. Okay, 13 more to go, 16 may say. So in all the 13, I will teach you all 3 forms. Adult, egg and larva. This is the adult of them. Egg of them, larva. But egg, whenever I will teach, you have to say 2 things about the egg. Yesterday also, I guess I taught you. Whenever we talk about the egg, we say the color of the egg. Okay, color is yellow, we say it bile stained. Bile stained means B positive. And color is transparent, we say it's B negative. B negative means not bile stained. It's transparent. So this is yellow, this is transparent. Yellow mane bile stained and transparent mane non bile stained. My coding, I write it B positive, B negative. So in my notes or anywhere, if you are finding B positive, I mean yellow egg. If I write B negative, I mean transparent egg. So you have to tell me the color of the egg. And this is the human bile which is coating the egg, because these eggs are present in human intestine. Okay. Yesterday I taught you. Watch my yesterday's session. And the same egg, suppose I'm having a patient in my clinic. My patient is complaining some GIT infection and I'm suspecting the patient is having parasitic infection. I will ask the patient to give a stool sample in a sterile container to submit the sample. The next day, patient have submitted the sample. Now I'm having stool sample of my patient. I'm a microbiologist. I will make the saline suspension of this and search for the eggs. Luckily, I got the egg. Okay, I will see the color of the egg in my microscope. Color is this or this. So I will label is it B positive or B negative. Then I will take that egg with a forceps and put it in saturated solution of NaCl, namak pani ka ghol. So I will take a container. I will make saturated solution of NaCl and the egg present in the feces of my patient, I will pick it with a forceps, a small egg, and put it there. And I will see whether it is floating or sinking. Ya to तैरेगा या डूबेगा. Whether it is floating or whether it is sinking, depending on the weight or specific gravity. If it is light, it will float. If it is heavy, it will sink. So if it is floating, I will call it F positive. If it is not floating, I will call it F negative. So for all egg, I will say, is it B positive, B negative, F positive or F negative? This is my coding, So you must understand my coding before I start. Let's start the cystores. People, with this introduction, we are ready to run now. So let's start the cystores. There are 4 cystores. Okay, what are the 4 cystores? Read the name. There is Taenia saginata, Taenia solium, Diphyllobrothium latum, you can call it D latum, and Hymenopsis nana, you can call it H nana. So Taenia solium, Taenia saginata, Diphyllobrothium latum, and H nana. Very simple. I will take these 2 parallel under these headings. These 2 parallel. These are close brothers, twin brothers. So let's take them parallel. You will have a better understanding. So these 2 we will cover together, and these 2 are small. Okay, so let's start it. Let's start the 2 Taenias. Please see, this is saginata, this is solium. Appreciate. This is saginata, this is solium. You can appreciate. This is saginata and this one is solium. This side I am taking. So in the introduction, tell me their other name. Other name. Tell me their other name in the introduction. Okay, Taenia saginata is known as beef tapeworm. It is known as beef because it occurs by eating the cow meat. Okay, uncooked cow meat, the person eat it. That's why it is known as beef tapeworm. This one is known as pork tapeworm because it occurs by eating the uncooked meat of the pig. That's why pork tapeworm. Learn one name. The other name, this is unarmed. This is armed. I will explain. Introduction is done. Come on, morphology. Morphology mein now there is no trophozoid, no cyst. वो सब protozoa में था. Protozoas are done. Trophozoid, cyst. Okay. Now we will have adult egg larva. In this sequence, I'd say first there is adult. They fertilize. Once they fertilize, they become gravid. They lay egg. Okay, egg will mature, egg will hatch, and the larva will come. And larva will again grow an adult. So my sequence is fixed, people. Adult, egg, larva. Learn it. Adult followed by egg followed by larva. So say the adult of both, say the egg of both, say the larva of both. Taenia saginata, Taenia solium. See the adult of both. See adult of both. Can you see the adult of both? Give me a minute. See the adult of both. See the diagram. This is only head, scolex. Scolex is known as head. Below which there are many proglatids. The chain of proglatids. Body I have not drawn. I have drawn only head. See the head here, see the head here. See the head of both. Taenia saginata and Taenia solium. You can see the head of the both. In the head, here also 4 suckers are there. 1, 2, 3, 4. In the head, here also 4 suckers are there. 1, 2, 3, 4. Appreciate 4 sucker, appreciate 4 sucker. In both of them, 4 suckers present. In both of them, 4 suckers present. In the head. Adult ka head man is colex. 4 sucker present, 4 sucker present. Okay, now, see. There is a pouting. Okay. It is known as rostulum. So can you appreciate rostulum is present here. Here we don't have any pouting. Rostulum is absent here. So here rostulum is absent. Rostulum is pouting in the mouth. It's like this. Okay. And here rostulum is present in solium. This is saginata. This is solium. And third is hooklet. Here we don't have hooklet. And here we have hooklet around the rostulum. So 4 suckers are present in both. Here also we have 4 sucker. Here also we have 4 sucker. But rostulum is not there. Hooklet is not there. Rostulum is there. Hooklet is there. You are getting my point. Hooklets are the arms. Hooklets. are the arms. Arms yani weapon. You know, army, arm, weapons. So this one have the weapons. This one is known as armed tapeworm. And this one don't have the weapon. It is known as unarmed. Now you understood the other name. This one is unarmed and this one is armed. Okay, learn two name. You will say, ma'am, how to learn? This is also Taenia saginata starting from S. This is also Taenia solium starting from S. In the exam, I can get confused. No worries. Have you seen in real life the cow and the pig? Have you seen? Cow is little bit larger and pig is little bit smaller if you have seen in real life, or if you know that. So see the spelling of saginata and see the spelling of solium. Count the letters. So that is the same difference in the spelling ka size. So see the saginata is little bit larger. You can understand saginata is the beef tapeworm and solium is small spelling. So it is pig tapeworm, pork tapeworm, number one. Okay. Now how to learn the second one? How to learn the second name? Cow is very seedha sada animal. We worship cow. You know, we worship cow. It is unarmed. It don't harm. It never bite. Cow never bite. It is unarmed. Okay. But pig, if you provocate a pig, pig will bite you. So this one is the armed. This is how you can learn. It's a way. You can make your own trick. So adult is done. You see, image-based questions are very common on the adult. Mind it. Adult is done. Based on the head, we can identify them. Foresucker. Rostulum absent, rostulum present. Hooklet absent, hooklet present. Okay, axe. Axe are same for both of them. That's why I have not run twice. I have not done the division of this page. Axe are same in both of them. Saginata, solium, both have the sac. Saginata also have the sac, solium also the sac. So ag may you always write B status, F status. See the color? What color you can see? Yellow, B positive. Now make a rule. Whenever it's B positive, F negative. Okay, with some exception. Whenever it's B negative, F positive. Okay, so the yellow eggs are lighter and the transparent eggs are heavier. Yes. Okay, so this is F negative because it is B positive, F negative. Okay, this one is lighter. It will float. I'm sorry. It will sink. It will sink. Okay, so B positive, F negative. Okay, and this one is having an embryo. Inside the egg, an embryo is there. Appreciate the embryo. It is having 6 hooklet. 1, 2, 3, 4, 5, 6. That's why it is known as hexacant embryo. Hexacant. So learn the B status, F status and a unique feature. The B status B positive, F status F negative. Unique feature, it's hexacant embryo. So that is the egg. Now tell me the larva. Larva ka we don't learn diagram. Okay, larva ka sirf naam. One larva. Okay, the name of the larva is Cysticircus. But the surname is different. Okay, in Saginata it's bovis. Bovis mane cattle, cow, bovis. Okay, and here it is Cysticircus cellulase. Cellulase mane pig. So Cysticircus bovis and Cysticircus cellulase. So this is the morphology. Okay, before I move on the mode of transmission, let's revise it. Let's revise morphology. So Taenia saginata, okay ji, and Taenia solium. Okay, I always take saginata here, solium here. You tell me their other names. This is beef tapeworm and this one is unarmed one. Unarmed tapeworm. This one is pork tapeworm and this one is armed. Okay, now tell me they are adult of both of them. I will say foresucker present, rostulum absent, hooklet absent. That's why it is unarmed. Here rostulum present, hooklet present. That's why it is gone. So adult is done. Intro is done, adult is done. Tell me their egg. Who will tell me their egg? Egg of both of them. Both the eggs are B positive. It's same. Both the eggs are F negative. B positive, F negative. And hexagon embryo. So see the intro, see the adult, see the egg. See the larva of both of them. Cysticircus bovis, Cysticircus cellulase. So till now I taught you intro, adult, egg, larva. Adult ka draw 2 diagram. Egg ka draw 1 diagram which is same. And larva we don't have diagram. Mode of transmission. Okay, here the person is eating cow ka meat. Here the person is eating pig ka meat. So ingestion, eating. Okay, infective stage. Okay, what does the cow ka meat and pork ka meat contain? Does it contain adult? No. Does it contain egg? No. It contains larva. Here the larva is Cysticircus bovis. Here the larva is Cysticircus cellulare. So here the meat of the cow contains Cysticircus bovis larva. And here the meat of the pig contains Cysticircus cellulare, the larva. Got it? So larva is the infective form in both of them. Life cycle. Let me tell you the life cycle quickly here. So that you can draw in your exam like this or like this. Okay? You have to draw like this. But I will explain you with simplicity like this. So this side, Taenia saginata, you can see. And this side Taenia solium, you can see. This is saginata. no need to say. You can see the cow. And this side, tinea solium, no need to say. You can see the pig. Okay, quickly. This person is eating the cow. The cow contains Cysticercus bovis, the larva. And this person is eating the pig. The pig contains Cysticercus cellulare, the larva. So infective form in both of them is larva. Cysticercus bovis, Cysticercus cellulare. Larva go inside. Larva go inside. Okay, larva bypass acidity. Okay, in the intestine, larva convert into adult. What's the next stage of larva? Larva convert into adult. Okay, so here adult is formed. Here also adult is formed. Adult cause diarrhea in both of them. The name of disease is teniasis. Here also the person have teniasis. Okay, mild diarrhea, no treatment required, ORS. Mild diarrhea, no treatment required, ORS. Teniasis. Okay, that is the adult form. Okay, adult will fertilize in the intestine. Okay, adult will fertilize in the intestine, male, female in the same form. They will fertilize. Eggs are laid in the feces. Eggs are laid in the feces. Eggs are laid. Okay, these eggs will be eaten by cow. They will contaminate the grass and the contaminated grass is eaten by cow. And here the eggs are eaten by the pig. Pig eat human feces. Pig eat. Cow don't eat human feces, but it is contaminating the grass. So infective stage for the animal is egg. Infective stage for animal is egg. Egg will go inside the animal. Egg will hatch. Okay, egg will hatch and larva will come. And the same larva is eaten by another human. Okay, egg will hatch inside it, convert into larva, and that is same eaten by another human. Life cycle continue. It's a circle. The starting point should be the end point. You got my point? So infective stage for man is larva present in the meat of the animal. And infective stage for animal is egg present in the feces of the human. I hope it's crystal clear. And disease is one, teniasis, which is caused by adult. But there is a catch in the solium. In the solium, along with teniasis, one more disease occurs. In the solium only, not in saginata. In the solium only, one more disease occurs. Imagine, what does this pig is eating? You tell me, what does this pig is eating? This pig is eating the eggs in the human feces. Pig eat human feces. Human feces contain egg. So pig is eating. Let's replace the pig with another human. Chi, Hana. Another human is eating not the human feces. Okay, say it decently. Another human is eating the food and water contaminated with human feces. Okay, the another human is eating the food and water contaminated with this human feces. So basically we are replacing the pig with another human. Okay, so whatever happened to pig, the same thing happened to this human. So this egg will go inside, egg will go inside, egg will go inside, egg will hatch. Once the egg will hatch, larva will come. Once the egg will hatch, larva will come. Larvae are dangerous. Adult remain in intestine only and cause diarrhea. Larvae don't remain in intestine. Once the larva form, it penetrate and go in multiple organs. It go in multiple organ. The first organ it goes is the brain, causing neurocysticercosis. Second organ it goes in eyes, orbital cysticercosis. Then it goes in subcutaneous tissue, subcutaneous cysticercosis. So the name of disease is cysticercosis caused by larva. Now you may be thinking, why larva they are eating, this is not causing. Now please learn the golden rule. Whatever person eat, the next stage cause the disease. Here the person is eating larva, the next adult cause teniasis. Here also person eating larva, the next adult cause disease. But here person is eating egg, so the next larva will cause the disease. So whatever person eat that do not cause a disease, the next stage cause a disease. Now you may be thinking, ma'am, why here you have replaced the pig with another man? Why here you are not replacing the cow with another man? Even if I replace it, here even if the human is eating the egg of Taenia saginata, they are non-infective in human. Here these eggs are destroyed by acidity, but these eggs are not destroyed. So the summary, saginata cause only teniasis. Solium cause teniasis and cysticercosis. I tried, people. I tried. Now read this. You will have a better understanding. So that is the life cycle of both of them. So this is a summary. Teniasis is caused by both of them. Both of them. The adult of both of them causes teniasis. Cysticercosis is caused only by solium. When the man is eating egg, not the larva. When the man is eating egg. When the man is eating larva, teniasis occurs. When the man is eating egg, cysticercosis occurs. That too in Taenia solium, not in Taenia saginata. I'm crystal clear in my concept. Read my notes, read my master charts now. For details, watch the main videos. Okay, so that is the life cycle. Lab diagnosis के लिए teniasis के लिए take the stool sample. You will get adult and egg. Based on egg, you cannot differentiate them. Now collect the stool sample over here. Collect the stool over here. Collect the stool sample over here. Collect the stool sample over here. Here in the stool, you will get egg and adult both. Here also you will get egg and adult both. Based on egg, you can't differentiate. Eggs are same. But based on adult ka head, sucker, rostulum, hooklets, you can differentiate. So same thing is written. So that is the diagnosis. For cysticercosis, we can do the biopsy of the brain, of subcutaneous tissue, orbit. We can do CT, MRI. So that is cysticercosis by Taenia solium. Drug of choice, praziquintal. Praziquintal. For all cystodes, the drug of choice is praziquintal. And if it is cysticercosis, excision may be required. Okay, so we are done with 2. Appreciate their adult, appreciate their act. Adult is different, acts are same. People 2 are done, 2 more to go. Can I move on the next parasite? The next parasite is D. latum, Diphyllobrothium latum. Let's start it. Let's quickly start the next one. The next one is Diphyllobrothium latum. Okay, what is the other name? Here the other name was beef tapeworm. Here, because it is occurring by eating beef. Here the other is pork tapeworm, by eating pork. Here the other name is fish tapeworm, because it occurs by eating fish. Okay, uncooked fish, raw fish, or partially cooked fish. So it is known as fish tapeworm. It is also known as broad tapeworm because this is head. Okay, the strobila, the proglottids, the width is more than length. Okay, these are proglottids, na? These are proglottids. In the proglottids, the width is more than the length. That's why it is known as broad tapeworm. Broad tapeworm. Learn the 2 names. It is known as fish tapeworm, broad tapeworm. Now see adult egg larva. Adult egg larva, standard. Okay, what is known as Diphyllobrothium? What is the name? The name is D. latum. Diphyllobrothium latum. Diphyllobrothium latum. What is dibrothia? What do you mean by dibrothia? It don't have sucker. Instead of sucker, it is having a slit. Instead of sucker, it is having a slit. One anterior, one posterior. 2 slit. The slit is known as brothia. And that's why it is known as Diphyllobrothium. It is having 2 brothia. It is having 2 brothia. Appreciate the 2. One anterior, one posterior. This is adult ka head. See the head. Appreciate unke head. Head of solium and saginata. See the head here. Okay. Rostulum absent, hooklet absent. Okay. Rostulum absent, hooklet absent. No rostulum, no hooklet. Okay. Acts. Acts are yellow in color. B positive. Whenever it's B positive, it's F negative. I told you. Whenever it's B positive, F negative. But the unique feature, it is having a cap, topi. It don't have hexacanth embryo. Learn the unique feature of all acts. There the acts were having hexacanth embryo. It is having a cap and a knob. Upper cap, operculum, niche knob. So operculum with a knob. There are 3 larvae, L1, L2, L3. There are 3 larvae, L1, L2, L3. Not 1 larva. L1, L2, L3. What is the mode of transmission? Eating the fish. What does the fish contain? Fish contain adult? No. Fish contain egg? No. Fish contain larva? Yes. Which larva? L1, L2, L3. It contains L3 larva. So infactive form is L3 present in the muscle of the fish. Infactive form is L3 larva. The name of L3 larva is pleurosarcoid larva, if you want to learn the name. Okay, now let me tell you the life cycle very quickly. So this is a human, the same human. You can see this human. This is the same human who is eating the fish. Now this fish contains L3 larva. Okay. Now you tell me the life cycle. First you tell me the general life cycle. So first there is adult. In the adult, male, female, both are in the same organism. They fertilize. They lay egg. Egg will hatch. So first L1 will come, then L2 will come, then L3 will come. L3 will convert into adult again. I guess this is clear to everyone. Now apply the same over here. This person eating L3. Okay, L3. L3 go inside. In the intestine. L3 का next क्या आएगा? L3 is next is adult. So it will convert into adult. Okay. This L3 convert into adult. This is adult. This is adult. Adult will fertilize. Adult will lay the egg. Eggs are passed in the feces. Okay. Let me pass the feces. Feces go in water. Egg go in water. In the water, egg will hatch. And L1 will come. So egg to L1 takes place in water. Okay, this L1 is ingested by snail. This egg is, not snail, cyclopes. This is cyclope. Okay, in the water we have cyclope. L1 ingested by cyclope. In the cyclope, L1 to L2 takes place. L1 to L2. This cyclope is ingested by fish. Like human eat fish, fish eat cyclope. Inside the body of fish, L2 to L3 conversion takes place. And this fish is eaten by this human again. So there are 2 intermediate hosts. The definitive host is human because fertilization takes place in human. Adult is in human. First intermediate host is cyclope and second intermediate host is fish. Okay, so this is the complete life cycle. If you have trouble, you have to write like this. If you have trouble, you can write like this also. See, fish, man eat fish containing L3. Now L3 में से बनेगा adult. Adult में से adult will fertilize and they lay egg. Okay, egg will hatch and form L1. L1 convert into L2. L2 convert into L3, and this is the same L3 which is present in fish. Now you tell me which conversion taking place where? Human eating fish. So this conversion takes place in mankind intestine. Okay, this conversion also takes place in man. Fertilization also takes place in mankind intestine. Okay, man will pass egg in the feces. Feces go in water. So egg will hatch in water. So egg to L1 in water. Now L1 is ingested by cyclone. Okay, so in the body of the cyclone, L1 to L2 and L2 to L3 is in the body of the fish. And this is the same fish eaten. You can learn the life cycle. It's your choice. Okay, the name of the disease it is causing. See here, the adult. Adult cause disease in human intestine. This adult is causing this disease in human intestine. This adult is causing a disease which is known as diphyllobrothiasis. Like their teniasis tha na, yahan pe diphyllobrothiasis. So person had diarrhea, simple mild diarrhea, no treatment requires. So teniasis, diphyllobrothiasis, hymenopsiasis, like that. These all are diarrheas caused by that. So here it is diphyllobrothiasis. Okay, the name of the disease. And this adult worm, the favorite dish of this adult worm is vitamin B12. So whatever vitamin B12 consumed by this human is ingested by this worm. So this person have vitamin B12 deficiency, that is megaloblastic anemia. So two things occurs in diphyllobrothiasis: mild diarrhea and megaloblastic anemia. Megaloblastic anemia is unique. You asked MCQs. Lab diagnosis is standard. Treatment. Praziquantel is drug of choice, but we will give parenteral vitamin B12 also, not oral. If oral is given, again the worm will eat it. So give it directly in the blood. So parenteral word is important. Parenteral injections of vitamin B12. So that is the treatment. I hope we are done. We are done with 3 till now. See the adult of all 3. Appreciate 4 sucker. No rostulum, no hooklet. 4 sucker. Rostulum present, hooklet present. No sucker. There are 2 brothia, but no rostulum, no hooklet. C B positive. C F negative. Hexacanth embryo, operculum with knob. I cannot make easy more than this. These are the heights, people. These are the heights. Got it? Can we go through the next? The last one. The last one here is H nana. Let's come on the last one. The last one is H nana. Okay, H nana, Hymenopsis nana. H nana is Hymenopsis nana. H nana. Okay. It is also known as dwarf tapeworm and is the smallest tapeworm because it is smallest among all. So learn the other name: dwarf tapeworm, smallest tapeworm, adult egg larva in all of them. See the adult. Here also 4 suckers. Appreciate 1, 2, 3, 4. Like the tinea, 4 sucker, but no rostulum, no hooklet. So it is like tinea saginata. 4 suckers there, but no rostulum, no hooklet. Okay, like saginata. Okay, but the main thing, eggs are transparent. First time I'm telling you eggs are transparent. So it is B negative, F positive. It's B negative, F positive. Appreciate. Whenever it's B negative, F positive. Till now we have B positive, F negative. Their eggs were yellow, but here eggs is transparent. And it is having the hexacanth embryo only, like the tenias. Like the tenias, it is having hexacanth embryo, but it is transparent. It is transparent. It is also having hexacanth embryo, but additionally it is having the polar filaments, which was not present in tenia, and it is having yolk granules. It is having yolk granules. So yolk granules and polar filaments is there. Larva is only one, cysticercoid, like the tenias. Like the tenias. Okay, there are only 4 eggs which are transparent. I guess everyone know the mnemonic Neha, N-E-H-A. So currently I am teaching you H nana. H nana I am teaching you, that is transparent. 3 more will come. Okay, 3 more will come in nematodes only. So these all are transparent egg. Rest all eggs are yellow. Only 4 are transparent. You can learn Neha is B negative. Neha is B negative, F positive. Neha is B negative, F positive, or you can learn Neha is transparent. Neha is transparent. Okay, so H is H nana. Mode of transmission is very simple. Okay, it is not any animal like pork or cow or fish. This person is eating simple food and water. Simple food and water, which is contaminated with the eggs. Here, infective stage is egg. Infective stage is not larva like the tenias, like the dilatum. Here, the infective stage is egg. This person is eating the food and water which contain egg. The infective stage is egg. Okay, egg will go inside, egg will hatch. From the egg, larva will come. Okay, larva will grow into adult, and adult again fertilize and lay egg. So Hindi में बोलो तो खा भी egg रहा है, निकाल भी egg रहा है. The person is eating the egg and passing the egg. Okay, the person is eating the egg. All the conversions taking place in human and passing the egg. Only one host, human. No intermediate host. No intermediate host. Only one host, human. No intermediate host. So that is a life cycle you have to draw like this. Okay. The name of the disease is Hymenopthiasis, like Tiniasis, Dactylobrythiasis, Hymenopthiasis. So all these are simple diarrhea. No treatment required. Okay. Lab diagnosis is standard. You will get the egg, and treatment is drug of choice for all of them is classical people. We are done with 4 cystodes with perfection. Appreciate the adult of all 4. Appreciate the egg of all 4. Appreciate the adult of all 4 and appreciate the egg of all 4. See Saginata solium, here 4 sucker, no rostulum, no hooklet. Okay, here 4 sucker, rostulum present, hooklet present. Okay, here no sucker, 2 brothia, 1 anterior, 1 posterior, no rostulum, no hooklet. And here again 4 sucker, no rostulum, no hooklet. So head of this is like this. Okay, so please appreciate. Learn the sucker, rostulum, hooklet, brothia. See the egg. B positive, F negative, B positive, F negative, B positive, F negative, B negative, F positive. Appreciate yellow, 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 transparent. Appreciate hexaconch embryo, appreciate hexaconch embryo, appreciate hexaconch embryo. Here also, but it's transparent. It's transparent along with polar filaments and yolk granules, which is additional, not there. And here there is an operculum and a knob. Got it? So please, we are done. We are done with the cystodes. Can I start trematodes? Can we start trematodes? Only 2 trematodes are there. So let's start trematodes. So I'm teaching you these trematodes, which are leaf shape. Give me 1 minute. I would like to drink water. Okay. So we will start the trematodes. Only 2 trematodes are there. Okay. After that, the last 5 nematodes. So let's finish these 2 trematodes. Okay. It will take another 15 minutes. After that, I think the 5 nematodes, it will take another half an hour or 40 minutes. That I will upload the recording tomorrow along with the CVSK parasites. So CVSK parasites along with the nematodes of GIT that I will upload. Like we will complete trematode today only. It will take another 15 minutes or maximum 20 minutes. So kindly cooperate. Just give me 1 minute. We will quickly finish. So let's come on the trematodes. Cystodes, we have seen 4 cystodes. Quickly, we will come on trematodes. In trematodes, we have 3 trematodes. So we are covering 3. These 2 we will take together. Okay, so 4 we have taken there, 3 we will take together here. So Cystosoma mansoni, Cystosoma japonica, and Fistula busky. These 2 we will take together because everything is same. That's why we take together, but we will count them as 3 out of 14. 4 are done, 3 more are done. So 7 will be done. Okay, so chalo. So let's start the trematodes. Let's start Cystosoma mansoni and japonicum. These are blood flukes. These are blood flukes. You can see in the classification. This is the trematodes. I guess you know the classification. This is cystosoma. Cystosoma is coming in the blood fluke. Mansoni and japonicum. In which blood vessel they remain? First you tell me they remain in artery or vein. They remain in vein. The blood flukes remain in vein, not in artery, because veins have the sluggish blood flow. Artery have fast blood flow. Okay, in which vein they remain? Hematobium. I'm not teaching you hematobium right now. Hematobium remain in the bladder ka vein. Okay, and Mansoni and japonicum remain in the intestine ka vein. The intestine vein is known as mesenteric vein. So same thing is written. All 3 remain in the veins. They remain in the veins, in the blood, in the veins. Okay, so hematobium remains in the bladder ka veins. These 2, japonicum and mansoni, remains in the GIT ka veins. That is mesenteric veins. Okay, so that is regarding the introduction. Now coming on morphology. So I told you all the trematodes are leaf shaped except cystosoma. Cystosoma are not leaf shaped. So these are coming in the exception. These are leaf shape only. I will tell you this is leaf shape, but this is not leaf shape. So cystosoma are not leaf shape. You can see they are not leaf shape and they are not even monoecious. The male is separate, the female is separate. In this diagram, male, female both are shown. The fat, the thicker one is the male. This one is male. And the lean thin is the female. So you can see the lean thin is the female. Okay. Now there is a unique feature. Inside the male body, there is a sex canal, which is known as gynecoporic canal. Sex canal or gynecoid. So for performing sexual intercourse, the female enters in the gynecoporic canal, performs the sexual intercourse and come back. So gynecophoric canal is present in the male, in which female enter. So you can see this diagram, female have already entered inside the male body. So very unique feature. All three cystosoma, they are dioecious. The sexes are separate. They are not leaf shaped. Okay, and inside the male body there is a gynecophoric canal. What about the egg? Yellow egg. B positive, F negative. B positive, F negative. Both the eggs are yellow. Okay, so how to differentiate the egg of Manzoni from Japonicum? Manzoni have a lateral spine. Lateral. Side में lateral spine Manzoni. You can learn the mnemonic Tedi Mansi. Tedi Mansi. Okay, lateral spine. And Japonicum have a knob. Japonicum have a knob. No spine, a knob. So there is a knob in Japan. Make a mnemonic. So you can see Manzoni have lateral spine and Japonicum have a knob. So that is adult, egg, larva. Larva have three stages over here. Mycelium, sporocyst and cercaria. Yesterday, I taught you Paragonimus westermani, which was also a fluke. So there were 5 stages in there. Okay, here larva have 3 stages. Mycelium, sporocyst, cercaria. You can learn the mnemonic MSc. MSc is the degree, na? Master in Science, MSc, BSc, MSc. You can learn the mnemonic. Okay, L1, L2, L3. 3 stages of larva. So see the adult, see the egg, see the larva. See the adult, male, female. Okay, see the egg. Don't see the egg of Hematobium. See these 2 eggs. This is Tedi Mansi and this is Japonicum have a knob. And see the larva. So morpho is done. Mode of transmission is very unique. Mode of transmission. So person is swimming in the water. When the person is swimming in the water, the infective form is Cercaria larva. Cercaria larva make a hole in the skin and penetrate directly inside. It's not ingestion. No. It's not inhalation. It's skin penetration. Cercaria larva secrete enzyme, make a hole and go inside. So skin penetration on contact with water. It's not swimming. Skin penetration. Whatever part of the body is in contact with contaminated water, that can get infected. So the infective form is Cercaria larva. This Cercaria larva is present in the water. You can see this Cercaria larva is the infective form. It is present in the water. Okay, so the infective form is Cercaria larva. Okay, now let me tell you the life cycle. Here I will draw it. You can see I have already drawn it for you. You can see in the life cycle, the Cercaria larva I have drawn with red color. Okay, it will penetrate, make a hole. It is present in the water. It is making a hole. It is secreting enzyme and enter in the vein. Okay, enter. We have veins everywhere. So whatever portion, it entered that vein. It entered that vein. Whatever portion in water. In that vein, it will go to portal vein. It will go to portal vein. From the portal vein, it go to the liver. From the portal vein, it go to the liver. In liver, the Cercaria larva grows into adult. This is L3 now. L3 will grow to adult. In the liver, it grow to adult. From liver, it will go to the veins of intestine. See, this is intestine. Okay, this is the vein of intestine. In the vein of intestine, they will fertilize. Male, female. So male is also growing, female is also growing. The couple is going in the veins of the intestine and they fertilize. Gynecophoric canal, female enter inside male. The female become gravid. She become pregnant after fertilization. After that, she lay egg. The eggs are laid in the intestine. Okay, they are fertilizing in the veins of intestine, but the eggs are laid in the intestine and eggs are passed in the feces. In the feces, feces will go in water. Okay, in the water, the egg will hatch. So L1 will form. This L1 is ingested by snail. Inside the snail body, L1 convert to L2. That is Mycelium convert into sporocyst. Okay, and sporocyst is released out. And then sporocyst convert into cercaria, the same cercaria. Okay, if you don't want to learn it, it's your choice. But sometimes it is asked in your exams. Okay, so man is the definitive host, intermediate host is snail. Okay, so this is the complete life cycle. You have to write like this in your exam. This is the complete life cycle. Okay, the name of the disease it is causing is schistosomiasis, like teniasis, hymenoptosiasis, diphilobrythiasis. This is schistosomiasis, in which the person have dermatitis. You can see small holes are there from which the Cercaria larva enters. Number 1, Cercaria dermatitis. There is acute schistosomiasis and chronic schistosomiasis. Acute schistosomiasis also known as Katayama fever. Patient have fever, and chronic में multiple granulomas are formed. And apart from that, it is the oncogenic parasite. I taught you oncogenic virus also. I taught you oncogenic bacteria today, H. pylori. And this is an oncogenic parasite. It is colorectal carcinoma, but it is caused only by Japonicum, not by Manzoni. Lab diagnosis, the same standard. Okay, you will get the X, either lateral spine or Japonicum. Ke. Drug of choice is prosequentin. So we are done with cystosoma. The last parasite is Fasciola busci. So can we start? Fasciola busci. Okay, the last parasite we are starting, another 5 minutes, Fasciola busci. So Fasciola busci, learn the other name. In the introduction, always write down the other name. The other name of Fasciola busci is giant intestinal fluke. These were blood fluke, these are giant intestinal fluke. Okay, again adult egg larva. Adult is leaf sheath, standard leaf sheath, monoecious, male female is the same organ. Okay, and unsegmented. It is having two suckers. You can see the two suckers. Okay, eggs are yellow. B positive, F negative. All eggs are yellow except Neha. It is not coming in Neha. So B positive, F negative, and it is having an operculum. It is having an operculum like D lata. Yes, it is having an operculum like the D lata. So it is having a gap. Okay, learn the unique feature. Learn B, learn F, learn unique feature. My master charts people, I have given months to create it. You know, in a standard format. All X ka B status, F status, unique feature with the diagram. Very easy to remember. Very easy. Okay. Larva. Here we have 5 stages. In cystosoma, we have 3 stages. What were the 3 stages? M, S, C. MSc, BSc, MSc. Now one before C and one after C. So total 5 stages. Before C we have radia, and after C this is Cercaria, this is Metacercaria. Cercaria, this is Cercaria ka bada bhai, Metacercaria. So total 5 stages. The same 5 stages I told you yesterday in Paragonimus Vesternary. I asked you to make a mnemonic from the chat GPT. Someone of you have made the mnemonic also. This is the same. MSRCM. You can learn. Okay. So this is the 5 stages of the larva we are having. So see the adult. See, this is cystosoma, this is fasciola. Appreciate the adult. These are dioecious, these are monoecious. Here male female separate, here male female same organ. This one is the tube shaped, this is leaf shaped. Okay, here gynecophoric canal present in the male, here no gynecophoric canal. See the egg. All 3 egg are B positive, F negative. But here the unique feature, here the unique feature is lateral spine in Mansoni, knob in Japonicum, and here the unique feature is operculum, topi, operculum. All are B positive, F negative, but see the unique feature. Here we have 3 stages of larva, and here we have 5 stages of larva. Here additional one above, one below, Cercaria. Okay, radia and Metacercaria. That is the additional. So compare the morpho. See the adult, see the egg, see the larva. Mode of transmission by eating aquatic plants. Some people make vegetables of aquatic plants, or they eat it, water plants, aquatic plants, by eating contaminated aquatic plants. Okay, what among them, among these 5 is present? Metacercaria larva. So it is the Metacercaria larva which is present on the leaves of the aquatic plant, and the person is eating that. It is the Metacercaria larva which is present on the leaves of the aquatic plant, and person is eating it. So Metacercaria larva, not Cercaria. There the infective form was Cercaria in cystosoma. Here it is Metacercaria. Okay, now the life cycle, don't go in the life cycle. I have not put the image, but the life cycle, let me see. Yeah, this is the life cycle. The person is eating the Metacercaria larva. Now you can make of your own. Now till now you got accustomed how to make the life cycle. So person is eating the aquatic plants containing Metacercaria larva. It is L5. So this L5 will go inside. L5 will convert into adult. Adult will fertilize. Adult will lay the egg. Egg will pass in feces. Feces will come in water. Eggs always hatch in water. Egg will hatch in water, and L1 will form. L1 is taken by the snail. In the snail, L1 to L2, L2 to L3, L3 to L4, L4 to L5 conversion takes place. And L5 again deposit over here. So same life cycle is written. I hope in short I told you. So the name of the disease is fasciolopsis. The same, hymenolopsis, schistosomiasis. So here fasciolopsis. Patient have intestinal ulcer, dysentery, and malabsorption. So that is the clinical feature. Okay. Lab diagnosis, standard. Drug of choice is standard. Praziquantel. Drug of choice of all of them is praziquantel. All of them praziquantel. So we are done with trematodes.